I'm very happy to chair this first session of the second day, which is devoted to bioinformatics and imaging. And we may start right away with the first keynote address of the day, which is entitled Biomedical Informatics, Application in Neurodegenerative Diseases, and is addressed by Professor Exarchos, who is with the Department of Informatics at the Ionian University in Greece. Professor Exarchos, the floor is yours. Thank you very much.
Thank you very much, Professor Myronas. I will present you, the title of my presentation is Biomedical Informatics Applications in Neurodegenerative Diseases. And the idea is to present a kind of sort of a review in different terms relating biomedical informatics, treatment, diagnosis, management of neurodegenerative diseases, decision support systems, precision medicine, and how all these are tied together towards the respective outcomes in neurodegenerative diseases. So, neurodegenerative diseases is an umbrella term that is used to address several conditions that affect the neurons in the human brain that cause problems with movement. For example, the two most common NDDs are Parkinson's disease and Alzheimer's disease. Besides those two, there are many others, but these are the most common ones affecting most of the people. In the case of neurodegenerative diseases, there are effective therapies still remain elusive, leading to a need for an interdisciplinary perspective on data and information management in order to improve healthcare and quality of life. So as most of you know, currently there are no therapies that can cure, for example, Alzheimer's disease. So the focus is on other perspectives to ameliorate patients' lives. Clinical and biomedical data are now widely used to advance the existing medical practices and scientific research, having a threefold approach. First, to derive novel insights for the disease. Second, to generate new knowledge. And third, to facilitate potential treatment discovery and targets for treatment. Although neurodegenerative diseases there are not yet therapeutic agents in order to slow down or reverse the underlying pathology, there are several things from the biomedical informatics point of view that currently take place. For example, patient stratification based on population screening in order to develop tools capable of deriving individualized risk profiles. Second is to provide more accurate and more specific diagnosis based on clinical evaluation and laboratory results. Third one, the positive disease identification prior to the observable symptomatology, pathologic or genetic changes in the brain, which may occur years before symptoms. This is one of the most important aspects because currently there are several research attempts that try to identify disease prior to the existing phenotype, prior to the identification of symptoms. And the fourth one is predictive disease course algorithms using biomarkers in order to provide symptoms prognosis. So another useful task is to predict the course of a patient that has the disease in order to predict what would be the future symptoms and provide different medication, different therapeutic approaches to alleviate them. Since in neurodegenerative diseases, patient-specific factors can influence disease progression, being able to identify such factors associated with pathologic transitions can drastically improve this overall patient management. So here you see an overall overview of the different aspects when dealing with biomedical informatics in neurodegenerative diseases and other diseases, of course. So on the top, we have the individual data, the patient-specific data that can be used by the systems, by the clinicians for those four terms described above. So we have different data like demographics, like medical history, like cognitive and behavioral phenotypes, sensors and wearables, and we will see about that at the end. Some other lifestyle factors biomarkers, imaging data, and genetics. On the bottom, we have the population data that are collected and used to identify the knowledge that needs to match the individual data. So we have exposomic data, we have genomic data from large populations that we can identify, for example, single nucleotide polymorphisms. We have other omics data, for example, recently identified the metagenomics. We have fluid biomarkers from blood, from saliva. We have brain imaging, we have cognitive and behavioral data, and sensors and wearables. In the middle, we have the precision diagnostics and precision management tools supported by clinical decision supports towards providing the clinical data outcomes. And for the purely informatics point of view, we have data repositories, we have translational bioinformatics, and we have the data warehouses. So starting with the data, the individual and population data, and biomarkers first, the most common biomarkers in neurodegenerative diseases currently used are brain and body fluid analysis. For example, the most informative data that can be provided for a patient is the cerebrospinal fluid. Moreover, there are examinations coming from blood or saliva. Other types of biomarkers are from brain imaging, either structural and functional, so both magnetic resonance or functional magnetic resonance. We have genomics data, plus other omics. And we have, of course, cognitive and behavioral phenotypes, like clinical evaluation, lifestyle, etc. From all those four, and the other that will be discussed now, the most difficult to obtain, not in terms of obtaining, but in terms of evaluating, is the last one, the cognitive and behavioral phenotypes, because their identification and their evaluation is purely subjective. So the idea is to collect cognitive and behavioral phenotypes, but evaluate them as objectively as possible. Those data and biomarkers are complemented using demographics, where the patient stays, what's his age, the gender, etc., detailed medical history, and wearable and sensors for continuous monitoring. Starting with the fluid biomarkers, the cerebrospinal fluid has been a prominent target for the discovery of potential biomarkers in neurodegenerative diseases, as it can provide molecular insights into the pathologic process within the brain. For example, amyloid B42 and tau are the two of the most validated biomarkers in Alzheimer's disease that can demonstrate good sensitivity to disease pathology and are widely used in clinical practice and research. However, those have some kind of limited specificity that has mitigated their utility as standalone diagnostics, and coupled with their invasive nature, the efforts have shifted on the identification of biomarkers in peripheral fluids such as saliva or blood. So the collection of cerebrospinal fluid from a patient is a rather difficult and painful procedure for the patient. And the idea is to find surrogate biomarkers that can show, for example, the concentration of amyloid or of the protein tau in blood or in saliva, which of course is very, very difficult. Going now to imaging biomarkers, structural and functional brain imaging is widely used to extract some radiomic biomarkers in entities. MRI is used to measure regional and whole brain atrophy in order to inform differential diagnosis. For example, there are features like medial temporal structures in Alzheimer's disease or atrophy that can be shown in a 3D MRI in dementia, and several also molecular imaging techniques that have been validated for detecting Alzheimer's disease pathology, and specifically the B amyloid, including the Pittsburgh compound B, the fluorine 18 labeled radio traces such as fluorbetapir, fluorbetaben, and fluometamol, and cerebral glucose metabolism in combination with the positron emission tomography, the PET. Imaging biomarkers have an advantage and a disadvantage. The advantage is that it's non-invasive, so you can easily have magnetic resonance imaging or a PET study. so it's easier for the patient. The disadvantage is that they cannot be used. They can only be used for diagnostics. So when the disease has been established in the patient's brain, for example, the imaging can be used to identify the disease. It cannot be used for predicting what will be the next steps in the course of the disease. And you cannot identify, and you cannot predict if the patient will progress from normal cognition, for example, to a mild cognitive impairment situation. Going now to genomic biomarkers. Genomic information is commonly used to assist clinicians and researchers in guiding the differential diagnosis and in identifying at-risk individuals long before the symptoms emerge. Specific genes have been associated with early and late onset of Alzheimer's disease, while there are genetic markers that have been associated with the development of idiopathic Parkinson's disease, dementia with Lewy bodies, and frontotemporal dementia. It is very important in an early age to identify if a specific individual will present early or late Alzheimer's disease, because early Alzheimer's disease is much worse than late. So having specific genes which have been found to identify this situation is something very useful. And the common theme across neurodegenerative diseases is that there is a considerable heterogeneity in the possible genetic determinants, most of which are polygenic disorders, and there is a growing evidence of genetic overlap between those diseases. So as mentioned in the beginning, Alzheimer's and in general neurodegenerative diseases is not one disease, are several diseases. Going now to the overview, we will discuss about the biomedical informatics part, the repositories, the translational informatics, and the data warehouses, and how this feed the overall process. So biomedical informatics is a multidisciplinary field that falls at the intersection of IT, computer and data science, engineering, and healthcare. Biomedical informatics aim at the development of technology-driven resources for storage, access, ease of use, and analysis of medical data to derive information and generate new knowledge. As the volume of the medical data and genomics and everything grows, the knowledge extraction is based on detecting those deeply embedded patterns within the data and can translate them in a manner that can provide the patient-specific outcomes. So in biomedical informatics, we focus on two different types of analysis and two application areas: the translational bioinformatics, or translational biomedical informatics, and the clinical research biomedical informatics. Translational bioinformatics are those methods that are used for storing, for analyzing, and interpreting data to transform high volumes of them into proactive, predictive, preventive, and participatory healthcare. Given these increasing rates at which biomedical data are now being collected, and are of course cheaper, and their complexity, the extraction of insights that can be used to inform clinical practice is a big challenge, requiring substantial, of course, computing power. And the notion of translational biomedical informatics focuses on moving research evidence from bench to bedside, which means from the laboratory, and what is laboratory in the informatics science is actually an in silico experiment in a computer, to bedside, meaning to help directly the patient in order to support evidence-based practice with data-driven analysis. On the other hand, clinical research informatics is defined as the use of informatics in the discovery and management of new knowledge related to healthcare and diseases, focusing first and most importantly on clinical trials, and secondly on clinical data aggregation from everyday practice. By incorporating complex and unstructured clinical data into highly controlled and well-characterized data from clinical trials is a really demanding task called harmonization. Several procedures need to take place in order to achieve this harmonization and be able to use and incorporate those complex and unstructured data. The future utilization of the collected data is equally important, as the data reuse of data is in future applications, besides the ongoing research purpose, is a key factor. And of course, it's a matter of losing time and money if data are collected and that at a certain point they are erased. So the idea is that all those data are collected and kept to help future researchers, future clinical trials, to have some deeper knowledge. Clinical research informatics aim to generate complementary practice-based evidence and can serve as the foundation of the learning healthcare system. Then going to the middle part, the precision diagnostics and precision management. Precision diagnostics refers both to diagnosis and prognosis, while precision management refers to all those processes that can be used to manage a patient, including medication, including sleep consultation, diet consultation, exercise, rehabilitation, ADL assistance, genetic testing, counseling, and health education. All those defining the clinical data outcomes that we want to achieve. So the overall notion is about precision medicine, which provides a disease management plan that takes into account individual variability, for example, genes, environmental factors, lifestyle, but also other magnetic resonance imaging, etc., towards linking individual with the best possible treatment to maximize the clinical outcomes. The effective implementation of personalized medicine into clinical practice requires, of course, the integration of translational research from a diverse array of data sources in order to ensure that the personalized medicine approach is firmly rooted in strong data-oriented evidence. The key in advancing precision diagnostics and management is how to effectively integrate patients' individual biomedical data with general population biomedical data. Of course, due to the incredible diversity in disease manifestations in several neurodegenerative diseases, NDDs cannot be reduced to a single determinant, and attention has been shifted to integrating those multidisciplinary data. Neurodegenerative diseases present a great application of precision medicine interventions, and following the advances of big data and computational power, we are now able to acquire with relatively easily genetic information, concurrent growth in biomarker discovery, and develop systems that can better characterize cognitive and behavioral phenotypes. Another major challenge is that those multiparametric assessments can generate databases of incredibly complex big data, requiring, of course, computing power and infrastructure to be processed. And when studied at an individual level, this data can be manually integrated with relatively easily, but elucidating patterns within even the modest sample sizes can quickly exceed what is feasible with manually guided approaches. The efficient and effective separation of the signal from the noise in such datasets requires dedicated computing power with purpose-built biomedical informatics applications in order to analyze the data and derive novel insights, especially critical for personalized medicine. So the overall idea when having data is not only the amount, it's also the quality, and there are several research attempts trying to fix the medical quality by using several automated algorithms, and at the end be able to remove the noise, the incomplete data from the data that will be used further on for precision medicine. Then going to the most important part of the overview, the clinical decision support tools, which will combine the upper and the second level for providing the outcomes. These are tools built to deliver filtered information to providers at the point of care in order to enhance the patient management and healthcare for the patient. So given a specific problem, a clinical decision support system can be developed in order to guide clinicians in their clinical decision-making in different manners. First, by merging empirical evidence, medical knowledge, and experts human reasoning into an immediately usable format of rules and conditions. In that case, there are no previous data required. And secondly, by analyzing medical data using techniques like data mining, like machine learning, like artificial intelligence, in order to indirectly extract meaningful information that can be translated to rules and conditions. In the second case, no human knowledge is required. Of course, there is a third case not mentioned here, which is the combination of the first and the second. We mean the optimal, let's say, way to define a clinical decision support system is by combining both empirical evidence and automated knowledge extracted using ML techniques, machine learning techniques, in a combined schema that can address both the advantages and the disadvantages of the previous approaches. So the first approach has the disadvantage that is difficult to acquire empirical evidence and formulate it in a decision support system, but has the advantage that there are no data required. The second approach has the advantage that automated knowledge that is extracted can be used for DSS, but has the disadvantage that there are several big data need to be collected in order to be able to analyze them. And of course, we must say that a decision support system is not something to replace the clinician. It's not a replacement for clinical judgment, but rather is an additional source of information that can be integrated within the clinical judgment in an effort to transfer medical knowledge to non-expert physicians. So the commercial and currently working decision support systems come with very big disclaimers that show that this is a decision support tool and not a decision-making tool that can substitute the clinical diagnosis or prognosis or the clinical consultation. When we have patient-specific parameters, genetics, of course, and others, and if these are integrated into a clinical decision support system, the DSS can provide more personalized recommendations, adjusting the best evidence-based practices accordingly for the specific patient. The resulting models can be used to generate an empirically derived probability of the underlying pathology based solely on the clinical presentation, having as an outcome significant utility for both clinical trial recruitment and clinical data management. For example, a decision support system that can be used to predict the evolution of cognitively normal individuals into amyloid positive states makes them eligible for clinical trial participation and preventative treatment. In neurodegenerative diseases, especially where individual variations can influence this pathologic transition between symptoms, findings, and disease burdens, precision medicine and personalized decision support systems can increase the opportunities of a successful treatment intervention. Finally, for this, with the advent of the Internet of Things and technology of high throughput streams of user-generated data, for example, the wearables, these have also emerged, providing behavioral data in real-world settings that are being gradually integrated with clinical data sources and medical records, providing additional information useful for the clinicians. Machine learning algorithms are then used to automatically monitor those data streams and isolate patterns that can suggest an emerging transition, an alarm from a healthy to disease state, raising a flag both before the clinical symptoms emerge, both to the patient, to the individual that can become a patient, or to the clinician, or to the caregiver. There are also decision support systems with natural language processing capabilities that are deployed into electronic medical records in order to analyze clinical notes autonomously in the background by providing updated suggestions in real time as this new information is generated. So going now to some novel trends, is that we will see the digital biomarkers. So digital biomarkers is actually everything that comes from digital media. And you see here in the screen what this could be, from the microphone, from the touch screen, from the camera, all the different wearable sensors plus the sensors existing in the mobile phone, accelerometer, gyroscope, etc. Here is a list of what can be available from camera, for example, the saccades, novelty preferences. And interesting is the device used at the end of the first column, like the PIN and the passcode attempts, which shows the executive function and the memory. It assesses the executive function and memory. I won't stay a lot here. And based on these digital biomarkers, and a very interesting and recent work is from the company Altoida, where they have developed augmented reality biomarkers to measure cognitive outcomes. There are 3 easy to perform, to use games, hide and seek, fire evaluation drill. The hide and seek assesses the spatial memory. The fire evacuation drill assesses the prospective memory. And finally, there is a dual task test in order to assess the psychomotor processing speed. Altoida has gained the 8th Health Award in 2018, a digital biomarker for real-world evidence in Alzheimer's disease, which follows up also the currently working clinical study. What the claim is that qualify the cognitive assignment algorithm composed of Altoida's neuromotor index and age for prognosis of the risk of developing MCI due to AD within 5 years in cognitively normal individuals. Another novel trend is to combine in the network-based biomarkers from molecular omics level and brain connectivity level. So you see on the left of the screen, we have molecular omics deriving at the end differentially expressed sub-pathway biomarkers, and neuroimaging data deriving discriminative subnetwork biomarkers combined using support vector machines and leading to increased capabilities of prognosis and diagnosis. Finally, a landmark paper from presented in the Brain Journal, performed logistic regression using several different biomarkers like ApoE, like cognitive questionnaires, magnetic resonance imaging, CSF tau protein, and they developed models for predicting progression from normal cognition to mild cognitive impairment for individuals at 5, 7, and 10 years. And we can see on the left the data that have been used. For example, ApoE4 on its own was able to predict progression from normal cognition to mild cognitive impairment at 5 years using a sensitivity of 62.9. And at the end, here we have the ApoE4, the cognitive domain, the magnetic resonance imaging, and the CSF tau protein, leading to an overall model sensitivity after 5 years of almost 80%. And at this stage, I would like to finish my presentation. I hope I was on time.
Thank you, Professor Xechos, for the great keynote talk and for introducing us to these very interesting facts and elements of your recent research. Any questions from the audience?
If not, I would like to make a question to Themis.
Of course, Professor Hadjinikolaou.
Yes. Themis, would you like to comment on the difficulties that appear in reuse of data? You mentioned in your talk, but what about the open data movement and all this? Are there any prospects on building something that could be elaborate or could assist people using data? Do you know?
You mean the digital biomarkers that are presented at the end?
Yeah, that's it.
This is the prospect of the future. To be able to, this data are easily collected from smartwatches, from the mobile phone, from very cheap and easily acquired sensors. The difficult thing in this is how to objectively quantify them. So how can I objectively quantify that, for example, such a movement at the hand is a biomarker for progression of the disease of Parkinson, for example. So this is this science goes to be able to collect digital biomarkers and objectively quantify them and assess them.
Okay, thanks. Thank you.
See another
we have
screen
also
here.
Sorry, yes, of course. We have an audience.
Yes, please.
Which is, I will read it out loud. Can you please provide an example of DSS with natural language by Dionysios Hirdaris?
Yes. A DSS that incorporates natural language processing techniques is actually a DSS that, of course, combines several heterogeneous data, like those presented, genomics, imaging, etc. But there are cases where there are clinical notes written on paper, for example, for a specific patient, and natural language processing methods are used to extract information on those clinical notes. Not only that, even if not on paper, but the clinician writes something in the health record of the patient on text. Of course, the automated analysis techniques cannot be able to directly use this text. So we need natural language processing and text mining techniques in order to create features from this text, able to use them further on to the DSS procedure.
Okay, thank you very much once again for this very interesting first keynote today. I will move forward the session to the second presentation, to the second keynote address, which is entitled A Multi-Scale Mathematical Model for Tumor Growth, Incorporating the GLUT1 Expression, which is going to be given by Professor Maria Hadjinicolaou, who is a professor of applied mathematics at the School of Science and Technology by the Hellenic Open University. Professor Hadjinicolaou, the floor is yours. Thank you very much.
Thank you very much. I would like to say that I'm very happy to participate again in GeNeDis conference, the fourth in line, which held this time online, which must say it's a different experience. I would like to thank the organizers, and particularly Professor GeNeDis and my dear colleague Professor Vlamus for the invitation. And I take now the opportunity to inform you that much of the newly obtained knowledge presented in this conference will be transferred to our students in the master program Bioinformatics and Neuroinformatics that the Hellenic Open University offers jointly with the Ionian University. The chairman and the previous talker, Themis Exarchos, are part of this. And I thank them also for the collaboration towards this direction. Well, the subject of my talk is a work in collaboration with Dr. Pantelis Avadzoglou and Professor Fotini Kariortou, held in Applied Mathematics Laboratory of School of Science and Technology at Hellenic Open University. I'll give you an overview. We propose a multi-scale mathematical model for the avascular tumor growth that considers different scales, tissue and cellular scale, which at the cellular scale, the model takes into account the biochemical environment, the different phases of the tumor cell cycle, the cell signaling, and cellular mechanics. We adopt a bioenergetics approach, which means that we take into account the metabolic processes that take place within tumor cells, and we model the interplay of energy in these cellular biochemical processes. How do we do this? We employ a mathematical function, the so-called health function, through which we express the cell's biochemical energy in the different regions of the tumor. We also encounter in the model the glucose transporter GLUT1. GLUT1 is a membrane protein that facilitates the basal uptake of glucose in most of the cell types, ensuring this way the flux of sugar into metabolic pathways. The role that it plays in mitosis is investigated for the different kinds of tumor cell populations, as its overexpression in malignant cells is associated with cancer development. Therefore, GLUT1 is an important target in cancer treatment. Overall, by incorporating biochemical processes in multi-scale modeling of tumor growth, we aim to provide better understanding of the disease and support the assessment of possible targeted therapeutic strategies, such as those targeting GLUT1 activity. The basic assumptions of the model are that we consider the avascular period of a tumor, which means that the period before, we consider the period before the tumor finds access to the surrounding vessels and becomes an invasive carcinoma. And this restricts the size of the tumor to a maximum diameter of 2 millimeters. All tumor cells are characterized from the available amount of chemical energy in the form of the concentration of adenosine triphosphate molecules, ATP, which, according to knowledge, ATP is the cellular energy currency of intracellular energy transfer that is produced by the cells through metabolic pathways. We employ also the so-called health function, which correlates the energy level of each cell to the phase at which it exists. And in this way, health function controls the tumor evolution. This function was first introduced by Abadzoglu et al. And is based on Young et al. Work, and precisely describes the state of tumor cells with respect to the concentration of oxygen, glucose, waste, growth factors, and growth inhibitors. It also carries information for the metabolism rates. And all this also, we translate in the mathematical language the role that GLUT1 protein plays in the transportation of glucose into the cell and the glycolysis. Having all of this in mind and in the model, we are able to produce estimations for the evolution of the tumor cell populations. Here's a small figure that shows the work, the role of GLUT1 in the transportation of glucose molecules in the cell. At tissue level now, we assume that the tumor occupies a space domain omega surrounded by the boundary theta omega between the tumor and the host tissue. The tumor develops in the surround medium occupied by the extracellular matrix, the ECM. ECM serves as a substrate for the diffusion into the cell of all the biochemical species we consider, which are glucose, waste, growth factors, growth inhibitors, and their behavior are modeled through diffusion equations for each species I. Ci as the concentration of the biochemical species I, Di denotes the diffusion coefficient of the species I, while Qij is the production consumption rate of the biochemical species I, but it depends on the phase that the cell exists, J. We assume a Dirichlet boundary condition on the boundary for each of the species concentration, reassuring this way that there is available nutrition and also it remains constant on the boundary. Initial values for the concentrations of the biochemical species are given here, where all the other the values are nil unless the concentration of the glucose, which is this initial value. Values for the production and consumption rates for the biochemical species and also for the diffusion constants are provided by Young et al. And here are some indicative values. Now, at the cellular scale, since nutrition diffuses from the surrounding ECM into the tumor through its boundary, cells in the outer tumor regions have access to an abundance of nutrients, and thus they obtain higher metabolic rates than those existing in the inner areas. Consequently, all tumor cells are categorized in 5 phases. Cells in necrotic phase are completely depleted of ATP and are unable to sustain life. Cells in autophagy phase have access to a very limited amount of nutrients, and thus the autophagy process has begun. Quiescence is a phase where all cell exists at or near energy equilibrium between the available nutrients and the energy requirements for maintaining life. At the proliferation phase, cells enjoy a nutrient-rich environment and strives towards maturation and eventually mitosis. Mitosis, as you all may know, is the phase at which proliferating cell has matured and the mitosis process has begun. The health function now represents, as we said, the overall energy of the cell state of each cell, and takes into account the energy that is produced by the aerobic and the anaerobic glycolysis, and the energy consumed at its cell phase. The health function characterizes this way the tumor regions with respect to energy thresholds. We can see here, for example, QA denotes the HQA denotes the threshold between the quiescence and autophagy phases. We consider that when a tumor is evolving from cancer cells, absorbs glucose from the extracellular matrix, and through the process governed by glucose transport 1, the glucose is penetrates the membrane and enters the cell. Since GLUT1 is limited in supply in each cell, we consider in the model, we use the Briggs-Haldane kinetics in order to describe the glucose transportation from the extracellular environment to the cell. According to Briggs-Haldane kinetics model, the glucose reaches the outer surface of the cell, and it connects to the available GLUT1 molecules at a rate K1. The combined molecule is transported in the cell where glucose is released to be used in the cell's metabolic pathways, and eventually to convert adenosine diphosphate to ATP. There are a series of processes which I didn't show in this slide, but we can say that all these processes, we assume that can be summarized in this form, where G is the concentration of glucose, and Vm represents the maximum reaction velocity achieved by the systems, and Km are kinetics, a constant that have to determined on the basis of the substrate concentrations at which happens the reaction. some indicated values are shown on this slide. The energy level, as you said before, the energy level of each cell appears with the ATP concentration of ATP molecules. And the concentration of ATP molecules is a direct result of the amount of glucose delivered to the cell. The availability of ATP molecules to be converted to ATP and the energy requirements of the cells in its current phase, namely, which is we call here, we depicted here as DS. So the rate of change of concentration of ATP molecules is provided through this first order ODE. The exact solution of which, with respect to time, is given through this equation. And from this equation, we try to make some examples of the evolution of the values of the function ATP under different nutrient conditions of G and different consumption rates for the cells, considering them in different phases. Here you can see some the values that we use for these different phases of the cell cycle we mentioned, and with initial condition of ATP's 0.6. This behavior ATP is embedded in the in energy integrals epsilon 1 and epsilon 2, and determines the values of the health function over time. In this figure, we can see examples of the evolution of the health function values over time under different nutrient conditions and energy consumption rate with respect to the concentrations of ATP molecules given by the previous equation in different phases. Perhaps here I can notice that there are some the thresholds of the energy thresholds that we described previously seems to be in a kind of any sort of way to appear the four thresholds. Now, in this having all this in the model, we are able to make a population estimation. And in this figure 3, in this figure, we present some examples of the evolution of tumor populations from 8 simulations, assuming that at the 50 adjunct cells that form a sphere, each one is having an initial value for the health function 1, and all these are inside a rich biochemical environment. The blue, the green, and the black plot points and the plot show the populations for the proliferating, the quiescent, and the total population respectively. We can see in this figure that the population of proliferating cells increases rapidly and reaches a maximum almost immediately, where then it fluctuates around the central value. In the meantime, the tumor's quiescent cells and the total cell population increase until the extracellular matrix carrying capacity is reached, and then the population remains fluctuating around that value. We also observe that the tumor's regions firstly undergo a very high growth phase, as you mentioned, and this initial growth is a consequence of the high nutrient concentration that suffice for all regions to get high values of the health function, which means that the cells are in a proliferating phase and undergo mitosis very rapidly. Then the population growth slows down and is asymptotically approaches as we said, the carrying capacity of the extracellular matrix. These are the initial conditions assumed before. We start with this value. Now, from all this data we obtained through the model, we try to perform a care fitting to the results obtained from these simulations, employing the minimum mean square error metric, and we find that the tumor's evolution is best described by a von Bertalanffy function given in this equation, this form. So the stimulated tumor behavior follows the tumor population, the stimulated tumor population growth follows a behavior similar to that of analogous models that show exponential population growth for both quiescent and proliferating cell populations. And this growth, the growth asymptotically approaches the assumed carrying capacity. And at that point, the tumor's population reaches a steady state with minor fluctuations. Performing the care fitting, we doing numerical testing, we obtain the value for the exponent beta having this approximate approximately 0.02. And these are the characteristics of the fitting. Roughly saying, this behavior of the results of our simulations. We have here, I present here a cross-section of the tumor simulations, assuming a spherical tumor. And here is the necrotic, the quiescent, and the proliferating zones, phases. You see some quiescent here, small quiescent cells, tumor cells. And here is it's a photograph, it's a picture taken from the International Journal of Cell Biology, which we found some similarities. Of course, there's not a complete identification, but this is something encouraging to show that a model can predict clinical data in a sort of saying. Of course, they are far away from that. Now I'll come to the conclusions. That what we did is that we proposed a mathematical model for predicting the evolution of the avascular and avascular tumor, adapting a bioenergetics approach for describing the cellular cycle of tumor cells, which is based on the chemical energy level that the cells exhibit at each phase. This is a kind of is a newly, I can say, approach since this behavior of the tumor cells and the categorization usually is done by stochastic processes. And now we inserted a detailed equation and mechanism that describes this whole biochemical process. We also model the role of GLUT1 in the transportation of glucose in the cells and the glycolytic generation of ATP needed for the cellular function, which also something new. The new thing is that we integrated this into a multi-scale model. So having done this, we can say we found that the predicted tumor behavior is in line with the existing results. And furthermore, by taking into account particular tumor characteristics, of the model, we provide an analytical function for describing the evolution of tumor cells populations, which is a von Bertalanffy type. And this way, having this analytical expression, we provide the tool for analytical study of the phenomenon. The proposed model is a first step towards the embedding bioenergetics aspects in multi-scale modeling of the tumor growth, as I mentioned earlier, and it focuses to more accurate description of the biological processes of living tumor cells, allowing this way in silico testing of cancer metabolism targeted therapies, perhaps by suppressing the expression of GLUT1 through specialized inhibitors, or by revealing possible unexpected complexities, such as metabolic adaptability, in the development of effective strategies. Here's some indicative bibliography. More have been written down on the paper we submitted. And thank you very much for your attention. And I must say that this work has been partially supported by the special account of the Hellenic Open University and the project, which is called Bioinformatics and Neuroinformatics. Thank you very much.
Thank you very much, Professor Hadjinicolaou, for this very interesting talk and the presentation of this novel idea. Any questions from the audience? Not yet. So I have one personal question, because I thought that this research of yours is very challenging and very interesting. So a more qualitative question would be, what would be the major challenge in combining these two worlds of applying a mathematical model to the bioinformatics part? What do you think would have been the most important challenge that you faced in this process, and how easy is to apply this in a more generic manner with other sub-elements of this work?
The difficulties, till now we have researchers that are completely devoted to the describing the metabolic processes, and others that do the modeling, and they don't go into detail. When do this bioenergetics, the people who are researchers who are working on research on these biochemical processes, they do a very difficult and experimental work, and they produce results that are connected mostly with clinical data. So the difficulty is to use this data and then to integrate into the model, which always I think it's the major issue for all of us, how to use data and integrate it into a numerical model to simulate. So we can start from very special cases, and then integrate them, part of them into the model, and then try to make predictions that will be reconfirmed through the output of the experimental data. So I think this is the process, if I answer.
Mm-hmm. This is very, it's easy to understand your skepticism about this, because I guess it's also subjective, the way that the incorporation happens. It depends up to an extent from the human factor, isn't it?
Yes, you have to take. You need the data from human population if you want to be effective, because most of the experiments are not done to humans. So these reaction rates or metabolism rates are not must be detected, but it's not so hard. So perhaps it can go straight through the personalized medicine instead of having generic models. So I think it's easier to do this, to have the details for a person from each individual, and then try to produce, to incorporate in the model, then to have an first trial results, let's say.
Clear enough. Okay, thank you very much. I think that we do not have another question. So this concludes the second presentation of the day. Thank you, Professor Hadjinikolaou. And we'll shift directly to the third presentation, which is the third keynote address, which is entitled Looking and Listening to Tissue, Advances in Fluorescence and Optoacoustic Bioimaging. This keynote address is presented by Vasilios Giachristos, who is a professor of medicine and electrical engineering, and also a chair of biological imaging at the Technical University of Munich. Professor Giachristos, the floor is yours.
Hello, and welcome to my talk on advances in fluorescence and optoacoustic bioimaging that is done for GeNeDis 2020. I will focus in particular on optoacoustic imaging techniques. It's a new modality, optoacoustics. We are seeing considerable progress in recent years. And although the discussion will be general in the sense what the modality can achieve and where we are with this technique, certainly I am trying throughout the talk to identify what sort of contrast mechanisms we can detect altogether that can also be applied for geriatric research and healthcare, but also in the context of neurodegenerative diseases. So, for example, I will be showing how we can see inflammation or metabolism in tissues in general, but this could be ported in these particular areas as well. I would like to take this opportunity to thank Panagiotis Vlamos and Vaso Michopoulos for the invitation. It's a talk that it was supposed to happen in Crete, and certainly I regret the fact that I cannot be in Crete and meet everybody and interact with the participants of the meeting, but also with the island. But I would like also to thank the people that made this talk possible through this virtual portal, so at least we can go through the science. I would like to disclose financial interest to Search Vision. It's a company that commercializes fluorescence technology, and to Ithera Medical, which is a company that commercializes optoacoustic technology. So I'd like to give a bit of an overview on what it is that we do in my institute. We are concerned with basic research on technology and data processing. We want to link developments on technology with biological systems, cells, organoids, organ-on-a-chip, animals, but also we perform clinical studies ourselves. So we invent and innovate new methods, imaging systems, sensors, and combine this with genomic and general omic information as well. And we convert some of these methods to clinical systems so that we can actually perform measurements from humans, with an emphasis on advancing early detection, because early detection is a very efficient step towards improving treatment, but also for advancing intervention. We have a program on data analytics and quantification algorithms, also under the this evolution now of neural networks and artificial intelligence. Essentially, we want to drive out of these measurements quantitative observations for precision medicine. And we also have a program on developing contrast agents and contrast mechanisms. Today, I will focus more on technology and its application to imaging in clinical environments. In the past decade or so, we have introduced a number of new modalities. Multispectral optoacoustic tomography is a technique that I will focus in my talk. I will also mention briefly developments with fluorescence molecular imaging, but we have also been developing other techniques. For example, the combination of fluorescence tomography and X-ray tomography as a hybrid modality alternative to maybe SPECT-CT or PET-CT. We have been developing near-field thermoacoustic tomography. This is a technique that uses radio frequency excitation, but because of this thermoacoustic technology, we can visualize radio frequency absorption in tissue with resolutions of 100 to 300 microns. We have also developed ionoacoustic tomography. Again, it uses detection with acoustics, and the resolution is similarly at 100 to 300 microns. But in this case, the ultrasound waves are excited by high energy beams, and this proved to be very useful into proton beam therapy communities that they want to characterize beams and localize how a beam or how the energy propagates in tissue. We have scaled multispectral optoacoustic tomography down into this field of mesoscopy, and that is raster scan optoacoustic mesoscopy. I will also show some examples about that. And very recently, we have developed this new microscopy modality called mid-IR optoacoustic microscopy, and I will also give some insights on this technology. Now, coming on the fluorescence molecular imaging, a very quick slide. This has been an earlier development that started many years ago, and the idea here is to inject fluorochromes into humans during surgery or during endoscopy in order to localize disease, cancer in this case. So we had shown early on, 10 years ago or so, that by using a fluorescence label, you can much better visualize tumor spots, tumor foci, as you can see on the image on the bottom right side, versus the typical surgical view that doesn't have this fluorescent signal that is shown there in pseudocreme. This is a technology that started from basic developments of hardware and animal work. It went all the way to the clinics, and it was commercialized by Search Vision and now has been taken up by Bracco, and we hope to have this technology into the clinics soon through this pipeline. It is an example of basic developments in a lab that they hopefully can make it into the clinic in the near future. The other technology that we have placed a lot of attention on is multispectral optoacoustic tomography. This is a technique that illuminates tissue with lights. This is our light pulses, and they excite ultrasound waves inside the tissue. So we capture the ultrasound waves on the surface with an ultrasound detector. Because we see tissue excitation by light using ultrasound, we listen to it, the resolution of the images we form is the ultrasonic resolution. So in a way, optoacoustics is a way to do high-resolution optical imaging much deeper than optical microscopy. I will discuss analytically about the contrast that we're seeing, which starts becoming very interesting. And in particular, I will focus on label-free imaging. So imaging without contrast agents. What it is that we can visualize with this technique if we are not using contrast agents. This is a technology that was also licensed by Ithera Medical in the laboratory, but also from the company. There have been different versions of first animal systems, animal prototypes, and then also systems for human measurements. And as you can see, as the years go by here, the number of channels improve, the speed of the laser improves, the tuning speed improves as well. And we have now handheld optoacoustic systems that work in parallel with ultrasound. We also have received several awards. And they work both in the what we call macroscopic scale, that it goes from 1 to 4 centimeters depth, or the mesoscopic scale that is limited to 1 to 10 millimeters, but with much superior resolution. This technology has been placed around the world in many labs and clinical centers via Ithera Medical. And in the Institute, we have been focusing on identifying clinical applications and how we can drive patient benefit with this technique. We are also doing basic research and animal research, but our primary focus is to understand if we can impact patient and healthcare with this technology. To develop this technology, we have been essentially developing all the individual components. We have been developing a new class of lasers that it can do up to 100 different wavelengths per second. Each pulse is about 100 nanosecond pulse, but it has a high energy, 50 millijoules, which is needed for optoacoustics. It's still safe for human use, but you need this type of energy in order to excite ultrasound in tissue. We have been developing detection channels up to 512 parallel detection of high frequency ultrasound. And this technology has allowed to do single shot frames, which means a single pulse is enough for generating a cross-sectional image of a tissue. We have also been developing the computational tools for this technique. I will not go into the details, but there has been a lot of computational effort and novel modeling and novel algorithms in order to formulate it. The end result is the following. Even 10 years or 15 years ago, optical imaging was this very diffusive image that you see on the top left image. Because of photon scattering, photons diffuse and you lose resolution. Using this technology, using these engineered lasers and new detectors and new tomographic methods, we converted optoacoustic imaging to this very high resolution imaging, cross-sectional imaging that you see on the right side of the top panel here. These are 3 cross-sectional images through the mouse. You see on the top the head and the brain. You see in the middle the spine and the spinal cord and the liver and so on. And then at the bottom you see the kidneys, again the spine and other structures, the spleen. So this is an optical image with a resolution of about 200 to 300 micrometers. And it was the first time that we could take anatomical images of such quality with using light. You can also see at the bottom 3-dimensional imaging, essentially real-time 3-dimensional imaging of a mouse. You see the nice structure of the vessels and also the beating heart. But as I mentioned, our primary drive was to understand if such a technology could be used for patient measurements. So we have been applying this to different regimes, and we have been showing that we can image metabolism with this technology. This has been work where we could see the activation of brown fat after exposure to cold. Independently, other investigators in Erlangen, they got our technology, and they have been publishing high-impact studies visualizing inflammation in the gut of Crohn's disease patients or muscular dystrophy. And when one plays with the frequency and the scale, we can get these very beautiful images. This is now mesoscopy. The bottom image on the right is a cross-section of the human skin in ways that it was never possible to visualize before. You see the epidermis and the capillary loops and the dermal vasculature. We can also see beautifully the angiogenesis in cancers and so on. I could start explaining all of the different applications and all of the different technologies, but for this plenary talk, I thought of presenting it in the sense of breaking barriers. The first barrier we definitely break with optoacoustics is depth. Optoacoustics gives the resolution at much deeper than any other optical technique. And this was evident already in the images I showed. But the second interesting area and barrier that we would like to address is contrast. So I would like to paint a picture of what sort of contrast do we see especially in regard to human measurements using this technology. So I tried to build this table where I have on here contrast that could be achieved when we use a single wavelength, contrast that could be achieved when we combine multiple wavelengths, and contrast that we get when we actually consider dynamic measurements. This is a fast technique, like ultrasound. We can have images up to 100 hertz, as I mentioned, with these lasers. So we can capture dynamic phenomena. And also, as you see here in the column on the left, you have primary features that can be resolved and possibly features that can be resolved after computation. So the first thing that you see if you do an imaging session with optoacoustics and a single wavelength is that you can very nicely visualize blood vessels. So the column on the left is optoacoustic images of a blood vessel that has also been visualized by ultrasound. This is what you see on the right column. Now, for ultrasound to see blood vessels when you don't use contrast agents, in this case, these are Doppler images, and they are shown in color, in red color, superimposed on the ultrasound image. Already, we notice, if you see the row on B, that when the vessel starts getting smaller, MSOT shows a much better appearance of this vessel compared to ultrasound. And as these vessels get even smaller, we showed in this publication in 2016 that ultrasound may fail altogether to see the smaller vessels, but optoacoustics, as you see, for example, on panel C, nicely resolves even much smaller vessels. There are many reasons for this. Vessels contain hemoglobin. Hemoglobin is a primary contrast for optoacoustics, so the signal is very high. But also, optoacoustics generally operate with a much broader bandwidth compared to ultrasound. So it has the ability to see a much broader range of resolutions. So it is clear, and everybody who has performed an optoacoustic experiment can tell that optoacoustics visualize blood vessels very nicely. This is, I showed this image briefly on the generic panel there. It has been now published as well by collaborators at Memorial Sloan Kettering in New York. This is a CT26 cancer, and you nicely see the angiogenesis, the blood vessels forming around this cancer. So it is clear that we can visualize blood vessels. So I have put it here on the table. We can do vasculature imaging, and obviously, since we have this high resolution, we can very nicely calculate vessel density and vessel size spread. So we can essentially understand how many vessels and what is the size of these vessels. So if we now perform computation on this with some sort of image processing tool, we can possibly also see dilation or vessel constriction because we can see the size very nicely. And this essentially can lead into understanding inflammation, either as dilation of vessels or as a total blood volume signal that is calculated by taking the aggregate of all of these vessels. And this is shown in many examples. This is the study I briefly mentioned, where this total blood volume signal corresponded to inflammation in the colon. And this was done in about 130 patients here, where that, what you see here on the top row is ultrasound images, and superimposed in them in color is an optoacoustic signal representative of total blood volume, which essentially means inflammation. And there's a nice correlation between this optoacoustic signal and invasive assessment of these tumors using endoscopy. This ability to visualize inflammation was also shown in a different publication here on, for example, using mesoscopy, so within the few millimeters and higher even resolution, between psoriasis and healthy patients. What you see here on the top, these are ultrasound images. It's very difficult to have a detailed understanding of vasculature here. However, when you switch to optoacoustics, you have, first of all, a very nice picture of vasculature, but a very also striking difference between psoriatic patients and healthy patients. And in this publication, calculations using blood vessels helped to essentially quantify inflammation burden. And here's a little comparison that between ultrasound and optoacoustic, where optoacoustic provides speckle-free morphology, can nicely see the vascular network and characterize inflammation. You can also see melanin because it's also an absorber of light. And I will tell you a little bit more about oxygenation and metabolism. Now, we populated the first column of single wavelength, but of course we can perform imaging at multiple wavelengths. And once we do that, we can start unmixing the contributions of different chromophores. And the most common chromophores in tissue that they essentially can be resolved by optoacoustics is oxyhemoglobin versus deoxyhemoglobin, because they have this very characteristic difference of spectrum in the near infrared that you see here with the blue versus the red line. And this is one of the driving forces of development of optoacoustics, because there's no other modality that can visualize oxy and deoxyhemoglobin. So in that sense, it can visualize tissue oxygenation without using contrast agents. Essentially, optoacoustics use hemoglobin as an internal biomarker and to sense oxygen. And we have shown, and others have shown, that you can quantify oxygenation. Now, it's not as easy as it sounds, because as light propagates deeper in tissue, it gets differentially attenuated depending on the wavelength. So in order to be accurate in quantifying oxygenation, you have to account for this effect. Essentially, it's called spectral coloring. And in this publication in particular, we presented a method called EMSOT, where, as you see here on the graph on the panel G, it can much better quantify the actual state of a dead mouse being, having zero oxygenation versus regular oxygenation or 100% oxygen supply versus a linear fitting algorithm that that shows a trend but is not quantitatively accurate. It's not only oxy and deoxyhemoglobin we can see. As you see in this graph on the top right side, we can also visualize water that has an absorption peak in the near infrared at 960. And I apologize, there's a fat signal here showing in yellow, a lipid signal. This has an absorption peak at 930. So we can also unmix the contributions of fat and water. This is from a breast cancer study. We have a handheld scanner where we scan together with ultrasound the breast. We use 28 wavelengths here. And we can resolve images of oxy and deoxyhemoglobin, of fat and water. So I have added now, in the second column, those signals, oxy and deoxyhemoglobin, and lipid and water content, which can help also to characterize ectopic fat accumulation. Now, if we start calculating based on the signals, if we take the oxy plus the deoxy signal, this is total blood volume, and the change of this over time is perfusion, the rate of total blood volume. If we take the oxy over total blood volume, this is oxygen saturation. And because we have resolution to see individual vessels, we can calculate the oxygenation of different vessels, but overall the oxygenation of tissue as well. And this is a driving reason of developing optoacoustics. It's the only modality that can visualize tissue oxygenation without using contrast agents, essentially using the hemoglobin as an internal sensor of oxygen. We can, in this way, see not only the oxygenation of arteries, this is what the pulse oximeter does, but also the oxygenation of veins, but also generally in tissue, produce images of oxygenation and hypoxia. We're using these signals in different applications. This is just an example again from the breast cancer study. These preliminary results were published already, but this is an ongoing study. This is now from a tumor contained in the breast. You have the ultrasound image. This is about 1 to 2 centimeters deep. And you can see an increased angiogenesis, an increased oxygen signal here, oxygenated hemoglobin signal here, but also alterations in the fat and water signal. Now, as I mentioned, optoacoustic, it's a fast technique. And because we have these very fast lasers, not only we can visualize, in this case, the arterial pulsation in a single wavelength, we can do it in multiple wavelengths. But certainly this is a technique that can capture dynamic phenomena. We have been applying it in different studies of seeing, for example, changes in oxygenation in exercising muscle, or the recovery of blood vessels due to an occlusion. The study that is shown here is because of is due to arterial occlusion. So we can take images of hemoglobin in the column A, of oxygenated hemoglobin in column B, and we can also calculate the total blood volume. And we can find changes and dynamic changes of the response of the vessel. And this has different implications. First of all, by seeing the rate of this change, we can extract parameters about endothelial function and the condition and the elasticity of the vessel. But we can also monitor generally dynamic events. So I have added also now this contrast into the table on the time domain column, where we can see dynamic responses. And we can characterize, as I mentioned, endothelial function, which is the rate of vascular response. But we can also see aerobic metabolism, because since we have the ability to see oxygenation over time, we can take the rate of oxy-to-deoxy change, deoxyhemoglobin change, and that is, of course, oxygen demand. So again, optoacoustic has a unique ability here. Not only is it tissue oxygenation, but it's probably the only modality that can accurately visualize aerobic metabolism. There were optical methods like near-infrared spectroscopy that also attempt to do this, but they do not have the resolution. Here, we can do this with high resolution, and high resolution also means ability to have accurate quantification. We are not affected by scattering here, so the measurements we're getting are much more accurate than an purely optical technique. Again, we're using this signal. I'll give you some examples. We have been monitoring brown fat activation using optoacoustics. Essentially, we have been looking at oxy and deoxyhemoglobin signals as a function of time. These were participants that they were exposed to cold, and we had a baseline measurement, and then we were looking at the activation after the cold exposure. What you see on the right column on the bottom is an area of the brown fat prior to activation and post-activation, and you see this increases signal because of blood influx and elevated oxygen demand. The muscle area that you see at the bottom the signals did not change, and they did not increase post-activation. If you look now at the left panel, at panels E and F, E is a PET image superimposed on MRI. MRI has taken here an oblique image, so it's a very low resolution image. That's the gray signal. And this yellow-red signal is the PET increase. So PET also very nicely capture the activation of the brown fat. But if you look at the MSOT image at F, first of all, what you see is that it's a higher resolution image, and it nicely see the muscle versus the adipose, the fat tissue. And this is the area where it's shown with a white arrow, where we actually collected the prior and post-activation images with the optoacoustics.
To demonstrate that we can carry out such a study in mice, what you see here on the top left panel is an anatomical optoacoustic image. It's 150 nanometers. These brighter areas are the brown fat deposits. We could also see the suture vein. We can resolve signals in arteries of the mouse. So we monitored the activation of oxy and deoxyhemoglobin as a function of time in response to different stimuli. We found, for example, that secretin activates brown fat, and we could visualize and take images of this activation in the brown fat deposits, but we could also see and monitor the oxygenation of the suture vein. So we could get signals that are organ-specific from the brown fat, but also systemic responses by monitoring the signal in the vein or the arteries. And interestingly, of course, the signal was changing in the veins depending on the oxygen demand. So MSOT can be used as a tool to visualize this activation in response to these different stimuli. We also used norepinephrine and in the humans the cold activation protocol. As I mentioned, we can also visualize lipids. So there's a potential here to see lipid distribution and track lipid metabolism. In the study that we performed in human volunteers using the handheld acoustic setup, we took baseline measurements and then administered a high-fat meal and monitored lipid distribution over the next 6 hours, sampling points of about half an hour apart. We could track the hemoglobins, but also the lipids at 930 nanometers. And since we have images, we do not just get a single time point, a single measurement in each time point, but we can see how lipids distribute in veins, arteries, in muscle, in adipose tissue, and so on. The graphs that are shown on the right side of the slide show how the lipid appearance was found in the cephalic vein and the radial artery here. So some hours after the meal, we see this peak in lipids coming into the vascular system. Combined, therefore, we can see not only the hemoglobins, but the lipids. So I have added also the lipid signal on the time domain column of the table, and the compartmental variance, which means that we can see relative dynamics of the signals, either lipids or hemoglobins, in the different compartments of tissues. So here, there is a table that shows what we have seen so far with these studies. They have all been confirmed in humans, but also in animals. At the single wavelength perceived vasculature, and this can associate to inflammation, but also the status of the vascular system from an anatomical standpoint. In the spectral domain, we can resolve oxy and deoxyhemoglobin, lipids, and water, and this can give us images of tissue oxygenation and of total blood volume. And in the time domain, we can see dynamic responses that are associated with endothelial function, with aerobic metabolism, and lipid metabolism. Therefore, this table shows the ability of label-free, but also possible clinical applications. The inflammation signals we have used, as I mentioned, for characterizing different inflammatory diseases. They can also be applied for cardiovascular imaging, peripheral arterial disease, and so on. We can capture the oxygenation that associates with exercise, but also with hypoxia, tissue hypoxia, associated also with oncology. And here we have a unique capacity to see label-free metabolism, but also endothelial function, also associating with cardiovascular diseases, endocrinology, and overall diseases associated with the metabolic syndrome and diabetes.
Okay, at this point, I think we need to have a short interruption to Professor Gianchristos' talk, because we need to connect on time with Canada and Professor Ilias Kotsirias. So this would allow me as a session chair to interrupt the third part, and we shall continue after Professor Kotsirias' presentation. Let me introduce now the fourth keynote address, which is entitled Constraint Satisfaction Problems, and is going to be presented by Professor Ilias Kotsirias, as already mentioned, who is a professor in physics and computer science at Wilfrid Laurier University, Waterloo, Ontario, Canada. So, Professor Kotsirias, the floor is yours. Thank you very much for attending this conference.
Thank you, Phibos. Is the sound okay?
Yes, everything is perfect. Thank you.
Thank you. Thank you for the introduction. And I want to start by thanking Professor Vlamos for the kind invitation to speak here at GeNeDis. I will start by saying that I was delighted to hear in some of the previous talks yesterday by Dr. Pagaria from Siemens, and also at the opening remarks by Deputy Minister Zarifopoulos, remarks on machine learning, AI, and so on, and how these technologies affect, everyday life, but also healthcare and essentially all medical professions. And in terms of AI, we did hear about deep learning and neural networks technologies that are technologies developed in the realm of AI, but widely used or starting to be used in various medical applications. So today I wanted to discuss another AI technology called CSP. CSP, abbreviated as CSP stands for Constraint Satisfaction Problems, abbreviated as CSP. And I want to mention my sources. My sources for this talk essentially come from this book over here on the bottom right. This is a well-known artificial intelligence textbook used in over 1,500 universities and colleges around the world. So let's start with the outline of the talk. I want to say a few things about what CSPs are, these constraint satisfaction problems, and maybe then discuss constraint propagation and inference, and some local search and backtracking search if time allows. So what exactly is a constraint satisfaction problem? Well, one starts with a standard search problem over here, where a state of the problem in the state space is a black box. In other words, we have some data structures to support perhaps a heuristic function, and we have the problem definition, and that's all that we have. In the CSP side of things, in constraint satisfaction problems, a state or a node in the state space of the problem is given by a so-called factored representation, which means that the state is not a black box anymore. Instead, it is a set of variables, and every variable has a value. So this is a pretty simple setup, right? And a problem is solved when all the variables have values that satisfy all of the constraints on the variables. So a state in a CSP formalism is defined by variables x1, x2, xn. Each one of those variables takes values from the corresponding n domains d1, d2, dn, and we found a solution, a goal, if the set of constraints is satisfied for all for an assignment of variables of values to variables. The CSP formalism is a simple example of what we call a formal representation language. And the whole point of this exercise, of this endeavor, if you like, is to allow useful and very efficient general purpose algorithms to solve problems that are formulated as CSPs very, very effectively, and I hope to demonstrate this with some specific examples. So this is the more formal definition, if you like, of CSPs. A CSP consists of three components that we call X, D, and C. X is a set of variables x1, x2, xn. D is a set of domains d1, d2, dn. And C is a set of constraints that specify allowable combinations of values and forbid other combinations. Every domain consists of a set of allowable values, U1, U2, UK, that K, K doesn't have to be the same for all the domains. And every constraint consists of a pair of scope and relationship. Scope is a tuple of variables, so a subset of the variables, let's say x1, x2, and relationship is what defines the values that these variables can take on or not. So there are two ways to represent a relationship: as an explicit set of tuples or as an abstract relation. For example, we can have x1, x2 as the variables. The domains can be A, B, and the constraint C1 can be that, in fact, x1 is different than x2. Okay, so that leaves us with two choices for x1 and x2, A and B, or BA. So this is quite simple definition, right? And in fact, its power and expressive power emanates exactly from the fact that it is so simple. So a constraint satisfaction problem requires some terminology. Once we have defined these three components, okay, X, D, and C, variables, domains, and constraints, then a state in a CSP is defined by an assignment of variables to some or all of the variables x1, x2, xn. A consistent assignment is one that does not violate any of the constraints. And a complete assignment is one where every variable is assigned a value, so all of the n variables have a value. A partial assignment is one in which only some variables are assigned specific values. And we will see later on why partial assignments are also interesting and useful. A solution to a CSP is a consistent and complete assignment. So if we find an assignment of the n variables to some values so that none of the constraints are violated, this is what we call a solution. So this is pretty much the theoretical background, if you like, to start discussing CSPs. And now we're going to illustrate all of this with an example to make it, of course, a lot more specific. And this example is the famous map coloring problem from AI and graph theory, of course. So what we have here is a map of beautiful Australia. You recognize the states, the territories: Western Australia over here, Northern Territory, South Australia, Queensland, New South Wales, Victoria, and Tasmania. Okay, so the map coloring problem is asking to color this map with, let's say, three colors: red, green, and blue, so that none of the adjacent states are colored with the same color. Okay, this is obviously how we color various maps in the literature, and this is the map coloring problem in discrete mathematics, computer science, AI, and graph theory. So the variables here are the abbreviations of these seven states: WA, NT, Q, NSW, V for Victoria, SA, Southern Australia, and Tasmania. The domains D1, D2, D7, they are all equal to the three colors red, green, and blue. Okay, and now the constraints. The constraints, we have to sort of take this sentence here and translate it into a set of constraints. So adjacent regions must have different colors. So we end up with these 9 constraints. So this is C, the set of 9 constraints. Southern Australia must be different. The value of Southern Australia variable must be different than the value of Western Australia, etc., etc. And the last one is, of course, NSW must be different color than Victoria to the south. So what we mean by this constraint here, let's say, let's make it more specific. When we say Western Australia is different than Northern Territory, we mean that the only possibilities for these two variables are to be colored red and green, red and blue, green red, green blue, blue red, and blue green, all of these 6 combinations. Okay, so this is a specific example of a CSP, a very simple one, right? This problem is very easy to formulate as a CSP. The advantage of doing that is that we can then hit it with a CSP solver that we can use as a black box. We don't need to know the internal details of how CSP algorithms work. And then we can try to find solutions. Of course, this is a simple problem, right? But imagine that you have a much bigger map, like the map of the United States or the map of China, where you have roughly 40 to 50 states and territories. Then this problem becomes a lot more difficult, and you really need an algorithm to solve it. Also, this problem, as I was saying, it's very simple to formulate as a CSP. There are other problems where the CSP formulation is not as immediate, right? It's a lot more difficult, but I can assure you that people have been very inventive in figuring out how to formulate a problem as a CSP. So let's proceed to look at a solution. So this is a solution to the CSP of the Australia map coloring problem CSP. It is a complete and consistent assignment. So this is the solution. In fact, Western Australia is red, Northern Territory is green, Queensland is red, NSW green, and so on. Tasmania, well, it doesn't matter what color you use for Tasmania, because you can clearly see that this is sort of disconnected from the mainland, right? So this is one solution. It is complete. All of the variables have values and consistent. It does satisfy all the 9 constraints, okay? And of course, there are many other possible solutions to this problem that we can find either by hand in this case, or by running various other algorithms. So this is a quick intro to CSP plus, this easy example. And I will skip a few things here in the interest of time. So why formulate the problem as a CSP? Well, because CSP solvers have this nice property here. They can quickly eliminate large parts of the search space because they realize that there are no solutions located there. And this process is called pruning. The term for that in AI is pruning. So everything in AI, at least in the sub area of search in AI, has to do with effectively pruning large state spaces. So the main idea is to eliminate large portions of the search space at once by identifying variable and value combinations that violate constraints. As an example, assume that we choose this assignment, Southern Australia is colored blue, then we know that none of the 5 neighbors can take on this value, the blue value. And this idea is called constraint propagation. So several CSP solvers work by conducting constraint propagation systematically. And why do we do that? Well, because for these 5 neighbors, for instance, in this case, we would have 3 to the 5 assignments, but when we do constraint propagation, we remove the blue value from the domains of these 5 neighbors. So we have 2 to the 5th, so 32 assignments, which is an 87% reduction. So this is a significant achievement, and this is just with a very small step on constraint propagation step. Right, so in the usual state space search, okay, what we have is we answer the question, is a specific state a goal state? In other words, did we find the solution? And the answer is yes or no. In the CSP search, we have a different way of thinking. If a partial assignment, in other words, an assignment of some variables to values, violates constraints, then we can immediately discard further refinements of this assignment because we know that it will not lead to a solution. Okay? Moreover, we can see which variables violate a constraint and which one or more constraints, and we can design heuristics accordingly. I will skip a few things. My apologies, but my overheads are available and I'm happy to share them with whoever is interested. So what kind of constraints do we have? Well, we have unary constraints, and these are constraints that involve a single variable. For example, we can have a constraint, Southern Australia different than green, okay? We can have binary constraints. These involve two variables exclusively. For example, Southern Australia different than Western Australia. The values must be different colors. And notice that in the Australia CSP, all of the constraints were binary, and this property is really nice, and it has a name that you can fathom what it is, binary CSP, right? So a binary CSP is a CSP with only binary constraints. Why is this a good property to have? Because many CSP algorithms described in this book that I told you in the beginning work directly on binary CSPs exclusively. Of course, we can have higher order constraints that involve three or more variables. Sometimes they're called global constraints as well, okay? For example, we can have this ternary constraint over here between x, y, z, and that means x is less than or equal to y, less than or equal to z. These are the three variables involved in this constraint. They have to be in increasing order. Another type of constraint is the so-called all-diff constraint. This is used in Sudoku. Sudoku is the well-known Japanese puzzle, right, that became famous and still is very popular. You see when you travel in planes and so on, you see people around you solving Sudokus and so on. In any case, so in Sudoku, you know that all variables must have different values, all variables in a row, in a column, and in every 3 by 3 square, right? So all diff is another type of constraint with a lot more than 3 variables. And the point is that with a little bit of ingenuity, you can transform all the higher order constraints into binary. This takes some effort, but when you do that, of course, you add a lot more binary constraints, but then you're ready to input your binary CSP in a CSP algorithm. I will skip a few things, unfortunately, again, in the interest of time. So I just want to mention a few more things on the algorithmic front of CSP formulations. We have constraint propagation and inference. What do we mean by that? We mean that in CSP search, we look for new variables assignments, or we perform some inference, and this is called constraint propagation. Inference is a well-known technique, of course, inference deduction and so on. All of these AI concepts are inspired from mathematical logic. So constraint propagation means we use the constraint to reduce the number of legal values for a variable. And that can have the same effect on another variable and so on and so forth. Okay. There is also the fundamental idea of local consistency, which means that if we treat every variable as a node and every binary constraint as an arc, then we use a graph to represent the CSP. And there are different types of local consistency. There is the node consistency that I want to talk about. So a single variable in a CSP is node consistent if all the values in the variable's domain satisfy the variable's unary constraints. Unary means constraints with one variable exclusively participating in the constraint. For example, if Southern Australia cannot be colored green, then its initial domain is red and blue. We don't need the color green. A network of nodes is node consistent, or variables is node consistent if every variable in it is node consistent. In other words, it's possible to eliminate all unary constraints in a CSP by enforcing node consistency, which means moving the effect of the constraint in the domain. This is what I was telling you. Every n-ary constraint can be transformed into binary ones. This is an example. Suppose that we have the ternary constraint A equals B plus C. Then we introduce a new variable AB with the domain, the Cartesian product of the domain A and the domain B. And we set up three binary constraints, AAB, BAB, and the sum of AB is equal to C. So we've translated the ternary constraint into three binary ones. Therefore, this slide tells us that node consistency plus these processes of transforming global constraints to binary ones allow us to use this assumption from now on that all CSPs will have binary constraints only. Okay. There is another concept called arc consistency that we won't have time to discuss today. So I would rather finish my talk with this concept of local search for CSPs. So this is one of the algorithms that we use, and I do want to mention it to show you the amazing power of CSP solvers. So we have this famous 4 queens problem, right? So this one here. You see on this board, this is a 4x4 board, and we placed 4 queens. Every queen is on her own column, right? And if you count the number of attacking queens, you find that you have five attacks. So 5 pairs of queens are attacking each other. Now, if we take this queen over here on the second column and move it up here, then we reduce the number of attacks to 2. And finally, if we take this queen over here and place it down here, then we reduce the number of the pairs of attacking queens to 0. So this over here, this board, this 4x4 board is a solution of the 4 queens problem, where we are able to place 4 queens on a 4x4 board so that none of them is attacking each other. And you know that in chess, of course, queens attack each other vertically, horizontally, and on the diagonal, on all diagonals. So it's a complicated problem, right? And this approach that we try to reduce the number, this evaluation heuristic function h of n, which is the number of attacking pairs of attacking queens, right? This approach is called, in fact, let me go back here. This is the minimal conflicts heuristic, a famous heuristic in the realm of CSP programming. And the point is that this heuristic allows you to solve big instances. So let's say n, the number of queens, 10 million, right? So imagine a board with 10 million rows, 10 million columns, and 10 million queens. How would you place those 10 million queens in order to solve this problem? Well, this algorithm allows you to do that. This is the pseudocode for the min conflicts algorithm. And this is the example that I wanted to show. This is an 8x8, so an 8 queens problem. This is not a solution, clearly here on the left. But if we take this last queen here and number the remaining squares in the column with the value of h of n, then we see that, in fact, if we move this queen up here, this is 1, this is the minimum number of conflicts, so we move it up here. Then we do the same for this queen, and we see a 0 over here. So we move this queen down here, and we get a solution in 2 steps. So the runtime of the mean conflicts algorithm is essentially independent of problem size, and it actually solves the 1 million queens problem in an average of 50 steps, which is an amazing achievement if you think about it. Again, think of a board with 1 million queens, 1 million rows, 1 million columns, and you have to place them so that none of them is attacking each other. Of course, this problem has some features that allow this kind of behavior. The solutions are densely populated throughout the search space. So there are many solutions. Some of them are equivalent and so on. There are some other technologies that allow us to escape from various plateau areas in the landscape, in the state space, and we can use other technologies like taboo search, etc. So this is pretty much what I wanted to discuss today. Again, CSPs are a very special kind of problem where we have states defined by values of a fixed set of variables. We can use all these techniques here: backtracking, arc path and node consistency, constraint propagation, and so on. And finally, mean conflicts. This is most amazing local search algorithm variation, if you like, on the theme to solve CSP problems very effectively. And to conclude my talk, I would like to say that I think there is a lot of space here in the realm of CSP programming for medical applications. So whenever you believe that you can formulate a problem as a CSP, then there are very effective libraries and algorithms to provide solutions very quickly. I will keep the time. It's 11:04 in Athens, so I will stop here and thank you very much.
Thank you very much, Professor Kotsireas, both for the very interesting talk and additionally for being on time, especially in this series of keynote addresses. Any questions from the audience? Not yet. So I would like to thank you for introducing us to the constraint satisfaction problem world, and also for letting us know how effective this is for a multitude of computational problems of modern bioinformatics. And these problems are typically complex and often hard to solve. So my qualitative question would be, where do you see that CSPs are fit mostly in the bioinformatics subdomains? For example, would it be protein structure prediction? Would it be sequence analysis? Would it be biological system simulation? What would be the killer app, let's say, the killer domain to apply a CSP?
Well, very good question, Philippos. Thank you. And the answer is I don't really know, but I'm hoping to find out, by interacting with GeNeDis participants. That is my hope. That's why I proposed this topic, because as I mentioned in the beginning, most people outside of AI know deep learning and neural networks, and that's it, right? And maybe some data mining, as we heard in previous talks. But I haven't seen people talking about CSPs. And the issue with CSPs is that they have a very wide range of applicability, right? So if you look at this book that I mentioned, there are dozens of problems formulated as CSPs. So I don't see why not. We can't formulate problems in the realm of medical research, and you are the experts. I'm not a doctor, unfortunately. I'm not a neuroscientist. Regrettably, right? But you are, and so I came here to the experts to seek advice and figure out what kind of problems are amenable to CSP formulation, with the idea that if we are able to do that, then we will be able to solve very large instances of those problems very effectively using CSP solvers. CSP research is a very active area of research, right? There are conferences, there is a handbook of constraint problems programming, and there is a journal called Constraints, a Springer journal, and so on and so forth. It's a very active community, and the research in CSP is definitely very interesting. And it's also related with other fundamental core theoretical computer science problems like the satisfiability, the SAT, the satisfiability problem, right? So the SAT is a very special case of CSP. CSP is a lot more general. In any case, I don't want to monopolize the time, so I will stop here.
It was clear enough. Thank you very much.
Thank you.
At this point, I would like to thank you for joining us from Canada in a difficult time. And we may return to Professor Geriatrics, who we interrupted earlier, to continue with his keynote talk. Thank you very much, Professor Kotsirias. And we can shift the break a little bit later after Professor Geriatrics concludes.
Using the contrast barrier, we have been doing work also in microscopy. And in this area, we have been trying to push the envelope of speed in fluorescence microscopy, so you can see fast dynamic profiles, especially associated with neuronal responses and behavioral studies. And our general philosophy here is to combine optoacoustic measurements with fluorescence measurements, building hybrid systems, so that we can have microscopy that can capture the classical fluorescence signal, but also a lot of other signals in label-free mode. So within microscopy, as you can see on the right panel here, we can have the classical 2-photon together with second harmonic and third harmonic. We can add the optoacoustic microscopy signal in order to start resolving these labels I showed you, such as hemoglobin, lipids, and so on. And we can also play with the scale, so we can switch from a microscopy to a mesoscopy scale and visualize also larger organisms. In terms of scale, as a side, we have been developing these hybrid systems that they have on one axis the microscopy modalities, both optical microscopy and optoacoustic microscopy. In this schematic, you see the objective marked by the letters OL. And this is an objective where we can direct both the light to excite classical optical microscopy signals, such as 2-photon, as I mentioned, and second harmonic, but also the optoacoustic signals. On the other side, we're adding a mesoscopy detector, so not only we can see the large appearance of an animal. Here we have a zebrafish, and we can visualize the entire zebrafish on panel A on the right side of the panels, but also zoom in an area of interest and obtain optoacoustic images and other microscopy images. We have been working together with CHICE as well in order to take this idea of multimodal and multi-scale microscopy / mesoscopy into a system that can do both single plane illumination microscopy, but also optoacoustic microscopy. And we have been showing, for example, that we can monitor optoacoustic and fluorescent signals in developing zebrafish in this case, but we are not limited in the very small zebrafish anymore, the ones that they are transparent, maybe up to 6 days of development. But in this case, we monitored fish that they were growing and becoming adult, up to 47 days, the fluorescence signal in this case becomes more diffusive, as you can see in the panels E to H. But this reddish signal is the optoacoustic signal that remains high resolution because optoacoustics is not affected so much by depth and scatter. So we have a modality that potentially could follow development much beyond what is allowed by current microscopy systems. Now, coming back to the contrast that I mentioned, the classical microscopy technique, a lot of its value in terms of monitoring molecular contrast comes from using labels, fluorescent proteins, for example. And this has been a powerful modality that truly revolutionized the way that we can visualize cells and tissues in vivo. But what if we had a modality that can see molecular contrast directly without the use of labels? In this manner, we wouldn't have to create transgenic lines of animals, and potentially we could see a much larger number of molecular species than what is allowed by having 2, 3, 4 fluorescent proteins in the cell or the animal. To achieve this molecular specific detection without labels, we illuminated cells and tissues with mid-infrared pulsed lasers. This mid-infrared part of the radiation is known to detect molecules based on their specific absorption spectrum due to their vibrational modes. So we imaged cells. This is shown on the left panel, where we could see the characteristic peaks of lipids. This is shown in panels D and I, simultaneously with the peaks of proteins by switching the wavelength. So we could resolve contrast from proteins and lipids from cells without any labels. And the panel on the right shows the same concept in tissue slices. This is white adipose tissue. This is about a centimeter by a centimeter area, and we can visualize the distribution of carbohydrates, proteins, and lipids in the specimen, providing, in a way, histological slices here without the use of labels. Appearance of muscle, gastrocnemius muscle from a mouse, again seeing carbohydrates, protein, and lipid. So we have a technique that can visualize these species, and now we move into methods that they can also see within the lipids different lipid molecules or different proteins, and it converts in a way this mid-IR imaging to a reflectance microscopy. Because of the use of ultrasound, we can also penetrate deeper than what is allowed by purely optical methods. And we see this as a universal microscopy method to move into label-free imaging of molecules. Away from contrast, I would like to discuss another barrier, a resolution barrier. There has been a lot of research and work done recently on breaking diffraction limits for optical microscopy with these super resolution techniques. But we have done work in breaking the diffraction limit in ultrasound imaging and therefore also in optoacoustic imaging. Consider an ultrasound system. It uses detectors typically based on piezoelectric technology, PZT, and these are essentially elements consisting of small PZT components, the physical dimension of which are in the order of tens of micrometers. Even when we have single detectors, the element, the active element, has areas in the tens of microns. And the reason for this is sensitivity. So even if we try to miniaturize the area further, it is possible technically. However, the sensitivity of an element like this depends on the active area, and therefore the sensitivity drops quadratically with reduction of size. So at some point you cannot miniaturize these elements significantly because you will lose a lot of sensitivity. A couple of years back, we reviewed techniques that they use optical components, all optical detection of ultrasound, typically based on interferometry. There are other techniques too. In this case, you have an optical component that is perturbed by the ultrasound wave, and this perturbation shifts the resonance of the interferometer, and this is detected then as the ultrasound wave.
We took a step towards this direction by using silicon on insulator technology. This is essentially silicon photonics. I will not go into the technical details of this development. You can find a lot of these details in the publication listed at the bottom of the slide. Just very briefly, we have a cavity surrounded by 2 mirrors, so we can establish resonance in this cavity, photon resonance, interferometry essentially. But when sound hits this detector, it perturbs this resonance. So by detecting the changes of the signal, essentially we detect sound. What is interesting about this detector is its physical dimensions. So as I mentioned, a classical ultrasound detector has dimensions in the tens of micrometers. And this is schematically shown on the right side of the slide, where you have a very small, essentially classical detector of dimensions 20 by 20 micrometers. By comparison, our detector is submicron. It's this little box on the bottom right side of the slide. Because we have so small dimensions, our detector is 300 nanometers by about 500 nanometers, we can push the resolution, the lateral resolution by which we can measure ultrasound waves.
We characterize these detectors, and they have a very broad bandwidth. They go up to 230, 250 megahertz. But the interesting thing is that we can achieve resolution that is well beyond what is predicted just by the frequency conversion to resolution. And this is shown in the panel on the right side, where we have this structure. The panel K is a panel imaging by a conventional optical microscope, and the panel L is the image that we obtained with this detector. Now it looks a little bit more noisy, but the K is an optical image from a high quality optical microscope, and the image L is an optoacoustic image using our detector. So if you look at panel N, essentially the resolution we achieve now in ultrasound imaging is very similar to the resolution achieved with a good optical microscope with an objective of 60x. Which means that we can see optical type of resolution, but using ultrasound waves in the megahertz range, not in the gigahertz range that would be predicting a resolution like this. And such a detector would have a lot of different applications, from miniaturized components to possibly stacking a lot of these detectors next to each other and building small arrays or even ultrasound cameras. Of course, there's a lot of work that needs to be done in terms of multiplexing signals once you start using multiple detectors, but certainly it's an area that has attracted our interest, and we are also looking at possibilities in terms of both ultrasound, but of course, optoacoustic detection. I would like to end my talk by discussing another barrier. This is not a technical barrier, but it's a cultural barrier. And in Munich, we're investing a lot in breaking this barrier, because we realize when it comes to disease, the traditional ways of research, the technical people building a new technical evolution and the biologists building new knowledge, it's not the fastest way for improving healthcare. The way to demonstrate this is by showing an airplane. The airplane is a fantastic human achievement. You have 100 tons of weight flying in the sky at 900 kilometers per hour or more. the plane is based on physics, aerodynamics, forces and flows, and so on. However, the plane is not built by physicists. It's built by engineers. Why is that? Physics is a culture, is a research culture of discovery, of building new knowledge. Physicists are fundamentally interested in observing the world and discovering new aspects of nature. But you do not ask them to build planes or cars or phones. You interface here a whole different discipline that is based on physics, but instead of being interested in discovery, it's interested in problem solving. And this also means that these people will develop a new set of tools that will help them in this problem solving. So the classical engineering is based on physics, but has developed its own tools in order to do this translation of physics to solutions. And 100 years ago, there were many more physicists than engineers, but now, of course, the world is full with engineers and less physicists. If we look at what happens in medicine, there is a discrepancy. Biology, like physics, is a culture of discovery and knowledge. And in many cases, we think that these biologists will also find the solutions, but this is not what happened in the natural sciences. We here a need to interface a new culture of problem solving with background in, let's say, bioengineering, data science, but the background could be biology or physics or electrical engineering. The interesting aspect here is that these are people not defined by their background, but they are defined by building tools that will accelerate the conversion of knowledge to solutions. And in that respect, this bioengineering and data science, this engineering culture, for us is synonymous to translation. This is why translation does not work efficiently, because we do not have the same paradigm in the life sciences that has developed over the past 100 years in the natural sciences. So we are changing our message. We are talking now of discovery, and we're talking of solutions. And we believe that this word and generally this intermix of solutions and engineering, as I mentioned, serves the real meaning of translation. And we are also having conferences. This conference has been postponed due to COVID, but it will happen in the near future. And we are trying to find the people and the thinking that they set their minds and their goals based on finding a solution, starting from the problem, and finding a solution, not starting from a discovery standpoint. With this message, I've been fortunate enough to receive funding from German government and Bavarian government to build 3 centers now. We started first with Translatum. This is a translational oncology building that they build. It builds people, it brings together people from the life sciences and the natural sciences. We developed the Helmholtz Pioneer Campus, and now we also start developing the Bioengineering and Digital Health Center. All of these centers here, which I'm privileged to have set up and serve in a board of directors or director positions, they bring people from different disciplines, but they do not stop there. They want to develop a culture of solutions and not just a culture of discovery. Discovery is essential, is the basis of any development that engineering will do, but it's not enough in order to accelerate the path to translation. And we hope that with this support here, we will be able to accelerate how we bring solutions for the patient. This activity also generated a lot of open positions. So if you're interested in these new concepts and this area overall, feel free to get in touch, send your CV, inquire. We will be happy to see your interest into this activity. So there are many people I would like to thank, all of the researchers and the staff and our administration at the Institute and the chair. that they're responsible for the work I showed you. I appreciate all of the funding we received from Europe, from Germany, so that we are able to do our research, and I would like to thank you very much for your attention.
Okay, I would like to thank Professor Deliachristos for this very interesting keynote address, and also thank him for his patience regarding the schedule and the ordering of presentations. Any questions from the audience? Not yet. So, given the fact that we are already well behind schedule and we have delayed, and this is only the first session of the day, I would like to conclude the session here and thank all of you, all of the professors, for their very interesting works and presentations. And we continue with a short break to the next session that will start after the break. Thank you very much.
Also, thank you for chairing this session,
Thank
Philippos.
you.
Thank you.
it seems we are good to go for the second morning session. It's my great pleasure to introduce the next keynote speaker. Professor Michal Schwartz is the Maurice and Ilse Katz Professorial Chair in Neuroimmunology, Department of Neurobiology at Weizmann Institute of Science, Israel. And her title is Targeting Immune Checkpoint Mobilizes Immune Cells to Help the Brain to Fight Alzheimer's Disease and Dementia. Professor Schwartz, please.
Good morning. Can you hear me?
Yes, thank you.
Good. So my name is Michal Schwartz. I'm professor of neuroimmunology at the Weizmann Institute. The title of my presentation, Targeting Immune Checkpoint Mobilizes Immune Cells to Help the Brain Fight Alzheimer's Disease and Dementia. I think in this audience, there is no need for introduction. However, I would like to focus on few issues before I'm going to my own presentation. We all know that the disease is mostly sporadic, only 5 to 10% are inherited. The major risk factor is aging. What we know now, the disease is very heterogeneous among patients. It's very dynamic, which means that at different stage of the disease, the additional risk factors are added and contribute to disease progression. It's mostly associated with accumulation of waste, and there is a local inflammation. Since the disease was first discovered in 1905, 1906, there are numerous attempts to fight the disease by trying to reduce plaques, or either by preventing their formation or facilitating their removal. We know now that even if we manage to reduce the plaques, it doesn't affect the cognitive ability or behavior. In addition, numerous attempts were made to reduce inflammation, again, have fallen short. It left the community with a key question: what are we missing in understanding of the disease? In 2018, there was an article in Nature Medicine in which they interview 4 different approaches in searching for Alzheimer's disease therapy, one of which was our approach, immunotherapy. So our approach is a mechanism-driven, and it's based on really 2 decades of basic science discovery, where we started by asking a key question: how come that the brain cannot be assisted by the immune system? So the cartoon show you station in our work, in which each station represents another piece in the puzzle, which led us to the current approach. So briefly, what led us to the current approach, that in 1998, we discovered that macrophages are needed for brain repair, although 10% of the cells in the brain are microglia. Subsequently, a year later, we discovered that it's not only macrophages, but also T cells, CD4 positive T cells. And the major contribution in this set of articles was in 2006, where we discovered that even healthy brain plasticity, cognitive ability, neurogenesis are dependent on the integrity of the immune system. So although the immune cells from the circulation are basically excluded from the brain, we found that higher brain function are dependent on the immune system, which left us with key question: how does all it happen? So subsequently, we discovered that there is an interface between the brain and the immune system, which is an epithelial interface and not endothelial interface, which is the blood CSF barrier that can orchestrate the communication between the brain and the immune system. And in 2014, we discovered that this interface dysfunction in aging, which opened for us a new era in understanding Alzheimer's disease. So we assumed that in aging and in Alzheimer more so, we need communication between the brain and the immune system in order to facilitate maintenance and repair. And it's known that in aging, there is a systemic immune suppression, which is associated mainly with two major types of immune cells: elevation of suppressor cells from the T cell, which are Treg, and elevation of myeloid-derived suppressor cells. So altogether, led us to assume that maybe in Alzheimer, what is happening is the lack of communication between the brain and the immune system, which is needed for brain maintenance and repair. Over the years, since we first discovered that macrophages are needed for brain repair, this was at that time an acute injuries. Numerous papers were published by several groups all over the world, suggesting that recruitment of macrophages to the brain can facilitate removal of plaque. At that time, we didn't understand the mechanism, but this was the notion. So we and others demonstrated in Alzheimer over the years. So taken together, we decided to embark on Alzheimer's disease in order to understand, A, whether the communication is malfunctioning, and whether we can, by restoring this communication, we will be able to modify the disease. So this is one example in which we took 5X5 mice with 5 mutations, 2 in presenilin and 3 in APP. So in this mouse model, most of the features of Alzheimer's disease are there, except that it's happened in early age, which allows us, it's not a corona. I'm coughing not because of the corona. So we wanted to see whether in this mouse model in which the disease emerged early in life, it is associated with this communication between the brain and the immune system. So here we monitor expression of trafficking molecules by the choroid plexus, and the red show you that with the disease progression, we are losing trafficking molecules expressed by the epithelial layer. We found a similar phenomena in human section that we got from post-mortem patient with Alzheimer's disease, age-matched aging with no Alzheimer, and young people. So overall, the notion that we found that maybe there is early impairment of communication. We tested to see whether it's associated with any type of immune deficiency or immune suppression which develop in this disease. And we found that even before manifestation of cognitive ability, manifestation of cognitive ability in this colony is usually around 6 months. We see already 4 months deficiency in immune activity, and we see elevation in immunosuppressive cells, which for us was very intriguing. And independent work published by the group of Marquis showed the same phenomena in J20 mouse model. Independent study by another group demonstrated that if you breed the 5X5 mice with immune deficiency mice, the disease is more severe. So altogether, it led us that the systemic immune system is playing a key role in the disease, not as a primary cause, but it may determine the timing of the disease onset and the severity of the disease. So in our pilot study, published in 2015, we tested this working hypothesis. We boost the immune system, and we found that indeed we can modify the disease. This led us to suggest that maybe the best way to fight Alzheimer's is not by trying to neutralize what is going on in the brain, but boost the mechanism of repair. So the way that we decided to boost the mechanism of repair is by reducing suppression from the immune system, which is usually under strict control and more so in aging. One of the mechanisms that keep the immune system under strict control or strict control are inhibitory immune checkpoints. They are known very much in cancer, but they are not related to cancer. It's a general mechanism that keep the immune system under tight control. And we decided to adapt this mechanism of suppression of the immune system to explore its potential in Alzheimer's. So it's nothing to do with cancer. It's just because this is a mechanism that keeps immune system under tight control. So we decided to focus on a family of checkpoints known collectively as PD-1, PD-L1 pathway. They were discovered by a group in Japan with no connection to Alzheimer's, but mechanism. PD-1 stands for program death protein. So we adapted the antibody, and our first study was published in 2016, where we show that if we give a single injection of anti-PD-1 to an animal model of a 5X FAD in a relative advanced stage of the disease, and we tested a month later in a test of radial arm water maze, which is a 2-day test of learning and memory. So the black show you wild-type animal, the red show you Alzheimer animals. So over 2 days, they don't learn. The black show you that the animal learned to find the platform over 2 days. The last trial, they swim directly to the platform, which is hidden in this maze. If we give the control antibody to irrelevant antigen, we see that there is no effect, whereas when we give anti-PD-1, we see a robust improvement. So that's paved the way to continue. I know we can discuss it later. Initial trial to repeat our work measuring only plaque failed, but now there are several groups, independent groups, that repeated our work and discuss it later on. So the next step, what we did is to see whether PD-L1 can do the same, because PD-1 as a receptor can recognize more than one ligand, so we wanted to narrow down the activity. So we repeated the same animal model, the same type of animal model, the 5X FAD. Here you can see a dose dependency, and you can see that no effect of the control. With increasing dose, we see very nice effect on cognitive ability. And if we continue to test the animal, there will be no difference between the wild type and the Alzheimer mice. We will discuss later on how it happens. We wanted to see whether the effect beyond behavior, there is effect on pathology. So we measure soluble A-beta and insoluble A-beta by ELISA, and we saw about 40% reduction in the soluble A-beta as well as the insoluble. Now, because the treatment is not directed to the pathology in the brain, but boosts the immune system, we envision that it will be applicable to any type of Alzheimer or dementia. So we repeated our work and used a model of tau with 2 mutations in phospho-tau. Here we measure short-term memory by T-maze or Y-maze, again, single injection and a month later. So this is the behavior of wild-type animal, nice preference to the novel arm. In the Alzheimer model or in the tauopathy, no preference. When we treat with low dose, no preference. Increasing the dose, we see nice preference. Again, the MM behave is almost as good as wild-type animal. We continue here to see whether there is effect on pathology, and we found that indeed there is reduction in aggregated tau, both in the hippocampus and in the cortex, measured by thread-based ELISA. We measure also level of IL-1 beta, which is inflammatory cytokine, and we see decrease. We see that the majority of IL-1 beta is expressed by astrocytes, and very nicely, we see some rescue of neurons. I will discuss it in a few minutes. So overall, in 2 different mouse models, one of Alzheimer, one of amyloidosis, and one of tauopathy, we see effect on behavior, on cognitive ability, and on disease pathology. We lately tested the 3rd model, which is commonly used by the community. It's another model of tau, and so the same phenomena. So before dosing the animal, we see cognitive impairment, and after giving the anti-PD-L1, we see a slight improvement after 2 weeks and a full improvement after 4 weeks. This for us was very intriguing because in all models, we see improvement after 3 to 4 weeks. And we know that the antibody is cleared from the blood after 1 week, which suggested that the effect is mediated by messenger that induced by the treatment rather than by the treatment directly. So we repeated here in this model, again, the pathology. We see reduction of phospho-tau in the brain, and very nicely reduction of total tau in the CSF, although the treatment was not directed to any tau or aggregated tau or phospho-tau. So we see reduction. So we went to see what might be the messenger, and of course we consider that as a result of the treatment, we facilitate mobilization of immune cell to the brain. So we see elevation entrance of macrophages, and we see anti-inflammatory T cell. Now, because it's very difficult by flow cytometry to be 100% sure that when we see macrophages, it's pure macrophages and not microglia, we activated microglia, continue with single-cell RNA sequencing in order to make sure that the cells that enter are macrophages and not microglia. So by single-cell RNA sequencing, 4 clusters here are microglia, and these are macrophages. We see that the macrophages express unique molecules. And the most intriguing one are this one, MSR1, which is a scavenger receptor expressed by macrophages, which was reported already by the group in Harvard by El Khoury that they are needed for removal of soluble A-beta, not the insoluble A-beta. And we found that they also express LIVE1, which is classically anti-inflammatory cytokine or stain for lymphatic vessel. So there are other scavenger receptors that expressed by the macrophages and not by the microglia. This is MRC1, which is anti-inflammatory and again scavenger receptor. So the macrophages that enter to the brain express scavenger receptors that are not expressed by microglia. One of which is the MSR1, which is also known as CAR1, which was reported already. This is the paper in 2013 in vitro study that this receptor can facilitate clearance of soluble amyloid beta. So we decided to create mice in which their bone marrow will be replaced by bone marrow from MSR1 knockout mice. And thereby, we now have Alzheimer mice in which their bone marrow, only the macrophages, but not microglia, are deficient in MSR1. We wait 6 weeks for these mice to be created, and now we dose them with anti-PD-L1. So as control, we have Alzheimer mice with bone marrow that they received from wild-type animal. They responded as before to the anti-PD-L1. However, Alzheimer mice in which we replaced the bone marrow with bone marrow from MSR1 failed to respond to anti-PD-L1, which suggested that the macrophages are essential, and more so because MSR1 is one of the key receptors. We went further and repeated the experiment by eliminating monocytes completely using antibody that blocks CCR2. And here again, you can see, so this is a wild-type animal in a tau model, preference to the novel arm. This is IgG treatment. As before, impaired completely, anti-PD-L1 effect. And now we can see that when we eliminate monocytes, we lost the effect, suggesting that indeed the anti-PD-L1 effect is dependent on monocytes. And now we see that also the pathology was, the effect on pathology was abrogated. So this is a level of aggregated tau in wild type, aggregated tau in the Alzheimer, aggregated tau after PD-L1 and after PD-L1 and blocking the monocytes. Based on this, we envision that we have a therapy that is not dependent on maybe not dependent on microglia, but on macrophages. And we decided that this is the best time to check whether it's a TREM2 dependent, because we know already that in the 5xFAD model, microglia are activated in the TREM2 dependent manner. and they were claimed that they are needed for removal of plaque. And by many companies now and many groups, they think that the TREM2 is a good target for modifying the disease. Our results suggest that macrophages is a key player. So we got for Marco Colonna TREM2, 5xFAD TREM2 positive and TREM2 negative. So we repeated all the experiment using TREM2 positive mice and TREM2 negative mice. And this is IgG, no difference. And this was reported also by a recent paper by Marco Colonna and Tanzi, where they said that in terms of the behavior, there is no difference between TREM2 positive and TREM negative. When we gave them anti-PD-L1, we see improvement in the TREM2 positive, and we see also improvement in the TREM2 negative, slightly less, but very robust improvement. We measure by novel object recognition, which is working memory, and you can see it here. This is the wild type animal. These are the Alzheimer, the 5xFAD treated TREM positive, treated with IgG, and this is the TREM negative treated with IgG. So with and without TREM, the cognition is similarly impaired. When we gave anti-PD-L1, it improved cognition based on novel object recognition in the TREM positive and TREM negative. So we were very excited by these results because we knew already from the work of Tanzi and Colonna that in the TREM2 negative and TREM2 positive, the level of soluble A beta is the same, whereas plaque is higher in the absence of TREM2. So we measure soluble and insoluble A beta in the TREM negative mice, and you can see that the treatment reduced the soluble A beta by 30%, no effect on the insoluble. So the improvement of cognition in the TREM2 negative was not dependent on the insoluble, only on the soluble. We went to the TREM positive, and here we see reduction in both soluble and insoluble. So the effect of cognition in the TREM2 positive and negative is mainly due to the soluble A beta. So we continue in the TREM negative to see what is the effect on synapses, because we know that the soluble A beta is containing the oligomer, which are toxic to the neuron. So we measure synapses by measuring from 6 animals in each group, 4 slices from each animal, total 96 slides in completely blinded manner. We gave it to summer students, and we found that both in the antigyrus and in the CS3, we see a higher level of synaptophysin, which suggests rescue of neurons from the cytotoxicity of the oligomer. And this explains why we rescue function and can reverse function, loss of function in those neurons that when they are being treated, they are still alive and are surrounded by the toxic material. So finally, we repeated the experiment in the TREM negative and block macrophages. And what we wanted to see whether macrophages are sufficient for our observation. So here you can see repeated of the experiment. We have TREM2 negative with anti-PD-L1 and TREM2 negative when we block completely the monocyte recruitment. And this is the novel object recognition. So these are the wild type animal. This is TREM negative treated with IgG, TREM negative with anti-PD-L1, and TREM negative blocking the monocytes. We see that we also, as a result of blocking monocytes, we have higher level of soluble A beta, and there is a very, very nice correlation between soluble A beta and behavior in radial arm water maze. So this is the effect also in the TREM positive. We lost major part of the effect by blocking the monocytes in the radial arm water maze. This is the anti-PD-L1 in the TREM positive. And when we block monocytes, this is what we got. We lost most of the effect. And this is the novel object recognition. And finally, because we found that macrophages are doing the job in both TREM2 positive and TREM2 negative. We repeated the single cell RNA set. So what we see that indeed, in the TREM2 negative, we don't have the activated microglia, which we, together with Idomit, called them in a paper that we published in 2017 as disease-associated macrophages. So we have that in a TREM positive, we don't have them in TREM negative. However, the macrophages that we found in the two TREM2 positive and TREM2 negative have a unique signature. It's the same signature in both of them. And again, as we found before, these macrophages express unique scavenger receptor. These are the macrophages, these are the microglia that are not expressed microglia. Microglia express TREM2, macrophages don't express TREM2. So to summarize, this is what we are proposing. At any given time, we know that there is a soluble, there is the monomer of A-beta, which is needed for brain function, for synapse activity. If they are not cleared, they are aggregated to dimer, triplemer, up to dodecamer. The dodecamer is toxic to the neuron. So at any given time, we have still neuron that can still functioning. When we are giving anti-PD-1 to the blood, we facilitate migration homing of macrophages. The macrophages express unique scavenger receptor and anti-inflammatory activity. The scavenger receptor remove this aggregated form. They don't have any effect on the plaque. So the effect of a plaque is secondary because if we remove the oligomer, they are not accumulated to plaques. So our ability to take, at any given time that we give the treatment, we can rescue neurons. Not always we can see plaques depending on the speed of progression and the stage of the disease. For long-term effect, we found that we need to give periodically the treatment, which we are now doing, and we have developed a unique antibody, anti-PD-1, which is distinct from the one used in cancer, which has a better safety profile. The modification was possible because of the mechanism of action, and we are almost ready for clinical trial, and we believe that the antibody will be given periodically every 3 months. So basically what we are doing by the treatment is nothing else but activating a process of repair, and the antibody is needed only for the early stage. Thereafter is chain of event, which is sequential, and each step depends on the other step. This work was heavily supported by advanced ERC, which I received, and more than once, and also by private competitive grants, Israeli competitive grants, and lately also by private foundation. These are my team, current team. All the single cells were done in collaboration with the team of Idomit. And since it's a work that emerged after 20 years of intensive work, these are former graduate students and collaborators. And thank you.
Thank you very much, Professor Schwartz, for a stimulating and thought-provoking talk. Are there any questions for the speaker?
I thought that I'm not provocative anymore. It's the mainstream now.
I don't see any questions on the chat, but we are on time. Definitely, thank you for keeping the time, so we have time for a few questions.
Either it was too very clear or too fast.
I'm sure people have a lot of questions. Maybe they will contact you in person or later on, but I don't see anything on the chat right now. Can I ask the technical support? Are you seeing anything, any questions for the speaker? Okay, I'm getting a negative response. It is 12:15 here in Athens, so I mean in Athens time. Therefore, I think we're good to finish this second morning session and thank Professor Schwartz once more virtually. We will reconvene at 2:15 p.m. Athens time. Then, thank you.
Thank you.
We welcome you all to this third session of the GeNeDis conference, which is the third morning session of the third day of Saturday. I'm happy to see that I've been joined by the panelists, and we can start right away with the papers of the session, which is entitled Neurodevelopment, and obviously is dedicated to related topics. The first keynote address, entitled Molecular Function of Chromatin Regulators Linked to Neurodevelopment Disorders, is going to be presented by Dr. Kyung-Min No, who is currently leading a group at EMBL Heidelberg in Germany about the genetic mechanism of neurodevelopment and diseases. Dr. Min No, the floor is yours. Thank you very much.
Thank you so much for nice introduction. Can you hear me and can you see my presentations?
Yes, everything is fine.
Thank you. Thank you. So as introduced, my name is Kyung-Min No. Good afternoon, everyone. I'm the group leaders in the genome biology unit at EMBL Heidelberg. We are doing a very basic research, understand mechanistic elements of the individual's molecular level of the individual biological pathway. And my group is particularly interested in the intersecting area between the chromatin biology and neurobiology, where I did my PhD in the neuro and then postdoc in the chromatin biology. So I think this intersecting area could be the nice niche for further study my research. I will present you some of our study in the defining the molecular function of the chromatin regulators linked to the neurodevelopmental disorders. So neurodevelopmental disorders such as autism and intellectual disabilities are the quite common behavior phenotype. However, their molecular mechanism and cellular diversity are very, very different. So for example, some of the autism intellectual disability patients has differed or altered the dendritic morphology in their spine. And therefore, there is also different synaptic structural component and different postsynaptic and then presynaptic changes often observed. There is also growing evidence show that there is the firing rate difference between the excitatory and inhibitory neuron imbalance. There is also some patients show that the calcium channel activity changing. Last but not least, but there is also growing evidence that this patient show that altered the chromatin or epigenetic changes in the nucleus. Traditionally, these synaptic changes, spine morphology, firing rate, and calcium channels are very familiar to the neurobiologist how to tackle, how to study this element. However, this chromatin changes, somehow it's quite new to neurobiologists how to study that. So our group is focusing on to understand what is happening within the chromatin, within the nucleus. Because the chromatin is the basically physiological form of our genomes. It is myriad string of this basic unit called nucleosome, which is composed of chimeric histone protein wrapped around with the DNA. And then this histone protein is excellent substrate for the multiple enzyme. For example, histone can be further modified by different enzyme systems such as writer, which put the histone modifications, or eraser, which remove the histone modifications. And sometimes these reading modules recognize the specific histone modifications. Now often we call as a part of epigenetic systems. So these reading modules can directly impact on the gene expressions. There is also a specific protein complex called the histone remodeler, which can move around nucleosome, can make the space for the other proteins such as polymerase can be more active. The surprising event of this autism and intellectual disability case, because the parents are quite interested what makes these kids, their kids to more vulnerable to this disease. So they made this exome sequencing or sometimes whole genome sequencing with their parents and also the kids together. So even the number is quite small, their statistic power to find pinpoint which mutation can be causally associated with this autism or intellectual disability is quite high. And then now we know that actually this histone writer erase reader mutations quite often found in this disease patient. And that's the area that our lab is highly focused on the study. What is the molecular mechanism of those mutations in that disease setting? So past 5 years, our lab has actively studied some of these molecules. Today, I will mainly focus on this SETD5 protein. So SETD5 is a set domain contained protein. So set domain is the only known protein structures in this known named protein. Set domain is known for methyltransferase activity. So maybe it induced histone modifications or other non-histone substrate. So that is the basic idea about that time we start to study the SETD5. The mutations that happens throughout the entire genome of the SETD5. There's no mutation hotspot. So there is a two take-home message in these mutations. It happens and throughout the coding genes, and mainly it makes the truncated form of the protein. So therefore, it's a loss of function mutations. And these also mutations happen in the only one copy of the genome. So it is haploinsufficiency. Even they have functionally intact one copy of the other copy of the genome, SETD5 gene exists, this haploinsufficiency, half amount of the protein is not enough, so it caused disease. And of course, it happened as a de novo. It doesn't affect, it doesn't found their parents and the siblings, so it's highly relevant, statistically highly relevant mutations associated with that. So it accounts about 1% of the intellectual disability patients compared to the cancer like a P53 or these BRCA mutations make the very, very high penetrate. This 1% seems a very, very small number. However, consider the diversity of the intellectual disability and autism. This number is actually very high number. However, we don't know anything about the SETD5 that times when we started, so we want to know what is the exact function. So basically we thought the SETD5 could be the methyltransferase. So therefore, we purified this set domain, or we purified the entire protein complexes. And then we mixed with the mononucleosome, oligonucleosome, also the cofactor, tritium labeled methionine. So the idea is that after we mix together and then we expose, then the substrate and then enzyme will be labeled with that. So as you can see here, the positive control, that's the known methyltransferase activity protein complex polycomb. After you run the gel, you can see that the polycomb is labeled with the tritium methionine, and then also it transfers enzymatic activity happen in this lower molecular weight of the histone. However, the SETD5, we didn't see any enzymatic activity. The SAM didn't bind to the protein, neither to the transfer to the histones. So it means that this SETD5, at least in this setting, doesn't have any enzymatic activity. So this is surprise, but also it's not a super surprise because it is known that, for example, some kinase doesn't have any kinase activity, but now it worked as a platform protein to recruit the protein complex and work as a functional unit of the protein complex. So we thought SETD5 could be function like that. One of the hurdles of this new protein study is that we don't have a good antibody. Therefore, to purify the protein complex, we endogenously tag one of the genes, the flag and HA, and then we pull that out in the endogenous setting in the neuroprecursor and ES cells. And we also used a knockout of the SETI5 and as our control to validate our protein complexes. And then after IP, we found multiple protein complexes, and a lot of protein actually precipitate together with the SETI5, but two protein complexes quite distinct. One is the NCOR HDAC3 complexes, and then the other one is green dot, that's the PAF1 complex. These two protein complexes has very distinct functions in the chromatin. So NCOR HDAC3s is, as you can see, that HDAC is the enzyme that removed acetylations from the histone. So acetylation is often connected to the active state of the chromatin. So removing them is the repressive or inactive state of the chromatin. And also the PAF complex is the posing, involved in the posing state of the polymerase. So somehow these two protein complex has functions of repressed or posing transcriptions. So then what is the effect of these protein complex formations under the mutations of the SETI5? So to study that, we generate truncated mutations of this SETI5, or is found in the autism spectrum disorder patients. And then we now call the shorter one 308, and then longest one 1258. So this serial truncation of the protein, now we can see that what is the effect on the forming the protein complex. We also, first of all, we look at their protein expression level. So this is the full-length proteins, highly expressed, both cytoplasm and nuclear extract. However, this shortest form, you don't see, barely see any protein protein exist. So it is purely a loss of functions protein. And now the truncated forms start to get expressed, both, this very short truncated form cannot go to the nucleus where the function need to be the most. However, this other truncated form, now it can express both cytoplasm and nuclear. So we selected two of those. This is just ectopic expression protein, but these two, 720 and then one longest one, to now endogenously express at the MPC and ES cells. And then we perform the IP experiment. So now you can see the full length when we IP the endogenous setting, it now recruit HDAC3 complexes and PAF complex in here. However, in the truncated form of the SETI5 mutant, cannot make the complex with the HDAC3, and then there is also reduced of complex formation with these PAF1 complexes. Therefore, now we can say that SETI5 form the complex with the HDAC3 and PAF, mainly where the polymerase activity is highly abundant. And then it may be posing this polymerase to reduce their activity. So now the main question is, where is the target location of the SETI5 in the genome, and what is the target genes in here? And for tackle these questions, we performed a genome-wide assay where is the SETI5 located in the genome, and also using this heterozygous and homozygous knockout line that we generated, and then we performed the ChIP-seq experiment to find the location of individual protein and histone mark. Now let's go to the first. We found that the SETI5 is highly located, occupied in the genome where that the polymerase enriched transcription start site. So if we divide it in genomic regions, the polymerase occupied transcription start site, and then polymerase non-occupied transcription start site, and then transcription exit site, and also the enhancer located the polymerase and other enhancer, you can see the clear pattern that this green box, that's the chip materials, where the SETI5 occupied in the genome is more localized, highly enriched, and the polymerase occupied transcription start site and enhancer, but not other regions. If we profile transcription start site and then divide it to these three different regions, one region is that HDAC3 located and then POL2 occupied, double occupied transcription start site. So those are the most enrichment can be observed with the SETI5 occupancy. So that there is a clear trend that any transcription start site, both of them are highly located. There is also SETI5 is highly occupied on those transcription start sites. But even though one of them is there, there's still an enrichment we can observe. So how about this mark can change it upon SETI5 heterozygous and homozygous mutant? So the first idea is that, okay, if we located with this HDAC3 together, and then this truncated form lost HDAC3 binding, we thought maybe HDAC3 occupancy is get changed upon the SETI5 knockout. But that was not the case in the heterozygous, and neither the homozygous. The change of the HDAC3 occupancy in the genome is almost nothing. There is no change. Therefore, HDAC3 occupancy itself doesn't make any change. However, when we look the polymerase binding, polymerase occupancy, there is a massive changes upon this SETI5 dosages. We saw that haploid sufficiency case, there is a lot of changes as strong as a homozygous knockout. So somehow this polymerase activity is very, very sensitive for the SETI5 dosages. And to go further analyze it, now we try to find a transcription start site where that the SETI5 is highly located. And then with those regions, about 550 transcription site, we look at how is the polymerase get changed on those sites. And then we found that polymerase get even more increased on those regions, and HDAC3 it doesn't have any change, and neither with the acetylation level. So it seems that SETI5, loss of SETI5, main effect happen in the polymerase. And then those genes are highly enriched, marking this nervous system development, also synaptic activity, or this behavior. So as a overall model that this SETI5, HDAC3, and PAF complex formed, and then transcription start site holding the polymerase activity, in particular in the developmental genes, when this SETI5 is a haploid sufficiency happens or complete knockout happens, HDAC3 is maintained. So maybe the HDAC3 also contribute recruiting the SETI5 on those sites. However, upon these conditions, now you start to see that more active polymerase activity upon loss of the SETI5, we see the increase of the transcript generations. This is the obvious consequence of the gene expression. So when we differentiate those mouse ESL to the neurons and perform the RNA-seq, we start to see there is increase of the transcript, particularly at the heterozygous conditions and neural system development and then neurogenesis. However, in the homozygous condition, we start to see the different set of the genes more increased. So this one is also quite similar to the mouse phenotype, because our collaborator in the IST showed that heterozygous mouse is survive ratios. However, SETI5 homozygous knockout is embryonic lethal because they cannot make the proper heart formations. And with that, now we can get the summary that the SETI5 has a novel protein complex component in make the Ancora HDAC3 complexes and also PAF1 complexes. And SETI5 is not required for the HDAC3 recruitment. There is also no change of acetylations. Maybe HDAC3 is needed to recruit the HDAC SETI5 in the transcription start site. SETI5 disrupt the polymerase occupancy in the developmental genes. And also this might be potentially linked to the neural lineages potentially explaining the etiology. So I also go to quickly the part that we also found in the mouse study, because even this very pleiotropic effect happened in this SETD5 heterozygous and then knockout ES cells, and their phenotype is quite distinct in the mouse. So maybe there is a link between what happened in the chromatin and what happened in here, in this domain. So what we observed in the mouse, of course, that knockout mouse is embryonic lethal, but heterozygous mouse, we start to see they have enhanced LTP can be observed in here. So definitely there is more running or prone to running happening in here. And then more surprising happened when we performed the cognitive test. So when we tested the cognitive peer conditioning, putting the mouse in this context chamber, that wall is marked with the context symbol, and then electric shock happens, both heterozygous and wild type learned equally well. However, the challenge happens after 24 hours when we put the mouse back into the cages. As soon as they see this wall painted, and then they start to freeze without electric shocks. And then this SETD5 heterozygous mouse has even more higher freezing, so they can overrun. And even more surprise happened when we now train the mouse. This symbol is not linked with anymore with electric shock, and then wild type mouse learn adaptive running, and then now is they know that is the safe, so they don't have show any freezing behavior. However, this SETD5 heterozygous mouse still show strong freezing behavior. So there is definitely a less flexible behavior can be found in this mutant. And we also found that when we extracted out these hippocampal regions and do the profiling of the transcription of those regions, there is a gene expression change that happens. Now you can see that when we cluster those genes, 11 different their expression patterns. Now you can see clearly this red is the SETD5 heterozygous mouse gene expression pattern. Some of them after the shock get lower, but still get maintained increased, unlike the wild type that has a spike and then get decreased. So almost like an opposite pattern. When that things has to be downregulated, it cannot downregulate, it still maintain a high expression. So when we extracted those genes and then go to the pathway analysis and then look at the genes, and those genes are highly marked in the postsynaptic density. So maybe those are the one of the reasons why the mouse is overrun and why we see the enhanced LTP in that postsynaptic area. So now, as a second summary, there's a SETD5 showed enhanced LTP, and this is actually not very unique for the SETD5 mouse. We often observed this phenotype in the autism spectrum disorder model mouse, suggesting that this disrupted cellular or molecular synaptic plasticity may be associated with the abnormal learning and memory. And then SETD5 haploinsufficiency is associated with the better memory retentions, but it decrease the potential, the capacity to extinguish the existing memory. So therefore, the cognitive deficit of this heterozygous mouse accompanied this abnormal dynamics of the postsynaptic density gene expression. So maybe we can now make the global model, right? So this SETD5 definitely is pleiotropic protein. It has function of the gene regulations, polymerase activity. However, each stage of the cell development, each condition of the cells, they have different target genes. There's not a unique single target. It is changing depend on the cell's state or cell developmental stages. And that is one of the reasons why this survived mouse has constant changes upon the learning and then gene expression. Even they compromise and buffered each stage of the development, they have this prone effect still maintained. This increased polymerase activity can happen any stages when this mouse get more challenges and adapted upon this experiment, these challenging conditions. With that, I will just go to the outlook. Now we know that this is the chromatin regulators show the distinct phenotype in the gene expressions. How about other chromatin regulator? Is that also happen in the same way of what happened in the SETI5? I will just tell you that's not the case. Each chromatin regulator will show the very unique phenotype. And then now we see one of the example of the PHF14. It makes very distinct, different gene expression profile. We now know that the PHF14 is linked with the developmental disorder because it makes the DNA damage response change it. It linked with the DNA damage response. Okay, with all that, now I want to thanks for the lab who is continuously studying this chromatin regulator in the neuronal context. And then the majority of the work I presented is from my first PhD student, Nicolo. And then all the rest of the lab members support it, and then they also their own project on running. So maybe next time I will present also exciting new finding. And then I also need to appreciate the Gaia Novarino group in the IST. We work together to accomplish this SETI5 exploration. Okay, thanks for your attention. I will be happy to take any questions.
Thank you very much, Dr. Mignot, for being accurate on time and for allowing us to a new window to this subsection of the conference, of the topic. Any questions from the audience? Maybe? Not yet. In this case, I would like to ask a more qualitative question. I mean, obviously, your field of expertise is not the one that I share, but I understand that neurodevelopment disorders are in principle a bunch of disorders that affect the nervous system of the human. So they disrupt the normal brain function in a way, and this also control emotions and self-controls and this of the human. And the question would be, do you think that the field of personalized medicine would help all these beautiful things that you described today and the research that is being done in this field? Do you think that following the personalized medicine concept would aid the research and also the achievements in your field? Or would it be the other way around, that we need to have a more generic approach to the problem?
No, actually, it's excellent questions, right? How can overarching our research to the this personalized one? And basically, this area of the research is very, very active, particularly for the motivated parents. They want to understand what is happening in these kids, and individual kids already their development. And at this point, there is very hard to rebirth their symptom. However, the alleviations or sometimes if it's coupled with some strong hormone changes or adolescence period, whereas the second stimulations phases developmental happen, maybe we can add some additional drug treatment, for example, to potentially alleviate symptoms. If in this case, SETI5 is highly connected to the gene expression pattern, and it's obvious that there is more polymerase activity and more so therefore, we can reduce this spurious transcription. There is a way we can reduce these spurious transcriptions, not very specific, but it's often used in the cancer therapy. For example, the bromodomain inhibitors or JQ1, etc. So maybe there is a small window of time of during this developmental stage, we can personalize the treatment. Particularly for the SETI5 cases. but not other autism, but not other intellectual disability. For that cases, I think individually we have to understand what is the other chromatin regulator that mutate in this case happens.
Okay, then there is a lot to see in the near future, I guess, in this field of expertise.
Yeah, absolutely. Thank you.
Perfect. Thank you very much for your presentation. And then to be right on time, I'm going to move forward to the second keynote address of the day, which is entitled Hypoxia and the Energy Crisis View in Experimental Models of Multiple Sclerosis. It's going to be presented by Dr. Mariana Castí, who is a doctor and also a consultant neurologist and lead clinician at East Essex NHS Trust. So, Dr. Castí, the floor is yours.
Thank you very much. Can you see my slides, please?
Not yet.
Can you see my slides?
Not yet, actually. Now the presentation started, and we can see your desktop.
Now, can you see my slides, please?
Yes, perfect. Thank you very much.
Thank you very much. Thank you very much for inviting me to this very, very interesting Congress, which includes various aspects of neurobiology, and I'm very happy to present the ideas that they have been developed over the past 10, 15 years in the Department of Neuroinflammation in London, in UCL. And, I will try to give you an insight of these ideas in multiple sclerosis. So the title of the talk today is Hypoxia and the Energy Crisis View in the experimental models of multiple sclerosis and their therapeutic implications. As I'm a clinician, I would like to tell you a bit about the disease, what we see in clinic. Characteristically, we do have the benign multiple sclerosis, the relapsing remitting multiple sclerosis, the secondary progressive multiple sclerosis, and the primary progressive multiple sclerosis with different disease phenotypes and with different ways to disability. So what we see in clinic is this. We have pre-symptomatic patients. We have patients with CIS, which is a clinically isolated syndrome. Patients with relapsing remitting multiple sclerosis and secondary progressive multiple sclerosis. So they are coming to in the clinic when they have symptoms in the beginning. However, the pathophysiology of the disease starts early on and is not visible, is not symptomatic. What we see is what we do. We follow the patients with MRI scans. We see an increase in T2 lesion load along with the disability and the brain atrophy. Our motto is to treat as early as we can. I like showing this picture where we see here a classic MRI scan of a patient with relapsing remitting multiple sclerosis, given in the baseline the treatment. And as you see in the first 3 years, we have a reduction in brain atrophy of the level of 0.089%. But when the people stop their treatment, in a matter of a year, the brain atrophy increases dramatically by 3.8% in this case. So with the neurologists, we would like to treat as early as we can. We have this type of anxiety, if you wish. Now, let's go to to understand a bit more about the disease, and indeed the pathological hallmarks in multiple sclerosis is inflammation, demyelination, and axonal degeneration. It is considered a primarily demyelinating disease. However, these three components are considered as the main important pathological markers. The multiple sclerosis is a disease which will lead sooner or later to brain atrophy, given the neurodegeneration. This can happen in a matter of years or in a matter of decades. We see typically a blood-brain barrier disruption with perivascular inflammation leading to what we see in clinic, the gadolinium enhancement. Now, what we do in MS is we try to treat, and this is what we consider the therapeutic pyramid. So what we would like ideally to do is to give anti-inflammatory drugs, to have neuroprotective drugs, remyelinating drugs, and neuroreparative drugs. At the moment, we treat MS with anti-inflammatory, in a way, drugs. I'm not an immunologist. I'm not going to give you the insights of the MS treatment today. However, just to say that we have drugs that they do target the peripheral immune system, drugs that they do target the blood-brain barrier, which is disrupted in a way, like natalizumab, for example, where there is an inhibition of diapedesis of immune cells within the brain. So far, we target the immune system, and it has been successful. So indeed, switching off adaptive immune response inhibits MS. And these are different drugs that we use successfully, like the dimethyl fumarate, newer drugs like the ocrelizumab, fingolimod, alemtuzumab, importantly, and so on. However, although there is a huge progress between the situation in the MS world 20 years ago and now, still there is no cure. There are different other approaches, such as the neuroprotection, remyelination approaches, like the LINGO studies, neuroreparative approaches. I'm going to focus to neuroprotection, and I'm going to show you what likely is going to be part of the future. So there are clinical studies that they do target the ongoing chronic neurodegeneration. We like in the MS world saying that there is a post-inflammatory slow burning of the neurons. So new approaches, they should target inflammation, axonal mechanisms, like the mitochondrion and the energetics, axonal targets, remyelination, other comorbidities like aging, for example, the simvastatin study, which happens now in secondary progressive MS. Acute neuroprotection would be the way to target events that they do happen acutely and try to protect the actions. And going to go back to this. We have the trial of phenytoin trial, which has been developed from Raj Kapoor over the last 10 years in the Department of Neuroinflammation, in the Department of Neurology. It has been the first study where in optic neuritis, it has been given an anti-epileptic with some good results. If you see, there was a 30% reduction of atrophy of the optic nerve in the active group. So how comes an anti-epileptic drug to a voltage-gated sodium channel blocker to provide neuroprotection in MS? And how comes that if someone takes an anti-epileptic drug, recovers, his optic nerve recovers by 30%? This is like these ideas, they have been developed over the last decade in the lab and in the department, and promote these ideas. They do promote the idea of this energy perspective in multiple sclerosis. And indeed, we have to go back to neurobiology to understand why this clinical trial has had this promising result. So multiple sclerosis, in the neuropathology of multiple sclerosis, we have four patterns, four distinct patterns of MS lesions. Two of those, pattern 1 and pattern 2, are mediated by or a cytokine or an antibody, by macrophages, for instance. But the other two patterns, pattern 3 and pattern 4, they are very distinct ones, as it looks like that the oligodendrocyte by itself degenerates. And importantly, in the pattern 3, it has been found that there were features of hypoxia and features of viral features that we find in viral infections and in ischemia. The next step was to go to neuropathology and see what is going on, and indeed, from two different labs, one from Germany, the other one from Vienna, from Hans Lassmann's group, they found HIF-1A reactivity in pattern 3 lesions in Hans Lassmann's group, and in the normal appearing white matter ischemic preconditioning situations in MS lesions, in MS neuropathology studies. Other studies by other groups, the Prineas group in Australia, showed that there is an extensive oligodendrocyte apoptosis due to changes in the local environment. Other studies previously showed also that inflammation alone may be sufficient to cause significant clinical deficit without demyelination. Demyelination in the MS world is the dogma. when we go to university, we hear that we have a relapse because there is demyelination. However, demyelination likely is not a sufficient explanation for relapses because some relapses in MS are only brief, because there are data encephalomyelitis that they say that without demyelination we have clinical deficit, and because we have biopsy observations in MS. So demyelination is not the only player. At the same time, past decade, 1997, 2005, 2003, it was known that there is a nitrative and oxidative damage implicated in the explanation of hypoxia-like observations in autopsy and microarray studies. So the whole thinking was to think the pathophysiology of the MS in a different way. So traditionally, the MS is considered a chronic neuroinflammatory disease. There was, however, growing evidence of a mismatch. I'm going to explain why between energy supply and energy demand in MS lesions. Nitric oxide by the INOS is found in MS lesions. Nitric oxide, we know that binds competitively with oxygen at mitochondrial complex 4, inhibiting the production of ATP. We also know that the nitric oxide and the peroxynitrite inhibit the mitochondrial electron transport and therefore the oxidative phosphorylation and therefore less ATP. We knew also that there was an impaired mitochondrial activity of complexes 1 and 3 in MS lesions, and in experimental lesions of the pattern 3, there was a major reduction of complex 4 in the absence of neuronal pathology. So we, for several years, the MS community started to think MS in a different way. What it was pivotal before our studies, it was this knowledge. Here it is the respiratory rate of the mitochondria. And here we see whether this is affected by nitric oxide. And we see that around 250 nanomolars of nitric oxide, half of the respiratory rate is affected, so less ATP. What we saw is that if we decrease the oxygen concentration, only 60 nanomolars of nitric oxide do the same job. So the nitric oxide becomes more potent in inhibiting mitochondria as the oxygen decreases. So there is a kind of synergistic effect. We knew from studies in animal models that the inflamed spinal cord has a 16 millimolars of oxygen concentration. So the idea was that in culture, neither nitric oxide nor hypoxia are sufficient perhaps to kill neurons, but the combination may be lethal. So we decided to look to true hypoxia, whether in neuroinflammatory lesions it was true hypoxia. We knew that there was histotoxic hypoxia, but not true hypoxia, because nitric oxide and reactive oxygen species, they lead to histotoxic hypoxia and energy crisis. The question was then, is true hypoxia present in neuroinflammatory lesions? And the answer was yes. There was a series of experiments when we used pimonidazole which was given IV in these experimental models of MS in EAE, in different models of EAE, which binds to proteins if the pO of oxygen is less than 10 millimeters of mercurium. As you see, this was our model, our baseline, which is the kidney, which is naturally hypoxic, and you see this adductor of pimonidazole. So here is an example of MOG-EAE, one of the models of multiple sclerosis, and the labeling for hypoxia, it was quantitatively related to the magnitude of neurological deficit in EAE, was temporally related to the expression of the neurological deficit, was spatially related to the expression of neurological deficits. We know in the lumbar spine, more inflammation, more disease in the spinal, in the lower part of the spinal cord in the EAE. And it was less hypoxia in remission and more in relapses. If you see, this is the black line is the control, and this is the average pimonidazole labeling, very high in EAE in the first peak of the disease and in relapses. This is a series of experiments to see whether there is inflammation in EAE, and it was, of course. And importantly, the correlation between disability score and PIMO, intensity of hypoxia, it was stronger than inflammation. The hypoxia was present in the gray matter and the white matter. Here we see an optic nerve, and the inflamed optic nerve, this is the normal, the inflamed optic nerve is hypoxic. You may want to remember the trial with the optic neuritis. And here are double labeling studies where we see with co-labeling with PIMO, beta-tubulin, GFAP, suggesting that a number of cells, they are, let's say, they colocalize, they are hypoxic in the central nervous system. The oxygen concentration, these are experiments with oxygen probes in vivo in the animals. Oxygen concentration with the gray matter in EAE drops dramatically. Hypoxia contributes to the neurological deficit, yes, but can we reverse it? Importantly, we could, because when we were able to treat the disease animals with oxygen in EAE, we were able to reverse the disability. And this is after room air and after oxygen in control and EAE in score 7. I will go faster here. When we gave oxygen, but also other agents, like, for example, the MitoQ or other agents that they inhibit nitric oxide, like, for example, this was the 1400W, or the MitoQ, who was affecting oxidative phosphorylation in the mitochondria, then we saw a better result in terms of clinical disability. So going back, tissue true hypoxia is, yes, present in neuroinflammatory lesions. The toxic hypoxia leads to energy crisis, and we put a question mark is tissue hypoxia raises the hypoxic threshold. Why this is important? What are the consequences of hypoxia in the loss of function? Because always we need to keep in mind that the 80-90% of the ATP in the axon goes pumped to keep the ion channels alive. So if the ATP is affected in a way is less, then the first element that will understand the problem of less ATP are the ion channels. And of course, there are a lot of deleterious events that they do happen when the ion channels, they are affected, they don't work properly. One of those is the influx of calcium. So this is a very important thing to remember, that the energy crisis affects a number of elements in the neuron, and first is the electric behavior of the neuron, and this is instant. It's not demyelination, it's longer. This is instant. And now we go back to the previous slide and say, okay, perhaps now we understand a bit more why we used phenytoin, sodium channel blocker, as a neuroprotecting agent in optic neuritis. My last two slides, they say something that perhaps we do know. The central nervous system has little oxygen reserve, and the signaling related energy is 30 moles ATP per gram per minute is the energy used by the leg muscle running the marathon. It's a very expensive tissue, the CNS. These are the conclusions. The conclusions are that the oxygen concentration can be inadequate to maintain normal function. Oxygen is a player. The true hypoxia can be a player in the pathophysiology of multiple sclerosis. And we need to think MS in terms of the pathophysiology in a different way. All this work has been done in the Department of Neuroinflammation in UCL, and I would like to thank all my colleagues for this wonderful journey. Thank you very much.
Thank you very much, Dr. Casti. It was a detailed presentation and a thorough one, so we all got a very good view on the topic. Any questions from the audience first, if any? I don't see something. So again, I have a question that deals with a more qualitative approach. I understand that and the diagnosis of multiple sclerosis is quite trivial at this point, or if I may be able to say that. And depending on this topic, I don't know, but very well, but I guess that we are not we're not expected to see huge innovations in the near future. On the other hand, I know that you're also active in the biomarkers subtopic, sub-area, and I would like to hear your opinion about whether biomarkers are expected to help with diagnosis and prediction of this disease, of these symptoms.
Yes, lovely. Thank you very much. Yes, in multiple sclerosis, we need biomarkers, and to be honest, we don't have a lot. One of the last biomarkers that they are used now is the level of neurofilaments in the cerebrospinal fluid, which looks like that it predicts it is higher, let's say, if there is a lot of damage and brain damage. And it is not a diagnostic marker, but it is a marker that we use during the follow-up of our drugs, of our therapies, of our treatments. However, we are in desperate need of new biomarkers because, as we saw, the disease is very active in very, very early stages. For instance, the clinically isolated syndrome is a person who is totally ambulant, no issues, has had a bit of blurred vision, let's say, for 2-3 days, and then goes back to work. And this particular person has a disease which is going on and on and on, and it can develop in a very debilitating form of disease. So yes, we need new biomarkers, and we are in collaboration with other centers and looking into it.
Okay, perfect. I think we have to see a lot there as well in the future, I guess. Thank you very much for your detailed presentation. I will move forward now to the third presentation, the third keynote address of the day, which is entitled Pathogenic Mechanisms and Novel Therapeutic Approaches for Alzheimer's Disease, which is introduced by Professor Tatsuki Tomita, who is a professor by the Laboratory of Neuropathology and Neuroscience at the University of Tokyo. So, Professor Tomita, the floor is yours.
My name is Daisuke Tomita from the University of Tokyo. First of all, I would like to thank the organizing committee for organizing such a great conference in this difficult COVID-19 situation. As I have never been to Creta, I would like to attend the next conference there and enjoy my stay in Greece. Today I am going to talk about the pathogenesis of Alzheimer's disease and novel therapeutic approach. Dementia has become a major social problem worldwide, and the number of the patients is increasing every year due to the lack of effective treatment and prevention methods. Countries around the world are continuing to build social systems to deal with dementia and to develop drugs to treat the disease. Various studies have shown that dementia is a proteinopathy caused by the accumulation of abnormal proteins in the brain. Abnormal protein aggregation causes abnormalities in neurological dysfunction, chronic inflammatory reactions, and neuronal cell death, leading to a decline in cognitive function. There are several types of diseases that can cause dementia. The major ones are Alzheimer's disease, dementia with Lewy bodies, and frontotemporal dementia. In each of them, abnormal protein accumulation pathology has been observed in the brain, and their constituent proteins have been identified. It is noteworthy that genetic mutations in genes that encoding these accumulated proteins themselves or related molecules are linked to the familial dementia. Especially in Alzheimer's disease, biomarker and brain imaging studies suggest that A-beta accumulates as synaptic plaques and causes the aggregation of tau in neurons, resulting in neurofibrillary tangles that lead to neuronal cell death. In other words, A-beta is considered to be a trigger for the Alzheimer's disease, and tau is thought to cause neurodegeneration. Therefore, approaches to reduce the amount of A-beta in the brain have been developed as a treatment method. However, unfortunately, there are still no approved drugs. In particular, anti-A-beta antibodies have been found to be effective in eliminating signal plaques, but they have struggled to have any effect on cognitive function. However, last year, significant progress was made in aducanumab, one of the anti-A-beta antibodies, and I would like to show about a few of them. In March 2019, Biogen, which is developing aducanumab, halted 2 large phase 3 trials, citing lack of inhibition of cognitive decline based on the results of fluidity analysis during the trials. However, in October, the company resumed the trials, reporting that inclusion of the larger study result showed that the drug was effective in patients receiving the highest dose. Furthermore, at the December meeting, they reported that results of the small group of tau PET imaging results showed a reduction in tau accumulation in the highest dose group, and announced that the FDA will grant priority review of the drug in August 2020. The future development of the anti-A-beta antibody drugs has been the focus of much attention. On the other hand, there are several problems with the removal of A-beta by antibody drugs. The biggest problems are the high cost, the need for prolonged intravenous infusion, and the very low level of penetration into the brain. In addition, the mechanism of action of anti-A-beta drugs is still unclear, and different strategies for amyloid removal are required to be developed. So I would like to show about photocatalysis, an innovative compound that facilitates phagocytosis by microglia. As we all know, proteins fold and form higher order structures based on primary sequence information. However, some types of peptides and proteins undergo successive folding to form oligomers and fibrils called amyloid aggregates. The higher order structure of the amyloid aggregates, which is not primary sequence dependent, is commonly characterized by the stacking of beta sheet structures called cross beta sheets. Amyloid aggregates are generally cytotoxic and are thus responsible for the variety of diseases, not only for Alzheimer's and Parkinson's, but for the several peripheral diseases. We thought that if we could artificially induce a chemical reaction that inhibits amyloid fibrillation, it could be a novel treatment for these amyloid-related diseases. We envisioned that the photocatalyzed aerobic oxygenation is a suitable chemical reaction to attenuate the pathogenic aggregate properties under physiological conditions. Because protein aggregation generally depends on intramolecular hydrophobic interactions, covalent installation of hydrophilic oxygen atoms to a protein would decrease the aggregative property via stabilizing hydrolyzed states. To this end, we are developing photocatalysts, small molecules that are activated by light to oxygenate proteins in the vicinity. The figure shown here shows a compound that is called thioflavin T, a dye that recognizes the beta sheet structure. Usually, thioflavin T has no fluorescence. However, once thioflavin T binds to the amyloid fibrils, the rotation within the molecule can be stabilized to generate the fluorescence under the irradiation. So this is a specific character of the thioflavin T in generating the fluorescence. We modified the structure with thioflavin T to generate the singlet oxygen after the irradiation. When this compound is mixed with a beta aggregate and irradiated the light, it is activated by the stabilization of its structure, and A beta in the vicinity is oxygenated. Importantly, the oxygenated A beta loses its aggregation ability and is unable to exhibit cytotoxicity in vitro. However, in vivo application of this cyflavin T-based catalyst is still difficult because irradiation of visible light at 500 nanometers light, which avers low tissue penetration, is necessary for the catalyst excitation. In addition, the highly specific reaction against the target should be achieved because non-specific reaction might be harmful to the cells by the reactive oxygen generation of reactive oxidation species. To overcome these issues, we have developed a bunch of the compounds and also derivatives of several fluorescent PET probes. And finally, we are now testing these photocatalysts that can be activated by near-infrared light. Because near-infrared light can penetrate into the tissues, we envision that these compounds can be applicable for the in vivo photooxygenation. First, we tested whether these photocatalysts can photooxygenate the amyloid fibrils that is generated in the brain. We mixed the brain lysate from the AD model mice together with the photocatalyst and irradiated by light. Mass spectrometry analysis revealed that the catalyst is capable of oxygenating the A beta accumulated in the brain of the AD model mice. Then we tested the photocatalyst in vivo. By applying optogenetics technology, we injected the photocatalyst and simultaneously delivered light into the brain through an optical fiber to oxygenate A beta in the brain. After the photooxygenation was carried out once a day for 7 days, the brain was removed and analyzed by immunological methods. We confirmed that the level of the A beta in the brain were significantly reduced, especially in the formic acid soluble fraction. Because in formic acid soluble fraction, aggregated A beta fibrils is enriched. These data strongly support our notion that the photocatalyst targets to the amyloid fibrils in the brain of the living mice. However, in contrast to the in vitro reaction using the brain lysate, we cannot detect the oxygenated A beta species by the mass spectrometry. Concerning the result of the total level of the A beta was significantly reduced by oxygenation reaction, we hypothesized that the oxygenated A beta is rapidly removed by unknown mechanism only in living mouse brain. To test this hypothesis, we generated the aggregated synthetic A beta in vitro and irradiated together with the photocatalyst to generate the photooxygenated A beta fibrils. Then we injected the photooxygenated A beta fibrils into the wild-type mouse brain and analyzed the brain lysate after 1 hour of the injection. Surprisingly, we found that the injected photooxygenated A beta species were rapidly degraded in the living mouse brain compared to the non-photooxygenated A beta fibrils. Then we tested the metabolism of the oxygenated A beta mixed with the mouse brain lysate in vitro. However, the degradation rate of the aggregated A beta did not change compared to that of normal aggregated A-beta, suggesting that the photooxygenated A-beta was rapidly degraded only in the living mouse brain. We then consider the effect of microglia, because microglia is involved in the degradation of aggregated A-beta in the brain. When we added A-beta to the cultured microglial cells, we can observe the degradation of A-beta by the phagocytosis and intracellular degradation. Intriguingly, the degradation rate of the photooxygenated A-beta fibrils significantly faster compared to than that of the normal A-beta fibrils. So these data strongly suggest that the photocatalytic oxygenation facilitates the A-beta degradation by microglial cells. To further verify the effect of microglia in the living mouse brain, PLX3397 was used. PLX compound is an inhibitor of the CSF1R receptor, which is involved in the survival of microglia. When we mixed the PLX compound into the tube and treated with the wild-type mice for 3 weeks, we can observe significant reduction of the number of the microglia in the brain. Then we tested the metabolism of the photooxygenated A-beta in the PLX compound-treated mouse brain. As I mentioned, the oxygenated A-beta species is rapidly degraded in the mouse brain. However, the rapid degradation of the photooxygenated A-beta species was significantly abolished in the brains of the mice treated with the PLX compound. These data strongly support our notion that the microglia is involved in the rapid degradation of the photooxygenated A-beta species in living mouse brain. These data indicate that our novel photocatalyst can oxygenate A-beta fibrils selectively with spatial temporal control by light, and the oxygenated A-beta fibrils is rapidly removed by microglia only in living mouse brain. We are still working on the investigation of the molecular mechanism whereby microglia can rapidly degrade or remove the photooxygenated A-beta species. However, we think that the photooxygenation approach would be a novel therapeutic strategy against not only the Alzheimer's but also the several amyloid-related diseases. I would like to show another data about the photocatalyst targeting the different amyloidogenic proteins called tau. Tau and synuclein is an intracellular aggregate that causes the neurodegeneration in Alzheimer's, dementia with Lewy bodies, and frontotemporal dementia. In a similar manner to the A-beta aggregates, tau and synuclein also forms the fibrils that are enriched with the cross-beta sheet structure. Because our photocatalyst selectively targets the cross-beta sheet structure, we hypothesize that the tau and synuclein fibrils also can be photooxygenated by these catalysts. To test the effect of the photooxygenation for the tau aggregation, we incubated with the photocatalyst together with the tau that is made from the E. coli. Recombinant tau can form the fibrils after the incubation in tube. However, when we irradiated the tau together with the catalyst, we found that the fibril formation of tau was significantly inhibited by the photooxygenation reaction. Then we analyze the photooxygenated tau by biochemical method to identify which amino acid residue was photooxygenated by this reaction. We found that the several methionine and histidine residues are photooxygenated by the in vitro reaction. Importantly, some of the methionine and histidines are involved in the formation of the tau fibrils that can be resolved recently by the cryo-EM structural biological analysis. So we hypothesize that the fibril formation can be inhibited by installation of the oxygen atoms into these important histidine and methionine residues. We then further tested the effect of the photooxygenation into the fibril formation of tau using sheaths. Because fiber formation is based on the sheath, that is shorter form of the fibers, we generated the photooxygenated sheath and added the tau monomer in vitro. Usually, tau fibers can be generated after the addition of monomers. However, after the incubation together with the photooxygenated sheath, the aggregation of tau was significantly inhibited. These data strongly suggest that the photooxygenation inhibited the aggregation of tau. Then we analyze the therapeutic potency of the photocatalyst targeting tau. Tau forms intracellular aggregates as a neurofibril tangles. However, unfortunately, our photocatalyst cannot be applicable for the formation of the neurofibril tangles because our photocatalyst cannot penetrate into the cell. However, recently, the tau propagation hypothesis is highlighted. This hypothesis can explain the spreading of the tau pathology into the brain. The process of tau propagation still remains unclear. However, it is hypothesized that once cells are bearing aggregates, then release the seed to the extracellular site. The neighboring cells can uptake the seed and incorporate it in the cytoplasmic site by unknown mechanism. Then the seed induces more aggregation in the neighboring cells. So this is the hypothetical model of the tau propagation. Based on this model, we can target the extracellular tau as a seed that causes the spreading of the tau pathology. So if we can photooxygenate this extracellular tau sheath to inhibit the tau aggregation property, then our photocatalyst can be applicable as a therapeutic against the tau-related diseases. To test this possibility, we analyze the effect of the photooxygenation in propagation cellular model in vitro. This model, using the tau stably expressing cell, application of the tau sheath from the extracellular side can induce the intracellular tau aggregates. So we tested the photooxygenated sheath that is generated in vitro into this propagation model. And we confirm that after photooxygenation, the induction of the intracellular tau aggregates in the cells expressing tau was significantly reduced. So these data strongly suggest that the photooxygenation at the extracellular site also reduced the seeding activity of tau in the culture cell model. We are currently analyzing the possibility whether this photocatalyst can inhibit the tau propagation and the formation of the tau fibrils in the mouse brain. Okay, this is our summary. In conclusion, the several genetic and biochemical researches support the notion that the neurodegenerative diseases are proteinopathy-induced chronic diseases. We developed an artificial catalyst that can be selectively and reversibly turned on or off, depending on the conformation state of the substrate. We also developed a near-infrared light activatable oxygenation catalyst, which enables us to oxygenate A-beta and tau. Application of the photocatalyst diminished the aggregated A-beta in the mouse brain, suggesting that the enhanced clearance of the oxygenated A-beta by microglia in vivo. And artificial protein oxygenation by the photocatalyst would be a novel therapeutic approach against amyloid-related diseases, not only Alzheimer's, but also the dementia with Lewy bodies, frontotemporal dementia, and peripheral amyloid-related diseases as well. I would like to acknowledge my several collaborators, especially Dr. Motomu Kanai and Yohei Soma, for providing us such wonderful chance to collaborate related to the photocatalyst. Actually, the photocatalyst strategy was originally made by the Drs. Kanai and Soma. We would like to thank the collaborator for the Takashi Saito and Takami Saito for the providing the AD model mice, Toru Fukuyama and Satoshi Yokoshima for the providing compounds. This photocatalyst story has been led by Dr. Yukiko Hori. She is the instructor of our laboratory. And the photocatalyst against the A-beta species has been performed by the Mr. Shuta Ozawa, and the photocatalyst against tau was performed by Mr. Yu Nemoto and Takanobu Suzuki. I would like to thank the funding agency for JSPS and AMED. Thank you for your attention.
Okay, I would like to thank Professor Tomita for his great presentation that allow us to familiarize ourselves with the topic at hand. And since we are running a little bit late, I would like to shift forward to the fourth presentation, the fourth keynote address of the day, which is entitled Motor Learning as a Model for the Use of Dance in Rehabilitation, which is going to be presented by Dr. Rebecca Barnstaple from York University, Department of Dance in Canada. Dr. Barnstaple, the floor is yours.
Thank you very much, and thank you for having me here. I'm really honored to be part of this gathering. I'm
afraid we can't hear, Dr.
1 second. Can you hear me now? Is that better? Yes? No? Hello, you hear me now?
Okay, perfect. Thank you very much.
Excellent. Yes, thank you. So thank you very much for this invitation, and I am going to now share my screen. Great. Okay. thank you very much for inviting me to be here. I'm really thrilled to be part of this gathering. My research is really sort of a view from very high up of some of the things that people have been discussing from a more, much more close-up range. And I would really like to invite the researchers who are here today, whether you're listening now or later, to maybe take an opportunity to think with me about some of the interesting questions underlying this emerging field of dance in rehabilitative research. I also wanted to précis my presentation by sharing that I work in a community health setting as a clinician. So it's been very interesting hearing people's presentations that relate with some of the groups that I work with. So through dance, I've worked with people with multiple sclerosis, Parkinson's, chronic pain, and I really try to apply my understanding of how the brain works to how I work with people. So maybe we can be mutually inspiring to each other. So I'm going to give you a quick overview of this field of dance in rehabilitative research and some information from the lab that I work with at York University in Canada before diving into some research we conducted last year in Berlin that really tried to get at some of the mechanisms behind the observed effects of dance that are associated with neuroplasticity. So in 2015, our group first published a paper that was exploring the hypothesis as to how dance could be used effectively in rehabilitation. And what was put forward at that time was this idea that dance really fuses cognitive and physical rehabilitation, and drawing on the literature from music rehabilitation and music therapy, and also exercise, which has quite a lot of literature over the past 15 years that is showing that exercise supports a healthy nervous system. And I wanted to bring this up because one of the things that we explored in this paper was thinking about the idea that in animal models, we have seen that not only exercise, but exercise in an enriched environment is more conducive to neurodegeneration and the maintaining of new neurons. If there's an enriched environment, there's more support for the continuation of neurons that are created through exercise. And this was the beginning for me, as someone who works in the field of dance, to think about the idea that what is happening when we provide a dance-based intervention for people is that we're really offering and creating an enriched environment. Because dance often includes social elements, it includes musical elements. People are moving in time with each other and in time with the environment that has been created for them. And definitely in a practical way, it got me thinking about how and why dance specifically might be fostering neuroprotective factors for older adults. I wanted to point to some research that's come out recently that supports this hypothesis. So this is from a group in Magdeburg in Germany, and they really dove in to see if they could determine some of the effects of dance in comparison to exercise. And I wanted to bring this up because it's a very nice study design in that they actually split the group in half, and it worked like an RCT, and they were very specific about what happened in the exercise group versus what happened in the dance group. And they tried to match in terms of the actual physical output of people in each group. And I think this is very important because what I'm going to talk to later in the presentation is what is it specifically about dance that might make it something more than exercise that can be offered to people? What is the difference? So they actually found, in comparison to their dance group versus their sports group, that the dance group had a higher increase in BDNF, which is a key marker of neuroplasticity. And this is a really nice finding to think about as we move forward. They also saw changes in volume within the precentral gyrus and the parahippocampal gyrus. And it was nice because they kept this particular intervention running for a pretty long period of time. So they looked at 6 months and they looked at 18 months. And some of these findings were only in evidence at 18 months. So this is something else to think about, is, you know, in terms of creating a supportive rehabilitative environment, what is an optimal length of time? Is there a difference between someone attending a program like this for 6 weeks, or is it something that should be offered on an ongoing basis? And how are these changes fostered that really maybe take a lot longer to emerge? In the lab that I am associated with at York University, which is Joe D'Souza's lab, we started investigating this a few years ago, starting with expert dancers before moving to a clinical population. So we thought about this idea about what is underlying the changes that can be seen. And we started looking at a model that involves motor learning, because one aspect of dance is learning and remembering motor sequences and being able to reproduce them. So this was research that was done with a group of expert ballet dancers. They were students, they were young and healthy. It was an fMRI study. And what we did is they were put in a scanner, and at several points, they were scanned first when they had heard the music, but they had not yet learned to dance to it. Then after 1 week of learning the movements, at 7 weeks when they were learning them and they knew them fairly well, and then at 34 weeks, at which point they were performing on a regular basis. So these were like very learned movements. And we saw a really nice curve here in the SMA in terms of activation patterns. So we have increasing activity at 1 and 7 weeks, and then that activation pattern drops out at 34 weeks, suggesting that there is some underlying plasticity that is in evidence. We wanted to do the same study with people with Parkinson's, because National Ballet School in Toronto is the host to an ongoing dance program for people with Parkinson's. And we have seen some other measures that suggest that it is benefiting them, but we wanted to see if we could actually detect a brain signal change. So we did the same exact setup with people with Parkinson's over 8 months to see what we could find. And we saw, again, this increasing activation in the supplemental motor area from the initial acquisition of the movement when they first learn it to when they're practicing it a little, practicing it a lot. And we just finished looking at like a first parse through this data. And once again, we see this activity drops later on. So it does show that, once again, there's maybe some sort of network reorganization associated with learning and really knowing this series of movements. This is, I think, a nice thing to think about, because this is people coming to a dance class 1 time per week for 1 hour, and they're in a group. So it's a very accessible type of therapeutic that could be available to people that may be helping facilitate adaptive plasticity. We also measure UPDRS, United Parkinson's Disease Rating Scale, for anyone who doesn't know this one. And this is, we've been following people for 3 years now who've been attending these classes. And the yellow line is the slope. Once we average together, those are points of measurements of UPDRS for people who have been attending the class over the 3 years. And we found that the people who danced on the yellow line, their slope is very, very low. It was almost 0 in comparison to a group of controls that we gathered from the Michael J. Fox database. And we tried to match for types of activity, but these were people that did not dance. And there's a difference here. And as you can imagine, controlling diseases like Parkinson's is a burden to the healthcare system. And if we can, again, find something that's in a therapeutic like this that could help slow disease progression at a very reasonable cost, I think it's a very exciting thing to think about and investigate. Some of the other things that we've measured, and this is also things that are in a lot of literature and that are more maybe obvious when you're thinking about dance, is that there are improvements for people's balance. There's improvements in timed up and go, so time to come to standing. So these like motor measures, it's kind of expected that they would improve, and they do indeed improve, even in people. This is, again, Parkinson's data, so even with people who are living with a motor disorder. And we also look at UPDRS measures from pre to post in a single class, and we see a difference there. We also measured within a shortened time frame of people dancing every day for 2 weeks. We gave them the weekend off. And we see, again, this improvement in people's UPDRS scores over that period of time. So that's all interesting. In addition to that, with the ballet school, we have been collecting just resting EEG with people before and after class, seeing if there's any kind of signal change that we can detect on site with a very simple 14-channel EEG setup. It's just an emotive headset. And we have consistently found that there's an increase in frontal alpha power, more particularly for the people with Parkinson's from pre to post class. I think this is a very interesting effect. And as I was coming into my PhD, I really wanted to understand more about how and why this might be happening in a more situated setting. So that's all my background. Before I go into the experiment I want to describe, I also want to say that the group that I told you about in Magdeburg has also produced a very nice hypothesis that I thought might make sense to some of the people watching this lecture, that what is interesting about dance is that you can think of it as a form of simultaneous motor cognitive training. So as opposed to something that is sequential, like there's already evidence that motor cognitive training together is helpful for people. In a simultaneous motor cognitive task, the cognitive task actually maybe facilitates the motor task. So in dance, whether it's learning a series of movements, a sequence of movements, or even like setting your movement to the music or understanding a technique, the types of things that you're thinking about and how you're modulating your attention are going to help you in the execution of the motor task. And I think this is a very nice hypothesis for thinking about how and why dance may support the nervous system. So I went last year to Berlin to work with a group there that's at the B-Mobile Lab because all of this is very interesting emerging research, but what is not understood really are the mechanisms underneath why dance is perhaps effective in slowing something like Parkinson's disease. So I wanted to have access to the type of research they do at this lab, which is mobile neuroimaging, to look at what happens while people are engaged not only in dancing, but in the process of learning a novel motor sequence. So this lab is very nice, high-tech lab. They have a mobile EEG headset with 128 channels, which was much more than I had before. They have motion capture technology, and they also usually use VR with their setup. I did not use VR. Oh, and yes, this is the experiment design I'm going to tell you about is published in EJN. So if you're interested, you can check it out. Instead of using VR, what I did for my research is I actually choreographed my experiment. We set it up with 12 different phases, and in which we were always collecting data continuously. So we started and ended with a resting state to address the question that came up in our previous research about this bump in alpha power. We had the subject perform a spacewalk, because one of the things that is very important, I think, potentially about dance is that dance is spatially oriented. And it is spatially oriented by the participant. So rather than orienting to an object like a stationary bicycle or some kind of exercise equipment, the spatial orientation happens through the process of learning the dance and is refined over time. So to investigate signal change with that, we had the subject walk a predetermined path through the room that they did the first time, naive to what that path would be, and then they repeated it, learning it over time. And we repeated this again at the end of the experiment once the subject had been performing a dance within this space. We tried to replicate the setting and context of a studio. So after this walk, which functioned as a first warm-up, we did like a 4-minute warm-up where we moved different parts of the body progressively until we were moving the whole body in space. We showed 2 videos, one of the choreography that the subject would learn and one of a different choreography that was matched in terms of types of movement and the music and things like this. And they didn't know which one they were going to learn. The subjects then watched the choreography 6 times. It was a 30-second choreography, and it was somewhat dictated by the equipment. We couldn't do anything really with a lot of jerk. Everything had to be very smooth, but we tried to have flexion and extension, one-legged support, things you might find in many different types of dance forms without it being any specific type of dance form. So they watched a live performance 6 times. They then did it with the teacher 6 times. That was me, I taught it. Then they imagined doing it without moving in space. And I should also say in the learn condition, it actually was a very variable window. We went up to 20 trials, and some participants who had previous experience were able to master it within, I think, 4 trials. And people who are new to the idea of dance, it took them closer to 20. When the subject was satisfied that they knew the piece, and we also had an independent rater assessing their accuracy, once they got to about 80% accuracy and felt comfortable, they would imagine performing the dance while being still. And so this was in some way replicating what would be conducted in some of the fMRI studies, where the person is hearing the music and imagining performing the dance without actually moving in space. We then had a performance where the person performed the dance 6 times in space. And then once again, they watched the videos twice, each one. And now one would be very familiar, one that they have just learned this choreography and performed it a number of times. And the other one would remain unfamiliar. It's the same one they saw at the beginning that they haven't seen since that point. And we did the spacewalk again and a resting state on the way out. So we have a lot of data here and trying to address a number of questions within the literature. So we have some questions as to spatial orientation and spatial coding. There's some questions we're trying to address here as to like watching a video versus watching a live performance, and trying to collect data as the person is actually moving through space, from learning to becoming proficient at a brief sequence of movements that is affixed to music. So some of the questions that we had, I mentioned a couple, but here are a couple more. We really wanted to try and get at motor learning through this kind of setup, to see like how does motor learning affect brain dynamics both at rest and moving through space. We were interested to see if we can see any evidence of short-term plasticity, referring back to the literature that showed some more like long-term plasticity in ongoing studies. What is the difference between watching, doing, learning, visualizing, and performing a sequence of movements? What's the difference between watching a live person versus watching a video? Most of the dance research that's been conducted so far has been conducted with video stimulus, just because of the difficulties of doing real-world research. This is one of my pet questions, but can a dance, does it create an environment? Is similarly to, I said this lab often uses virtual reality to create their environment. We choreographed the environment. So this is, I can talk more about that if anyone's interested later. And I also asked each subject throughout the experiment, after each trial of the performance, to what extent they felt like they were fully dancing. And this is, again, some of the data I was most interested in, because I think there is a big difference between simply moving to music and going through the motions, so to speak, as opposed to becoming fully invested in the world of a movement, and whether that is a cultural world, as you might find in a folk dance, or a world that is like a choreography that is performed, but the person has had the time to really infuse the movement with their own sense of it and their own manner of performing it. This is 30 seconds of what we did, just to give you a sense of what it looks like. You can see the subject there behind me with a big setup on. It took about an hour to prepare each subject, and there I am in the front. So I'll just play it so you have a sense of what I'm talking about here. Okay, so you can see that was like a trial 6 or so, and the subject at that point had a pretty good sense of the movement, and they were able to replicate it fairly well, but they're also watching me a lot. They were a little bit correcting themselves as to the timing and things like that. I should also say that we specifically created music for this that we hoped would be low in emotional valence, because I was trying to separate this process of motor learning from some of the more emotion-laden associations that dance has. So we actually had the subjects rate the music before and after dancing to determine like what the effective impact was. And interestingly, it was usually very low at the beginning of the experiment, and it increased specifically for the piece of music that they learned the movement sequence to. This is from the motion capture data. We just had 5 pucks on each person. I'm the red person, the subject is green, but you can see we get some nice data here showing the movement. So we will be able to do some synchrony measures, I think, between the subject and myself, and there's also potential for us to look at some synchrony with the neuroimaging data. So I haven't analyzed this data yet. This concluded right before COVID shut everything down, and I am still determining ways to access my data and work with it, because I obviously cannot go back to Berlin right now from Canada. But one of the things, this is just a quick independent component analysis. This is not really totally cleaned up or anything, but I wanted to show this one because this was one of the sources that we isolated, and it looks like it could be pretty close to SMA. So this is interesting. Anyway, we have so much data, and I will probably be analyzing this for the rest of my life. If there are enough interesting questions, which I think there potentially are. So just as a final couple of slides, just to wrap up where I've taken you on this journey and where we're going. The Parkinson's research started in 2013 through a grant to the lab to train some people at National Ballet School to teach the Dance with Parkinson's program. That was when we did this early neuroimaging data that I showed you with people with Parkinson's. And the classes have been going since that time. We have our more than 3 years worth of data on things like UPDRS. And this was just last year that we conducted this study in Berlin with 20 subjects learning this dance individually. And I want to say, to acknowledge that, of course, I do not do this research alone at all. I am part of a group of people at York University. The head of the lab is Joe D'Souza, and there are many people who contribute to the work of the lab there. And that's all I have to say right now. So I would love to take questions or have a conversation if anyone would like to. Thank you.
Thank you very much, Dr. Barshapel. Your talk provided certainly another dimension or another perspective on the bioinformatics territory, domain that we are discussing in this conference for a couple of days now. It was very interesting to myself, of course, and hopefully to the audience. Any questions from the audience to start with? If any. We don't have any questions yet. In this case, I would like to pose a question that regards to your personal experience, maybe. It is clear that physical rehabilitation based on motor learning strategies, or how this subdomain is called, is clearly aids clearly the process. The question is, from your experience, does this aid is this aid applicable to all neurodegenerative diseases up to the same extent? Is it better in some cases where the disease itself provides some different input for the patient? And how does this go from your experience?
Yes, it's a very nice question. So one of the things I would like to say, one thing that motivated me to do this kind of research is with the dance program for people with Parkinson's. It contains always aspects of learning a choreography. I'm also part of another group that is doing dance-based research on people with MCI, mild cognitive impairment, like beginning of Alzheimer's, and their care partners, and that is using all improvisational movements. So it's a very different approach. And I think it is important for us to understand, and as someone who's working clinically with some of these populations, what is the contribution of these different modes of working? Because you can imagine that learning a choreographic sequence involves, remembering, spatially orienting, all of these things that I talked about. But in improvisational movement, there are other aspects like responding within a certain period of time to a prompt or an idea, responding creatively, pushing yourself to explore different ways of moving or different types of movement. So it's one of the things I'd really like to understand. I don't believe there is an answer for your question at this point, but I think having a better understanding of the contributions of the different ways we can work with dance could help us to develop better therapeutic applications.
Yes, of course, it's more than a topic for discussion or for future research. Continuing as a second question, did you also see patients suffering from the same disease respond in a different manner to the strategy applied? Meaning that maybe the personalized or the personal skills or other elements of each patient play a specific role to receiving this treatment?
This is also a good question. One thing I can say as a clinician that I find very interesting is that the less experience the person has with dance, the more potentially helpful it seems for them. So people who come to a program who maybe have never danced before, they never thought of dancing before, and it is entirely novel to them, are learning things, learning strategies of thinking about how they move, where they move, that I think maybe facilitate this process even further.
Okay, it's a very interesting field of research, and
I think
glad that you were here. Yes, of course. Dr. No.
Yes. Actually, it's a very interesting subject, and I'm quite fascinated about research. So the fact that you saw some this brain activity through the MRI and your EEG study, is it in any specific brain regions, and or just general this prefrontal cortex or some specific gyrus always show up as most affected area?
Yeah, in the fMRI research, we were really looking at SMA, supplemental motor area. There's, of course, activity in the auditory cortex as well, as you would expect when people are having an auditory input. But with the EEG research, we're also looking at changes mainly in the frontal area. So it's frontal alpha, and that is also what it looked like we saw when we have this bigger setup that we can get input from more electrodes. But I have a big learning in front of me to learn how to do more things with source localization and associations between electrodes, because I would really like to be able to do network analysis with some of this denser data that we have. The group that I have done some work with in the United States that's doing work with MCI has seen changes in specific networks like the default mode network and the salience network that have been associated with participation in like an 8-week improvisatory dance program.
Thank you.
Thanks for your question.
Okay, thank you very much again for the presentation, and I'm really glad I had all 4 of you in this session, and we heard and also familiarized ourselves with quite diverse, but at the same time very innovative and very inspirational presentations and the research work. Please keep up the good work. Thank you very much for presenting in this conference, and this concludes the session. There is a short break, and then the next one continues. Thank you very much.
Thank you.
Hello, everyone. Welcome to the Saturday afternoon session of GeNeDis 2020. My name is Ilias Kotsirias, and I have the distinct pleasure to chair this session. We have 4 keynote speakers in this afternoon session, and we will start with the first keynote by Professor Christos Plakiotis. His title is The National Mental Health Strategy, Past, Present and Future Directions in Mental Health Policy in Australia. Professor Plakiotis, you have the floor, please.
Hello, my name's Chris Plakiotis. I'm a psychiatrist specialising in both adult and old age psychiatry, and I'm the unit head of the Aged Persons Mental Health Service at Monash Health and an adjunct senior lecturer with the Department of Psychiatry at Monash University, both of which are based in Melbourne, Australia. I'd like to thank the organisers of GeNeDis 2020 for their kind invitation to present at this year's conference. I'm very pleased that the conference is going ahead, even if I can't be present in person to see you all in Heraklion, as was initially the plan. And today I'll be talking to you about a health policy related topic, and in particular Australia's National Mental Health Strategy, which has helped to shape the delivery of mental health care services in Australia for many years now. I'm going to begin by providing an overview of the policy, as well as discussing the background and setting against which it was developed. It's important to appreciate that the National Mental Health Strategy is a strategic health policy of the Federal Government of Australia, and it's currently comprised of a number of documents which include the National Mental Health Policy of 2008, the 4th National Mental Health Plan of 2009, the Mental Health Statement of Rights and Responsibilities of 2012, as well as the more recently released 5th National Mental Health and Suicide Prevention Plan of 2017. The development of the National Mental Health Strategy is an important milestone in the history of mental health service provision in Australia, because prior to its existence, state governments had been entirely responsible for mental health service provision within the country. It's also important to note that when the National Mental Health Strategy was being developed, an important inquiry took place, which was the National Inquiry into the Human Rights of People with Mental Illness, commonly known as the Burdekin Report. And this inquiry and the report that stemmed from it would have been important in shaping the direction of the National Mental Health Strategy. The National Mental Health Strategy represented a pledge by all levels of government within Australia to enhance the lives of individuals with a mental illness by making available to them better supports and services, preferably within the community, as opposed to hospital or asylum settings. In fact, criticisms of the earlier asylum era in the 1980s and 1990s were identified as having contributed to the policies of deinstitutionalisation, to the mainstreaming of psychiatric services within the general hospital sector, and to the integration between inpatient and community care components of mental health services. These changes had actually commenced prior to the National Mental Health Strategy. However, they had done so in an uncoordinated manner. So the introduction of the National Mental Health Strategy may have represented an attempt by governments to provide an overarching framework to progress these changes. When the National Mental Health Strategy first came into effect, the federal government allocated $189 million to states and territories for service remodelling, and an additional $80 million for nationwide coordination and major national projects, which included improved mental health training for general practitioners, the establishment of a national mental health minimum data set, the provision of funding for basic medical and health services research, an initiative to improve community mental health awareness and reduce stigma and inequality. Funding models were developed that were responsive to consumers' needs. Outcome measures and national standards were also developed for service performance. Improved services for non-English speaking people were established. A nationwide survey of mental illness prevalence and related disability and service use was also set up, as well as grants for service innovation. It's also important to appreciate that the National Mental Health Strategy doesn't operate in a silo of its own, but that there are in fact a number of factors and players which impact on and constrain its ongoing implementation, both in the state of Victoria and in other states from a legislative and policy perspective. And these include the Victorian Mental Health Act of 2014, which superseded the earlier Mental Health Act of 1986. There's the Charter of Human Rights and Responsibilities Act of 2006, as well as the recent Victorian Medical Treatment Planning and Decisions Act of 2016. We also need to take into account the Office of the Chief Psychiatrist of Victoria, which provides guidance to mental health services about the provision of high quality clinical care, as well as the Mental Health Tribunal, which oversees the provision of compulsory treatment in the state of Victoria. There's Victoria Legal Aid, which provides free legal services to patients who interact with the mental health sector should they require such assistance. There's the National Safety and Quality Health Service Standards, which must be taken into account. And previously there were also the National Standards for Mental Health Services of 2010, but these have now been effectively superseded by the previous standards. The National Disability Insurance Scheme is also of relevance, and I'll be talking a bit more about that later. The National Mental Health Strategy was developed as a framework to guide mental health reform, and it was initially endorsed in April 1992 by all Australian health ministers via the Australian Health Ministers Conference. In order to provide consumer and carer oversight of how the National Mental Health Strategy was progressing, a National Community Advisory Group on Mental Health was created in 1992 to report to the Department of Human Services and Health in this regard. However, despite the claimed emphasis on the consultation process that was purportedly followed, both within the National Mental Health Strategy documents themselves, as well as within the scientific literature, it's my impression that accountability, transparency, and documentation of this process is actually lacking. In terms of how the National Mental Health Strategy fits into the healthcare system and interacts with the broader policy environment, it was pointed out by Singh and McGorry that the first National Mental Health Plan constituted Schedule F of the Medicare agreement, which was applicable at the time. The second National Mental Health Plan was also intended to represent a schedule of the Australian Healthcare Agreement that would replace the Medicare agreement. In recognition of the need to have an integrated approach across a range of government departments through intersectoral policy reform, there was a meeting in early 1994 of health, community services and housing ministers, which approved the establishment of a national body to coordinate mental health services when defined from a narrow healthcare perspective, but also disability support more broadly under a community services portfolio, as well as accommodation under a housing portfolio. Next, I'll be considering the values and drivers underpinning the policy and the stakeholders who have an interest in it, and I'll also be commenting on its effectiveness. In the 30-year period prior to the National Mental Health Strategy being introduced, an agenda to reform the treatment of people with mental illness was noted to have taken hold in many developed countries. The National Mental Health Strategy is intended to benefit all Australians who are affected by mental illness by ensuring that they receive high quality care and are assisted to move towards recovery. This is important because severe mental illness can be very debilitating indeed. It can prevent those affected from functioning and living independently. It can place a significant burden of care on families, and it also has significant financial implications for society as a whole. It's therefore likely that the strategy was developed with all these issues in mind. The fourth National Mental Health Plan identified stakeholders that were relevant to its various priority areas, and these included funders, service providers, consumers and carers, as well as other stakeholders. In a review of the strategy undertaken by Singh in 2003, it was identified that consumers and carers had been involved in all of the national planning bodies that had been formed subsequent to the strategy's commencement, and that the mental health sector led the health sector as a whole in the area of consumer and carer involvement. In terms of the effectiveness of consumers and carers in advocating for mental health and in influencing policy development, I think it's certainly the case that they're given considerable opportunities to have their voices heard within the mental health sector, but that professional groups, health services and government departments still retain a dominant role in interpreting consumer and carer views and distilling these into policy. In 1998, Singh and McGorry reviewed the effectiveness of the National Mental Health Strategy in its first 5 years. They noted a 24% total increase in mental health service funding and a 55% increase in community mental health service funding since its inception. However, progress and service reform were uneven across states and territories. Furthermore, in their analysis, the authors asserted that patients with psychotic illnesses in particular now found themselves being treated in a less stigmatizing and more humane way as a result of funds that were previously provided to psychiatric hospitals now being channeled into community services via ring-fenced mental health budgets. The above notwithstanding, I would make the point, however, that the Victorian public mental health sector is still very much a psychosis service that markedly lacks inpatient beds, which are available only to the most unwell of patients. And it's also understaffed, both in the hospital setting and in community clinics. Furthermore, despite the clear rises in mental health service funding, with its redistribution from psychiatric to general hospitals and the community sector, there's little evidence that better quality services have actually resulted. Despite the good intentions of the National Mental Health Strategy, people haven't always been convinced that these intentions have been achieved. Ray and colleagues in 2004 reported that the perception that public psychiatry services had deteriorated was greatest among those who were the farthest removed from their administration. Whiteford and Buckingham in 2005 reassessed the impact of 10 years of mental health service reform since the inception of the National Mental Health Strategy. In the face of negative media attention, several national inquiries into mental health, and increasing professional unease regarding the state of the Australian mental health system. The authors asserted in their paper that concerns raised by increasingly vocal consumers, carers, and healthcare advocates regarding access to mental health care, alongside a surfacing of unmet need and rising service demand, were all attributable to an insufficient rate and scale of reform. The authors suggested that a solution could be found in an improved understanding of whether the 65% increase in mental health expenditure since 1993 was providing good value for money, and in ensuring the efficiency of the public mental health sector. Rosenberg and Hickey recently compared Australia's performance in the mental health field and its wider social context with that of other nations, looking at particular at issues related to mental health care spending, rehospitalisation, the excessive use of antidepressants and treatment for depression, and community mental health service access. They found that despite being one of the earliest nations to have a comprehensive national mental health strategy, Australia performed poorly in comparison to other nations across all of these domains, prompting the conclusion that no gold medals deserved to be awarded. It's also worth noting the sobering Not-for-Service report, which came out in 2005, and which clearly revealed a mental health service that remained in crisis through the views of consumers, carers, and other relevant stakeholders, such as police and prison officers. In the next section, I'll be looking at the factors which impact on the achievement of the goals and outcomes of the National Mental Health Strategy. In 2006, Rosen described how the National Mental Health Strategy had driven deinstitutionalisation via a natural experiment between states to translate the policy into practice. In my view, governments have distorted the aspirations of those with mental illness and their families to live independently in order to promote community living as an alternative to hospital and to achieve deinstitutionalisation, while at the same time systematically failing to invest in the community-based resources that are needed to provide good quality care. Hickey and Groom commented in 2004 on the commitment of Australian governments to the 5-year National Mental Health Plan 2003 to 2008, criticising the plan for not obliging governments to deliver on any particular items within a specified time frame. The authors asserted that the plan had not been influenced as they had hoped by their earlier out of hospital, out of mind report, which had systematically surveyed consumers, carers and healthcare providers and had identified a common failure to adequately invest in community mental health care provision as promoted in the first National Mental Health Plan. Other recommended actions included improved funding, national data sets regarding service provision, the strengthening of input from non-government organisations, and consumer and carer evaluation. One area in which progress has been made is that of suicide prevention, with the inception of a separate national suicide prevention strategy in the year 2000. Jorm asserted in 2014 that a reduction in the suicide rate from the late 1990s, corresponding to the introduction of the National Suicide Prevention Strategy, rather than the National Mental Health Strategy per se, is the only demonstrable improvement in mental health outcomes in Australia in the 20 years since community service provision has increased. The development of a national prevention strategy for mental disorders has been proposed from a population health perspective as a means of redressing this balance. It also appears that the above ideas have gained some traction in recent years, with the introduction of a combined 5th National Mental Health and Suicide Prevention Plan in 2017. So how can the deficiencies of the National Mental Health Strategy be addressed and its implementation enhanced? As I indicated earlier, mental health services in Victoria have been systematically underfunded for many years. For example, Victoria has a much lower number of acute psychiatric inpatient beds per head of population relative to most other states and territories in Australia. If we're truly serious about improving the situation of people with mental illness in this country, it would be helpful for governments to take the bold step of defining the lowest acceptable ranking for Australia in international comparisons of mental health services. And this is important because despite the level of advocacy for improved service provision within the mental health sector, mental health patients may be less capable than patients in other areas of health to be able to effectively stand up for their rights against government and healthcare services. A key objective should be to increase and ring-fence mental health funding to ensure that Australia can reliably improve its position in international rankings of mental health service provision. And this would require appropriate resource allocation per head of population across a variety of domains. For example, in psychiatrists and mental health clinicians of all disciplines, in outpatient and community clinics, in short, medium and long stay inpatient beds, in residential care accommodation and in housing options, and in educational and occupational opportunities. It has been suggested that the collection of benchmarking data by mental health services is an important first step in service improvement by allowing inter-service comparisons both within Australia and also internationally. Factors that contribute to the causation of mental illness and to the well-being of those affected are manifold, and there's no guarantee that the above measures will easily result in measurable improvements in outcome in some areas. Furthermore, and sadly, improvements in mental health service provision at this stage may be too little too late for the many individuals who have already committed suicide without coming into prior contact with mental health services. In 2004, Rosen and colleagues argued that a long-term National Mental Health Commission, similar to that which operates in New Zealand, should have been established alongside Australia's National Mental Health Plan 2003 to 2008, which has since been replaced by the 4th and 5th National Mental Health Plans. On a positive note, it appears that these calls have indeed been heeded, with the establishment of a National Mental Health Commission in 2012 to advise both government and the community about what is and isn't effective in the area of mental health service provision. So where to from here? In the next few slides, I'll briefly be examining a number of topics that are relevant to mental health policy and practice in Australia, and which offer the potential to deliver even better mental health services than those achieved under the National Mental Health Strategy to date. The establishment of the National Disability Insurance Scheme from 2013 onwards has been a major development in the provision of disability support services in Australia. From the perspective of patients with a mental illness, the NDIS funds disability supports that are not clinical in nature and that focus on a person's functional ability. And these include supports that enable a person with a mental illness or psychiatric condition to undertake activities of daily living and participate in the community and social and economic life. The Productivity Commission is an Australian government advisory body that is concerned with the economy as well as other societal and environmental issues. It has recently released a draft report on the mental health sector in which it identified a number of key factors driving poor outcomes in this area. These factors include an underinvestment in prevention and early intervention, clinical services that overlook other determinants of mental health, difficulties in finding and accessing suitable supports, the provision of supports that fall well below best practice, a persistence of stigma and discrimination in relation to mental illness, and a lack of clarity across the tiers of government about their various roles, responsibilities, and funding, which lead to problems with limited accountability. To overcome these difficulties, the Commission has identified a number of reform areas in its draft report, including prevention and early intervention for mental illness and suicide attempts, closing the critical gaps in healthcare services, investing in services beyond health, assisting people with mental illness to get into work, and enabling early treatment of work-related mental illness, and fundamental reform to care coordination, governance, and funding arrangements.
Okay, thank you. Thank you to Dr. Chris Plakiotis for his talk. We will continue on with the second keynote in the program, which is given by Professor Katerina Akasoglu from University of California, San Francisco. Her title is Neurovascular Interactions, Mechanisms, Imaging, and Therapeutics. Dr. Akasoglu, you have the floor, please.
Thank you. I'm Katerina Akasoglu. I'm a senior investigator at the Gladstone Institute and a professor of neurology at UCSF. And I would like to thank the organizers for the opportunity to present our work. Research in my lab focuses on neurovascular interactions and their contribution to neuroinflammation and neurodegeneration. And today we'll be presenting some exciting new studies on how changes at the neurovascular interface link to toxic neuroinflammation in the brain. These are my disclosures. I'm a co-founder of a university spin-out and a named inventor on patents and patent applications, some of which are related to this work. The normal brain is a very highly vascularized tissue, and here you can see a three-dimensional immunolabeling of a cleared control brain. And the main function of these blood vessels is to maintain a normal blood-brain barrier, and thus not allow toxic proteins from inside the bloodstream to extravasate into the brain parenchyma. This changes dramatically in the context of disease. Here you can see a cleared brain volume from a patient with Alzheimer's disease, and you can appreciate this dramatic change both in the structure of the blood vessels, but also this extravasation of blood proteins, and this is immunolabeling for the blood protein fibrinogen. And a key driving question in my lab is, what is the role of the leak of these blood proteins into the brain in the context of neurological disease? And indeed, traditionally, neurological diseases have been categorized in distinct categories as only autoimmune, or only vascular, or only neurodegenerative. But we now know that evidence from epidemiological studies identified that changes in blood-brain barrier disruption and vascular permeability are really a common thread, a common pathogenic thread among these multiple diseases. So the key question that appears is this chicken and the egg question. Is there more leak of blood in these diseases because the disease is worse? Or is it possible that these blood proteins might be causally linked to the neurologic disease? And in my lab over the past 20 years, we have identified the blood coagulation protein fibrinogen as being both necessary and sufficient to mediate several of these neuroinflammatory, neurodegenerative processes in the brain, be a critical mediator that controls the communication between peripheral immune responses with the brain, and also being a rich niche both for the development of imaging tools, as also the development of new therapeutics, both for targeting these processes in the CNS, the development of new animal models, as well as biomarkers. And there are three aspects of fibrinogen that really set it apart among other blood proteins. First, it's abundantly deposited in neurological diseases. And here you can see neuropathology in the human brain, in multiple sclerosis, traumatic brain injury, as well as Alzheimer's disease. Second, fibrinogen is converted to insoluble fibrin. It is a soluble molecule in the blood. It's a large molecular weight protein that with the action of thrombin is converted to this insoluble molecule. And we know that fibrin plays a role in coagulation, inflammation, as well as repair. But these processes had not been studied in the brain in the context of neurologic disease. So I became really interested to find what is the role of this in the context of brain diseases. Third, fibrinogen is a druggable target. Here you can see the gamma chain of fibrinogen, where you can see the 400, 411 binding site to the platelet receptor. And there is indeed a monoclonal antibody that blocks the interaction of platelets with this coagulation domain of fibrinogen. What I noticed was that fibrinogen has another domain, a cryptic epitope, the 377-395 epitope of fibrinogen, that binds to a different receptor, the inflammatory complement receptor. So a driving question in my lab is it possible that we can selectively block this potential damaging inflammatory functions of fibrin without interfering with its protective roles in hemostasis? And indeed, in the lab, we have taken both genetic and pharmacologic approaches to target this inflammatory domain of fibrinogen and explore its functions in the brain. This is a slide that shows the studies over the past 40 years that have identified the causative role for fibrin in different models of neurological diseases. And our lab performed foundational studies originally in fibrinogen knockout mice in 2002, and later in the fibrinogen knock-in mice, which are mice that have a specific mutation in the inflammatory fibrin domain that reduces paralysis, inflammation, microglia activation in the EAE model, and provide the first genetic evidence that by mutating the specific epitope on this molecule is sufficient to suppress neurologic diseases. And this later on became the foundation for our pharmacological approaches for selective targeting of this protein in the context of neurodegeneration. These models were later used by other labs also in the context of Alzheimer's disease, as well as also parasite deficient mice, which are models for leaky blood-brain barrier. When we started this work, no cell type had been identified in the brain that is affected by fibrinogen. So we asked what are the cellular mechanisms that are activated by this molecule in the brain. We identified the microglia cell as a central key cell target for fibrin, and we showed that fibrin activates both pro-inflammatory processes in microglia as well as pro-oxidant processes by activating NADPH oxidase and inducing release of reactive oxygen species, leading to a deleterious environment that inhibits repair, is promoting demyelination and neurodegeneration. And we show this in models of multiple sclerosis, Alzheimer's disease, as well as also brain injury. We became really interested in this link between vascular pathology, oxidative stress in neurodegeneration. And we first explored and asked the question, what is the link between fibrinogen and neurodegeneration at the level of neurons and synapses? And to address this question, we took a multi-pronged approach by combining in vivo to photon imaging with fibrin, three-dimensional fibrin immunolabeling in cleared human and mouse brain tissues, as well as cognitive and behavioral tests in animal models of Alzheimer's disease. And what we found was that an unexpected mechanism that fibrinogen can induce microglia-mediated spine elimination and cognitive impairment in Alzheimer's disease context. Here you can see baseline imaging of microglia shown here in red, spines in green, and vasculature in the normal brain. And you can appreciate these very close interactions between microglia and neurons. This is baseline imaging, and we can use the vasculature as a map. And if we inject here picoliters of fibrinogen, you can see that there is a dramatic change in the vasculature where the microglia have become amoeboid, and also at the same time there is loss of dendrites, and the remaining dendrites have lost their spines, showing for the first time that even in the healthy brain, delivering fibrinogen can cause neurodegenerative processes. We wanted to explore this also in a physiological model of neurodegeneration, and we selected the 5-XFAD animal model of Alzheimer's disease. We labeled endogenous fibrinogen, you can see it here in red, and amyloid plaques in blue. And we found this dystrophy around the amyloid plaques, which is a result that has been shown by many labs. But what was really interesting is that even in areas where there was only fibrin deposits alone, and you can see there these red fibrin deposits, these were also areas of dystrophy of neurites and synapse loss. So we decided to look into this further, and we performed longitudinal in vivo to photon imaging in these mice. You can see here day 0, these are 6-month-old 5-XFAD mice. We endogenously label fibrinogen, plaques, and neurites. And by using the vasculature as a map, we can do repetitive to photon imaging, so we can go back to the exact same area and see what these spines look like 2 weeks later after this labeling of these different deposits. And you can see in the second panel that there is significant spine loss around the amyloid plaques. But what was interesting is that also around the fibrin plaques on the upper panel, similarly, there is loss of spines over a 14-day period, even at areas that were not proximal to amyloid plaques. And as you can see at the bottom, at areas that there are no deposits, neither amyloid nor fibrin, the spines are pretty stable. Suggesting that these fibrin deposits are sites of spinal elimination in these mice. So this introduced a new model of fibrinogen as being a new culprit of spinal elimination and cognitive decline at sites of vascular damage. It identified fibrin as an apical signal that can induce oxidative stress and neurotoxic immune responses at the neurovascular interface. And it also showed that vascular pathology and oxidative stress could be a potential independent mechanism that can drive neurodegeneration and cognitive decline in the context of Alzheimer's disease. It's important to note that when we superimposed the genetic mutation to the 5-XFAD mouse, we had a significant protection in the number of synapses, but also increased cognition in these mice in behavioral tests. And these studies in mice could link with what there is emerging evidence about the importance of vascular pathology in Alzheimer's disease. Indeed, evidence from Alzheimer's patients, elderly individuals, and also APOE4 carriers, which is the leading genetic risk factor in AD, have identified that vascular pathology is an early, independent, and additive predictor of cognitive decline, also associated with high fibrinogen levels in the APOE4 mutants of Alzheimer's disease. And this has raised the question that it's possible that we need a new way to target these vascular processes in the Alzheimer's disease brain. So with this evidence from the biology, do we have a tool to be able to see whether we can target these upstream effects of vasculature in Alzheimer's disease? And of course, these are very complex pathologies. So one of the questions that always comes up is it enough to target only one protein at the neurovascular interface to be able to have a protective effect? And it was very inspiring to me that this is a question that actually has come up before in biology. This is a quote from the autobiography of Jan Wilczek. Jan Wilczek is the inventor of Remicade, the first anti-TNF therapeutic. And in the early 90s, Dr. Wilczek says that to most scientists and physicians, it seemed inconceivable that blocking a single cytokine could be beneficial for rheumatoid arthritis patients, when it was known that multiple cytokines are involved in the inflammatory process. But it appears when a molecule is really an important amplifier of the disease process, it can have important therapeutic benefit. So we asked the question, is it possible if we block fibrinogen with a pharmacologic tool, is it possible to have a therapeutic benefit in neurological diseases? And to be able to ask this question, we first had to develop a tool to efficiently and safely block this molecule. So we developed the first in its class fibrin targeting immunotherapy. And the challenge that we had to develop this was to be able to dissociate the protective role of this molecule in hemostasis from its damaging role in inflammation. And this was a large collaboration with three academic institutions and also pharma. And the way we overcame this challenge was to develop an epitope-specific antibody that targets only the critic inflammatory fibrin domain that is exposed only on the pathogenic fibrin form, but not on the soluble fibrinogen form that is circulating in the blood. The antibody targets selectively the inflammatory functions of fibrin without adverse anticoagulant effects. And it has a therapeutic administration in 4 different animal models of MS and also Alzheimer's disease, has shown protection from disability, demyelination, inflammatory processes, protection from neurodegeneration, and also suppression of inflammatory gene pathways that are linked with the neurodegenerative process. This study was selected to be featured in the 20th anniversary issue of Nature Immunology, where we had a chance to describe the steps that it took to go from basic science discovery to the development of a therapeutic and proposed that potentially fibrin targeting immunotherapy could be one of these upstream therapies to block vascular-induced inflammation. During the course of this work, we also became aware that this immunotherapy was also really efficient to suppress oxidative stress processes in the CNS. And this link between fibrin and vascular disruption with oxidative stress and neurodegeneration, we realized that this was actually not very well characterized, even though oxidative stress is such an important and critical mediator in neurological diseases. We didn't understand about the molecular mechanisms that mediate oxidative stress in neurologic disease. So we decided to take an unbiased approach to be able to understand the mechanisms that link innate immunity, oxidative stress, and neurodegeneration. And the questions we asked were, what is a transcriptomic landscape of oxidative stress producing innate immune cells? What are the signals that can polarize these innate immune cells towards oxidative stress production? And can we identify drugs to selectively target neurotoxic innate immunity? To address these questions, we established a blood-brain barrier and immunity multiomics analysis pipeline, and by combining single-cell transcriptomics, phosphoproteomics, as well as computational data integration and validation in animal models. These studies were led by Andrew Mendiola and Jake Curio in the lab, and with a fantastic group of collaborators with expertise in transcriptomics, drug discovery, as well as clinical research. We developed a functional transcriptomic analysis integrated with drug discovery. And the outcome of this work was the oxidative stress innate immune cell atlas. And we combined this with both human signatures as well as high throughput screens to be able to discover innate immune inhibitor neuroprotective strategies. The key aspect of this work was the development of TOC-seq, which is a combination of reactive oxygen species labeling and single-cell RNA-seq transcriptomics to be able to selectively identify the molecular map of oxidative stress production. In this case, we performed it in models of MS, but of course this platform can be used across the board of any neurologic disease. This study is open access. All the information is deposited in open access databases, either in Shiny, Wiki Pathways, as well as on the website of Nature Immunology. So using TOC-seq, we analyzed the transcriptomes of CD11b positive cells, and with unsupervised analysis,
here you can see distinct populations between the MS-EAE model and healthy tissue. And when we superimpose the reactive oxygen secreting cells, you can see that these are strikingly distinct ROS populations. By using canonical signatures and published markers, together with functional enrichment analysis, we identified that there were very specific populations of monocytes and macrophages, and one specific population of microglia that were sharing a distinct core oxidative stress signature associated with specific genes that can drive oxidative stress in these populations. We performed subcluster analysis, and subcluster analysis identified one specific microglia population, MG5, and one macrophage population, MP1, as being enriched in reactive oxygen species metabolic process. In contrast, other populations like MG3 and MP3 were enriched for antigen processing and presentation. We overlaid on the subclusters either oxidative stress genes like CyBB, and this is a gene encoding NADPH oxidase, one of the key subunits of NADPH expressed in multiple sclerosis, that also, if it's genetically depleted, it protects from MS and Alzheimer's disease. And we also used MHC class 2 to look for antigen-presenting genes. And here you can see that the MG5 population is highly expressing the NADPH oxidase gene, while the other populations like MG3 is expressing almost exclusively the antigen-presenting MHC class 2 gene. And these results showed for the first time that there is a functional heterogeneity in innate immunity in relation to oxidative stress, and there's a differential contribution of CNS innate immune clusters to oxidative stress and antigen presentation. And indeed, when we superimpose these signatures to human multiple sclerosis, the population MG5 exclusively expressed genes associated with glutathione pathway, oxidative stress, as well as iron uptake, and not more generic inflammatory genes that are identified in the lesions. The transcriptome signature of the MG5 population showed the highest expression of oxidative stress genes, and also pattern recognition markers like CLEG4, as well as oxidative stress and inflammatory markers. What was really intriguing is that we found that MG5 also expressed coagulation markers, and this is expression of factor 10, showing that oxidative stress activation could be also mechanistically linked to production of coagulation of genes that promote the coagulation cascade. We independently validated the signatures by doing bulk seq on FACT-sorted ROS and MHC class 2 positive cells. And here again, you can see that the ROS positive microglia were enriched in pathways of blood coagulation, phosphorylation, lipid biosynthesis, and had a decrease of antigen-presenting pathways in generic inflammatory responses and leukocyte activation. Since these studies identified this mechanistic link between oxidative stress and coagulation, we developed a high-throughput, high-content small molecule screen to activate microglia cells with fibrin, the final product of the coagulation cascade, and we screened 1,907 clinical drugs and bioactive compounds. From this screen, we identified 128 small molecules that could suppress either LPS or fibrin-induced activation of the microglia. And we developed microglia drug networks by using KEGG analysis. And we then overlaid the drug networks with the toxic data. And this led us to oxidative stress maps, where we could identify both the regulation of genes in the different oxidative stress populations, but also which of those had drugs that could be that were identified in the high throughput screen as hits for this pathway. This analysis revealed the GGT pathway and the hit acidicine as one of the key leads of this molecule, with the potential to suppress these oxidative stress responses. GGT is the gamma-glutamic transferase. It is an ectoenzyme that integrates glutathione and contributes to reactive oxygen species generation. There is extensive work for GGT in cancer, but it had not been studied before in the context of neurologic diseases. So we checked if it's even expressed in the neuroinflammation model. And as you can see here, there was abundant expression of GGT in the AE model of MS. And the GGT activity was also increased, and the drug activity that we had found in the HIT had the efficacy to actually suppress the activity of this enzyme in vivo. We tested its therapeutic efficacy, and here you can see in a model of chronic EAE, chronic multiple sclerosis model, we dosed the mice 80 days after onset of pathology. So even after such a late therapeutic administration, the drug is still sufficient to suppress paralysis in these mice, and also suppress NADPH oxidase, protect from demyelination, and also protect from axonal damage. Suggesting that, showing that suppressing this downstream mediator of oxidative stress that is induced by the fibrin pathway can also have a therapeutic effect. So these findings change the way we think about the interplay between neurovascular damage and oxidative stress in neurological diseases by identifying the microglia population MG5 as the oxidative stress innate immune population in the CNS, and showing that this population, in addition to mediating oxidative stress, is actually secreting procoagulant factors that can start this vicious loop, this positive feedback loop, to promote fibrin formation and therefore activate more oxidative stress processes in the brain. We also identify a new pathway, GGT, as a key molecule involved in oxidative stress induction in the CNS. And now we have multiple ways to target this, both upstream by blocking the interaction of fibrin with the microglia cells, but also downstream by blocking a selective intracellular pathway that can activate oxidative stress responses. And combined, these approaches could have been tested in multiple sclerosis, but I think could also be relevant for other neurological diseases. So I have spent this talk discussing with you the inflammatory functions of this pathway, and I want to highlight that 3 years ago we also published that fibrin is also inhibiting regeneration in the brain by suppressing remyelination in the CNS. And this study, the take-home message from this study was very nicely featured in the News and Views article, that it was described as the bloody break on myelin repair. And the take-home message I want to leave you today is that the mechanism of action of these pathways in the CNS is to be both a bloody break on repair, but also a gas pedal for toxic inflammation. And potentially this can open the path for a new way of thinking how to target neurovascular pathology in neurological diseases. I would like to thank the very talented people in my lab who have been involved in this work. The antibody was studies were headed by J.Q. Ryu and Takame Franke, and the toxic was developed by Andrew Mendiola and Mark Peterson, and Resmi Dognata are working on the remyelination aspects in the lab. I would like to thank our fantastic collaborators over the years that have supported these studies. All the past trainees, in particular Mario Merlini, who performed the, I showed data from Mario Merlini today, who performed the spinal elimination study, and he just started his lab this month at the University of Cannes. And finally, I would like to thank all our funding over the years for all the support to the lab. And thank you so much for your attention.
Thank you very much, Professor Akasoglu. Are there any questions for the speaker, please? Okay, I don't see any questions on the chat. We do have 1 minute. Are there any questions, comments from participants? Okay, I don't see any questions. So let us thank the speaker again and continue on with the program right on time. Thank you for keeping the time as well. By the way, I want to apologize for skipping the last 10 minutes of the pre-recorded video from Dr. Plakiotis' talk. Inadvertently, his video was 40 minutes. So whoever is interested, please contact this speaker individually. So we will continue on with the third keynote speaker, who is Professor Claude Frasson from Montreal. His talk is entitled Virtual Reality Intelligent Systems for Alzheimer.
Yes,
Professor, allez-y.
Thank you. Do you see the screen?
Yes, yes, thank you.
As Katerina did, I would like to thank the organizer. You are doing a fantastic job. I'm following the talk in parallel with another conference we organize also on brain function in Athens, and your talk are fantastic. I will talk today about virtual reality intelligent system for Alzheimer. In fact, there is an impact of negative emotion on the brain, particularly the frustration, the depression, and most of all, the empathy. They have an impact on certain regions of the brain and mostly in the limbic system, where stays the memory and the cognitive function. So there is an alteration by negative emotion. And for Alzheimer's disease, it's particularly important because the circuits are not installed, and this represents a problem. So the origin of Alzheimer's disease is in the brain and the neuron region. It's a deterioration of both short-term and long-term memory. And the disease affects also behavior, cognitive abilities, and also physical abilities. So presently there is no drugs, no treatment efficient for Alzheimer, and there is a shift to non-pharmacological treatment, and among them for virtual reality. Virtual reality was used, it was proven to be efficient for fear or octopus, for fear of flight, fear of maybe COVID-19. This is a reality, and it is an efficient treatment. So VR intervention for mild cognitive impairment seems to be positive, according to recent studies. And as I said, VR has been applied in the field of psychology to treat various disorders. And the most important thing is that the impact is on the right brain, mostly on the right brain of a patient in the subconscious part, subconscious brain. And we have designed in our laboratory an intelligent virtual reality platform with several environments. First, a virtual train in which the patient is able to travel and to relax, and music therapy in which the patient is able to hear some music, some songs, a zootherapy in which the patient is able to meet with animals, for example, horses, and also a space therapy where the patient is in a space station in space and able to try to catch some object. So it's a question of equilibrium in a space. But the most important thing is the contribution of artificial intelligence to this environment. In fact, we use sensors to catch the EEG signals But we also have an intelligent neurofeedback approach. The signals comes from EEG and has an impact on the virtual reality environment because an agent will modify the environment accordingly, according to the evolution of EEG. For instance, if a patient has a decrease of negative emotion, so this means that the treatment is good and can be reinforced. You know? Otherwise, if a patient does not like, so the environment moves differently. So this is an intelligent neurofeedback approach. This is the environment in which this is not a patient, this is one of my students. Wearing a headset. You see here the headset, the light headset, which is built by Emotive, and a VR headset in which we can see, in fact, you see what the patient is seeing. And for this headset, we have, in addition, eye tracking included in the VR headset. So this is very important, because if you ask a patient, Alzheimer patient, for some question, and you say, look at this picture on the right, and the patient is looking on the left, you detect that he is looking on the left. So it is not because he don't recognize the image, it's because he's not looking at the right direction. So it's important to detect and not to have false interpretation. And we have here the environment. You see two screens because it's for each eye. The measures are built by Emotive headset, Epoch headset, with 14 electrodes on the front, on the temporal, on parietal, occipital, on the left and on the right part of the brain, according to the well-known international 1020 system. And Emotive generate raw data at a sample of 120 hertz. What is important and useful is that the reaction of the brain is quite immediate, and you detect the reaction of the brain after 1 second. So you, in real time, you can have a measure signal, but of course you have to build the tools to interpret the signals, and build the tool to have a real-time interpretation and measure of the emotion. And particularly, we target negative emotion. So this is the process of the experiment. First, the patient fills a precision form without any equipment. After that, we install the Emotive Epoch headset on his head. Then we submit the patient to cognitive and memory exercise in order to know what he is doing, what are his performances without the intervention of VR. Then we store the result. Then we install the VR headset and we display the virtual environment. Here, it will be the virtual train, for instance. And remember that in parallel, we measure with the EEG system. So we detect all the signals from the learner. After that, it lasts about 10 minutes. After that, we remove the VR headset, and again, the patient is submitted to cognitive and memory exercise. Then he fills a post-session form. So this is a general process, able to compare what was before and what was after. For instance, memory exercise, this is an example of memory exercise. We ask the patient to memorize a series of objects presented visually or orally. Then there are presented a series of object image or words presented auditorily, and participants are asked to determine whether the object was previously seen visually, auditorily, or never presented. So it's an exercise of memorization. A cognitive exercise, we show images of different objects for a short time of period, and the image is then replaced by a series of 4 letters, and the participant is asked to select the first letter of the object. For instance, if I ask you about E, B, D, C, and I present a COVID-19 image, so you remember that it was C, which was presented. Sorry for the, the example. So, so for instance, a chair, and of course, the patient should click on the C. Let us see now a demonstration of virtual train. I hope you will hear. Do you hear? So this is the virtual train. And on the right side, you see a in real time measure of frustration. Frustration is decreasing. And valence, which means interest, motivation, is sometimes increasing. This is made in real time, taken from EEG measure. So this is, of course, the environment in Canada, what we know in general. But you see that the frustration has decreased because the patient is relaxed. So you see, so we analyze after that the evolution of frustration before and during and after the virtual train. And we saw a decrease of the frustration during the train. And after that, small increase, but the medium measure was also reduced frustration. So this means that the impact stays after the experience. So this effect is observed after the training. We analyze also the performance improvement on the first 3 exercises, and according to the measure, so we saw sometimes improvement in exercise and sometimes decreased performance. Sometimes for certain exercises improvement, and with some others, sometimes improvement, or sometimes the same measure. For memory exercise, we definitively, in memory, you see for the different exercise, we see an increase, you see an increase of improvement for quite all the exercise, except for some of them. But in general, it was an increase of 10–20% of improvement. For cognitive exercise and memory exercise, if we compare, we have also a clear difference between improvement of performance in memory exercise, you know, in red, memory exercise, versus cognitive exercise, where the improvement is small or sometimes not in the good direction. Now I would like to present another environment with music therapy, which is another capability to reach the subconscious brain of a learner.
trees of green, red roses too. I see them bloom for me and you.
So it seems that Armstrong has a good impact. A good impact on the reduction of negative emotion in general. So the results show that negative emotions like frustration are reduced, clearly reduced, and also a clear increase in memory performance following music therapy, and some improvement in some attention abilities. So we have studied with several of my students, master and PhD students, the involvement of brain reward system, which is at the basis of dopamine generation. And we saw that the brain reward system is at the center of this improvement by music. So I show you a recent paper published in the Journal of Biomedical Science and Engineering by my team. It's on the contribution of music therapy and virtual reality for improvement of cognition. And the brain reward system regions are in fact activated by music, increasing pleasure and reducing negative emotion. So the rewarding stimuli reduced the cortisol, which is a so-called stress hormone, and reduce the negative emotion to have a better problem-solving performance, inhibiting the negative emotion. And music activates the BRS through interaction between cortical networks related to audition and to reward. Now, I would like to introduce the contribution of animals for reducing negative emotions. So it exists presently in some hospital, the introduction of animal from pets, some birds, cats, and even horses for therapy animal. This seems to be positive for the patient who feel more relaxed and have more empathy, which is the most important default of emotion. So the problem is that this is expensive to care. Introducing horses in a hospital requires disinfection of a hospital, of a horse, and before and after. And so it's really a strong experiment requiring a lot of precaution. It can provoke zoonotic infection sometimes, and sometimes with the proximity of animals to the patient, we could have some animal bites, which is not recommended. So this is why we have looked at the virtual reality contribution, existing therapy with virtual reality, for instance, for treating the fear of height, not for Alzheimer's patient, but for the fear of height exists. For instance, trying to save a cat from a tree. It's a motivation to save a cat against the fear of elevation. So in the same direction, we have built animal-assisted therapy as a stimulation of psychosocial treatment between human and animals. And we have seen that animal therapy can increase the social interaction, decreasing the agitated behaviors of patients with Alzheimer's disease. They have a calming effect. So we have decided to use virtual reality in order to create a virtual animal-assisted therapy, but to increase the communication between the patient and the animal. Mostly, some patients are sometimes unable even to speak or to interact. So the only interaction is by gesture. So we have built a gesture recognition system able to recognize simple gesture from the patient. And so we have built this system with horses and submitted to the patient, where we measure the EEG, and we have also contribution of gesture recognition. Then we have an intelligent agent which is able to compute the signal from EEG and from gesture recognition. For instance, if the signal from EEG means that the frustration is in reduction, and the patient says, no, go away, go away. So it's in contradiction with what the patient is feeling, the result of the interaction. And so the agent will take into consideration the EEG signals, And according to the signals, the intelligent agent moves the horses or change the environment in order to optimize. So this is a real neurofeedback intelligence system. So we have created EEG Unity 3D in order to create domestic animals which react to patient with realistic sound. So we hope realistic sound. And the animals moves according to a dynamic path. So to recognize the gesture, instead of having computational effort using complex recognition system, we have used a depth camera able to recognize gesture. For instance, Intel RealSense depth camera, which provoke a skeleton of the movement of a body. And also lip motion camera, able to detect the gesture of a hand and making also a skeleton of a gesture. So we have experimented that for simple, for instance, left hand comb, which make the animal come near the user, and right hand comb, so simple gesture. The intelligent agent tracks the emotion and the gesture of the patient. So he intervenes in the environment in order to command the animals, taking into account this balance of EEG signals and what the patient wants to say. And sometimes when we recognize the movement of a patient, we see an impact on the EEG improvement. So we use a rule-based system in order to adapt the environment to the participant. And for instance, if the frustration of a participant increases when the animal approaches them, the agent makes the animal go away. So we combine hand gesture and emotion as an input in order to decide of an action. So we have made experiment on that. So now I would like to present you the system. We see two screens because it's what each eyes see. So it's a real immersive system. These are the hands of a patient. The patient selects a dog. So now the patient say, come in. So now the patient goes away. So now, if we can see, now the patient has activated a call for the horses. So this is an example of experiment with this environment. And so in conclusion, so we have presented a series of virtual environment with progressive intelligence. In virtual train, there is no too much intelligence. In music therapy, the system, the agent is able to detect which music provoke the most important decrease of negative emotion. And after that, the system will present this kind of music, for instance, Armstrong songs. And with zootherapy, there is a real neurofeedback intelligence system, as the agent is able to change not only the environment, but also the move of the animals into a virtual environment. So this is what we say at the center of this system. We have a brain. The brain is, in fact, the component of artificial intelligence. The brain is a real, it's not artificial, but it's a real AI and live component of intelligence, because we use what the learner, what the patient feels to improve the environment and the change of negative emotion. So this is the intelligent layer, and we have other system under development, the space therapy. We have also savanna therapy, in which the patient is able to travel into a savanna environment, and also underwater. The patient is able to go down in the water and see the different fishes. So a lot of environment driven by intelligent layer in virtual reality. Thank you very much.
Thank you very much, Professor Frasson. Merci beaucoup de votre exposé. Are there any questions, please, for Professor Frasson? Thank you for keeping the time as well. Yes. May I ask the technical staff to notify me if there are any questions, please, for the speaker? Okay, it looks like there are no questions, so thank you again very much.
Thank you.
Let's move on with last but not least, obviously, the keynote address by Dr. Ioannis Tarnanas from, he is the Chief Scientific Officer at Altoida and also affiliated with Texas Medical Center in Houston. His talk today is on digital biomarkers data from Alzheimer's clinical trials and the neuromotor index. Please go ahead.
Greetings. Greetings to the conference of GeNeDis. I'm going to present today our work with ADDF, the Alzheimer's Disease Discovery Fund, about a digitally enhanced personalized medicine platform for the accurate selection of subjective cognitive decline and measuring progression of mild cognitive impairment. The collaborators for this project is myself, Ioannis Tarnanas, PhD. I'm the Chief Scientific Officer in Altoida. José Luis Molenuevo, he's the PI and Director of the Barcelona Brain Research Center. And Mircea Balasa, he's a Director of EDPAPS, a Barcelona Clinic for Mild Cognitive Impairment and Dementia. We all know the background of Alzheimer's. We all know how terrible disease it is. It's the global, is one of the biggest global public health challenges. 60 to 70 cases of dementia are attributable to Alzheimer's disease. And what we see here is that usually the numbers are growing, and they are growing more aggressively in the low to middle income countries. The new conceptualization of Alzheimer's disease means that we are now looking at the pathological continuum. There are two stages that are considered to be preclinical, and one of the starting stages is basically normal, no symptoms at all, no amyloid in the brain. There is a stage that is considered to be a preclinical stage for AD, where you do see some amyloid pathology, but there are no symptoms. And when you are discussing clinical stages, then you are talking about the MCI, mild cognitive impairment due to AD, and of course dementia due to AD with moderate pathology and intense pathology, respectively, along the pathological continuum. The population that we target here with our platform for precision medicine is subjective cognitive decline and mild cognitive impairment. Those are some of the syndromes that have proven to be very useful to identify early individuals, subjects that are in the preclinical stages. And subjective cognitive decline, or SCD, is individuals that do have some complaints or some subjective perception of cognitive decline, but they don't show any other cognitive deficits in a formal neuropsychological assessment. On the contrary, mild cognitive impaired individuals, they do have a single or multi-domain cognitive deficit, and usually they preserve activities of daily living. What is the unmet need here is how can we develop sensitive measures for the early identification of those SCD and MCI individuals without the need of using invasive biomarkers, and also how to track disease progression during the pre-dementia stages in clinical practice and trials with measures that are sensitive enough to capture subtle changes. What we know today about the digital biomarkers in Alzheimer's, the diagnostic biomarkers in Alzheimer's, is that we do have a golden standard, and this is PET and CSF. Both are not necessarily suited for screening because they are both invasive and expensive. And you see here from the images, what do we mean by invasive? A novel biomarker, on the other hand, would be sensitive, non-invasive, patient-friendly, would allow for repetitive measurements, would be easy to work with. And there is some research in the area that indicates Such biomarkers could be digital devices that are enabling the collection of continuous, objective, and multidimensional data during daily activities. Such devices can capture subtle changes in cognition and functional capacity long before the onset of cognitive decline that is currently captured by existing paper and pencil neuropsychological tests. Such a tool is Altoida. So what is Altoida? Altoida is a risk assessment and mitigation tool. We have developed a term called Altoida Neuromotor Index. It's a platform that runs at a simple tablet and smartphone. It's an augmented reality technology that tests functional, motor, cognitive, and behavioral capabilities, and it's using machine learning for the scoring of your performance. Based on clinical trials that I will speak about in my presentation today, we have clinically validated a 94% accuracy rate at identifying individuals at risk of developing MCI due to AD at the prodromal stage. So we're talking about prognosis here. We have minimal practice effects. The test-retest variability in healthy controls are 0.158%. And this allows very precise patient monitoring and measuring true performance. On top of that, we are FDA cleared as a software, as a medical device. And right now, it's the fastest, most non-invasive and most scalable tool that you can use for screening in a medical environment or a clinical trial. The actual device consists of 2 motor tests and 2 voice tests, accompanied by 2 augmented reality tasks, and they are generating 1,000 features and fitting into 11 cognitive and functional domains that are generating a user-friendly report. I'm going to explain to you more what this means in the following slides. How do we collect 700 plus features? So while we are collecting traditional parameters such as reaction time, speed, attention, and memory while you are performing the virtual reality tasks, those conventional parameters are usually based on correct or incorrect responses and usually are capturing either a yes or no or a checkbox during this task execution. On the contrary, we are doing something more besides collecting those yes or no responses. We are collecting every input that you are giving our device, you are giving out sensors while you are holding it. So during the entire assessment, either while you are responding to stimuli or when you are not responding to stimuli, we are continuously collecting data from the accelerometer, gyroscope, magnetoscope, camera, microphone, and touchscreen of your iPad or of your smartphone. So we can assess things like tremor, gait, visual search patterns in real life, any hesitation, any directions, any change of directions, and the actual paths while you are hiding or discovering hidden objects. So in essence, it's the same process, but we are collecting much more information than just responses on a piece of paper. And in that way, within 10 minutes, we are able to collect 1,000 plus individual parameters, which are feeding our algorithms, our machine learning models. And we have developed a concept called micro errors for accuracy and micro movements for latency that are collected during the augmented reality task. If you want to see some of those motor tests, how do they look like? It's some simple exercises like the ones that you see on your left and on your right. So you need to use your finger to complete the cycle as fast as possible and as accurate as possible. And at the same time, next, the Serpentine test. And it's not just how accurately you complete the cycle or how accurately you complete the Serpentine test from start to finish, but it's also how are you holding the device? Are you tilting the device? Are you pressing the device hard? Are you pressing the device soft? Are you inside the cycle? Are you outside the cycle? So among those 1,000 parameters that we are collecting, is things that we are responding to and things that we are not responding to during the execution of your task. At the same time, we are having some classical motor processing speed and reaction time tasks. And at the same time, we are capturing your eyes, your gaze, while you're executing those tasks. Those are just two of the examples of our motor test. The actual motor test includes 11 exercises in total. The augmented reality test, which is following the execution of the motor test, might be a little bit more interesting for clinicians and for researchers to observe. It's actually an execution in real life of two very simple tests, two very simple exercises. One is forcing you to hide 3 items in real space and then find them back. So a very realistic representation of an everyday activity, just like losing one's key and finding them at the house. And this particular test is called BEAT, back in time, which besides controlling for how well are you finding the actual item, it also controls for things like gait, balance, visual spatial memory, orientation, planning, executive functioning, dual tasking based on some stimuli that are obscuring you during execution, working memory, and the micro errors, which, as I promised, I will explain later on. At the same test, or at the second part of the test, you are being asked to remember prospectively some items that you are asked to place in real space, in your real office, clinical office, or in your real house, and then find them back. And this is a prospective memory task that resembles the items that you go to buy at the supermarket. So again, there is a lot of ecological validity in the way that you are performing those 2 exercises. Same as before, besides measuring how well are you hiding and finding the objects and at what order, we are also measuring, like before, lots of other different parameters like gait, balance, orientation. And on top of what I said before, it's also the prospective memory aspect that we are measuring in particular, or focusing more in this particular task. Now, we've spoken about the micro errors, and micro errors is an Altoida patent. What micro errors means is that while you are walking in real space holding your iPad or your smartphone, the camera at the back of your smartphone is at the same time mapping the environment around you. So we get a very clear indication, a very objective way of knowing exactly where in real space you have placed your items that you're supposed to be finding next, and exactly how are you working around this space. Are you actually close to those objects? Are you far away? Taking into account, of course, during the mapping, any obstacles in the real space or any paths that are hidden or not. So it's basically a very accurate 3D picture of the environment around you. And this is feeding to our algorithms not only accelerometer data regarding your speed, but also accuracy data, and also the ratio between acceleration and latency, which has proven, based on previous research, to be a very sensitive marker of predicting individuals that are developing Alzheimer's in the future and the ones that are normal. And this has been published before. In a little bit more detail, if you look at the micro scale, because before it was the macro scale, NMI, the Altoida Neuromotor Index, is creating 3D trajectories in space during the item find. And you can see here 2 very distinctive profiles, one for the healthy, one for the individuals at risk. For the healthy individuals, when you examine the micro scale, you see that the range of motions when you are trying to find an item that you have previously hidden is very condensed and very close together to the actual position of the item that you successfully found, basically. If you take an individual at risk, then you observe a different pattern. And this pattern is starting around the area that is the actual hidden area, and it has a specific pattern as to how you are starting to search around there. So as you can see from the example here, you start from the left to the right, and this individual has 3 steps that is basically looking for around the area of the target. Other individuals have 4, other individuals have 2. So this creates a very specific signature or a very specific, let's say, fingerprint of your behavior, which is what we call microerrors. And it's a unique feature that we have patented. And it's a combination of perceptual motor coordination along with eye movements along this process. And it can be a very sensitive metric for accuracy when monitoring patients at a later stage. As a demonstration of the power of those microerrors and the previous measures that I mentioned, we have published results, which I will describe later at our validation studies. And we have done a clinical trial for 10 years in total, 8 years of original data collection and 4 years of validation. And in both those cases, we were demonstrating an area under the curve of 94% to be able to differentiate healthy controls with individuals that MCI due to AD from a prodromal stage, basically prognosing individuals when there was no other clinical symptom or cognitive symptom evident in a neuropsychological assessment. When you go to the MCI stage, we can differentiate between the amyloid positive and the amyloid negative individuals with an area under the curve of 91%. Those are the published results. I mean, along the published study, which it became published at August this year. I mean, the study, the clinical trials ended at January this year, and the results appeared in the journals at August. But between those 6 months, from January to August, we increased the accuracy to 96% based on additional data coming from our clinical trials. So the algorithm is evolving. The machine learning is actually learning how to differentiate more. But I'm showing here for transparency the published results. Don't take my word for the nuance. Just put a little bit of salt on top of that. If you combine NMI, our own metrics, with genotyping as well, so with APOE 4.4 status, you get an almost 100% accuracy in predicting individuals that will transition to MCI due to AD in the next 3 to 4 years from measurement. So the accuracy here is quite substantial, and the tool has already attracted some attention, which is why we are getting now the support from the Alzheimer's Disease Discovery Fund to further validate our tool to an earlier population than what we do now, essentially SSD population. A little bit about the clinical trials that we have used to get our data and to analyze our results. The first and the original study was an IMI grant called Pharmacog. It was a large collaboration which, as we can see, included most of the major pharma companies in Europe, together with some major academic institutions. We managed to link our tool with brain network dysfunction, among other biomarkers, in MCI due to AD. And we have shown by our data collection that hippocampal overactivity can be the best predictor of subsequent cognitive decline and conversion to Alzheimer's disease dementia. In our case, we managed to be the first in the world mapping of a translation of brain network dysfunction to everyday function, using our tool that assesses continuously motor coordination, planning, processing speed, inhibition, visual perception, so on and so forth. At the time, we had 774 features, but again, 11 domains mapped, 11 cognitive domains mapped in our reports. And consistent accuracy of 94% area under the curve for classifying individuals that are healthy and individuals that are going to be progressing to AD during the trial duration of 8 years. The validation study was done recently. So using the original study as a database for training, we trained our models, and then we applied our models to a new dataset, to a new real-world dataset. It was a prospective multi-center study performed in both memory clinics and primary care centers. The total population was 548 cognitively normal MCI and early dementia subjects. And 10 of the centers were at Europe and 2 primary care centers in the US, so quite a multinational representation in this validation study. The funding was given to us by EIT Health, Alzheimer's Society US, Takeda Pharmaceuticals, and the grant from the EIT Health was $806,999 for the years 2015 till 2019. The same tool, so we didn't change the tool significantly from the original study because we wanted to validate the models. And this is what gave the chance to the Alzheimer's discovery, this Alzheimer's Disease or Discovery Fund to adopt our tool and together with the Barcelona Brain Research Center, see if we can apply the same technology and the same performance into a better phenotype population that would be consisting mostly of subjective cognitive decline individuals that are very well characterized in the Barcelona beta cohort. What are the objectives here? The objectives are to test the accuracy of the Altoida NMI to identify patients with SCD or MCI that actually have underlying AD pathology. And the second objective is to test the capacity of the Altoida NMI to track disease progression in such patients. And we also have a couple of secondary objectives, which is basically correlate NMI with MRI measures as a function of AD pathology. Also study the impact of markers of neuronal death with plasma neurofilaments in both study groups against Altoida NMI performance. To explore the potential diagnostic value and accuracy of this particular metric that we have developed, NMI, but also other metrics from Altoida in their efficacy to detect ADI pathology in patients with SCD through complex machine learning. So a little bit of R&D there. And also track disease progressions in such patients. And of course, as a final goal, in order to test the applicability of the tool in real life practice, is to determine the ease of use and satisfaction of the Altoida system by its users, and also define the cost benefit of triaging patients through the Altoida system. So there is a very clear clinical and also commercial objective using these data collections that we have planned as the third major clinical trial that we are doing with our Altoida tool together with some very high reputable institutions like the Barcelona Brain Research Center and the EDPAPS. The study population, the one from the Barcelona Brain Research Center, is the so-called Barcelona beta cohort. They are 200 individuals, 60 years of age till 80 years of age. Most of them referred from primary care centers. And they do experience a decline in their cognitive abilities, majority SCD and some MCI as well. This is year one. We're going to collect clinical history, cognition, APOE genotyping, MRIs, environmental and lifestyle factors, CSF and blood sample, and of course the Altoida device. In year two, we are doing something similar, but we are only collecting clinical history and cognition, environmental and lifestyle factors, blood sampling, and the Altoida device. On top of those 200, there are another 50 individuals that are MCI. They are recruited through an outpatient clinic from the EDPAP's Hospital Clinic Barcelona cohort. And besides the clinical history and cognition and APOE genotyping, we want to know AD biomarker status, for year 1, so that we can identify MCI due to AD versus non-AD MCI. And there is also a regular follow-up for the next couple of years. It's going to go until year 5, with data collection of clinical history, cognition, environmental and lifestyle factors from these particular individuals that are the MCIs due to AD or non-AD. In our statistical analysis from those two groups, we obviously want to create predictive models. So we want to see some differences between the groups. For example, amyloid positive versus negative, SSD, or MCI, or stable versus progressive SSD or MCI that will be calculated, and the model will be created to test how well are we predicting or prognosing those two groups. I would like to add here that besides stable or progressive, we can also assess speed of progression using some additional matrices from NMI. And of course, we're going to do a logistic regression analysis for a model that can effectively separate SED and MCI subjects according to the amyloid status. Goodness of the LRI model will be assessed. Positive predictive value, negative predictive value, area under the curve will be constructed. The area under the curve of the rocks with 90% confidence interval will be calculated as well. And all of that will be done with SPSS and correlated among the two cohorts with other data that we are collecting here, like demographics, cognitive assessments, core AD CSF biomarkers, APOE data, serum NFL neurofilaments, MRI data, and of course other matrices besides the NMI index itself. What we plan to analyze is how well does Altoid NMI correlates with basal cognition, also correlate basal NMI with core CSF biomarkers in both groups, evaluate the Altoida performance according to the APOE genotype based on the promising results before. We hope that we will be able to showcase a similar performance here in those two groups. The impact of serum neurofilament light will be evaluated as well in combination with the Altoida test, or maybe as a predictive model to see if Altoida test can predict the scoring of the neurofilament light in serum. We are also going to correlate NMI with gray and white matter volume using a voxel-based analysis. And finally, as I said before, not only clinical measures, but also user and applicability measures. We're going to evaluate user-perceived invasiveness of the test, as well as their willingness to repeat it. So it's going to be a really good metric of the clinical and practical efficiency of the tool on its own, as well as potentially how well can it be adopted in the primary care setting. The study timeline in a little bit more detail is consisting of some main tasks, the milestones and deliverables. You can see here that it's going to be a busy couple of years. And we plan the start of the study this month, right after the GeNeDis conference. So it's going to be a very good timing, and that's why we are demonstrating this study for the first time in this conference now. Potentially, if there are interested individuals, they can contact us later on. And it's going to be finishing 2 years from now. So apparently in October 2022, we should be able to start generating some data and running our analysis, including the longitudinal and machine learning analysis that we have in mind. What is good with this data collection is that Altoid is providing a dashboard. Altoid is providing a dashboard to track patient development, And it really is objectively collecting real-world evidence, real-world data, because as I explained before, the exercise and the tasks that are performed under the Altoida examination are actually performed in a real-world setting. It's not a computerized test that you are having in front of you while you are sitting in your office and you are just interacting with a keyboard or a smartphone or a touchscreen in your iPad. You're actually doing stuff in everyday life, and we are actually measuring everyday function in a realistic environment in a very ecologically valid way. And we are seeing the development of your trajectories in real time, depending on how frequent or how often you are taking our examination. So this is the biggest value proposition, the biggest impact that we are creating with Altoida. On top of that, those are some of the additional matrices that we have created, especially for clinical trials and for research. We are creating a halo radar plot showing how your different scores are changing during time. And this enables us to measure slight variations in subjects' performance over time and potentially assess inter-individual variability, which might be an interesting metric to predict potentially conversion effects or conversion events in a disease continuum. We do have some data to showcase here from our MCI group. I didn't have the time to present them in a structured way, but I promise you that I will present those results from this SCD group in the next couple of years. And I'll go back to you with some more performance improvements from our machine learning models, especially suited for illustrations of conversion events for the MCI group most specifically, because the SSD group can only be converting to MCI at a later stage. This is the group of people that is making the science behind Altoida. Myself, I'm the Chief Science Officer in Altoida and acting as an interim CEO for the company currently. We have a full team of people for commercialization, sales, medical executives, and innovation, which you can see here. And Maximilian Bugler is our Chief Technology Officer. He's a PhD in computational modeling and simulation, and he has over 10 years of experience in the AI. Together with Robert Harms, they are the ones who are developing our solutions and our algorithms and our models behind our prognosis. And we recently published, as I mentioned before, our most recent results at the Alzheimer's and Dementia Journal. You can look for us under my name or Maximilian Bugler's name, and you will see the published research that is demonstrating the areas under the curve that I mentioned before, and our beautiful performance in prognosing MCI due to AD from a prodromal stage, as well as dementia, of course. This was my presentation today. If you have any questions, don't forget to write me an email at the address that you are seeing here, or call me for those of you that are in the US. Our company has offices both at the US, Houston, Texas Medical Center, as well as Switzerland, Lucerne. So depending on your preference, feel free to reach out. And I thank you all, and I feel very, very grateful to be invited in this high reputation and high impact conference that is called GeNeDis. Thank you.
Okay, thank you, Dr. Tarnanas, for the talk. Once again, for participants who are interested, you are welcome to contact Dr. Tarnanas and ask questions. I would like to thank, in closing, all of the 4 speakers of this afternoon's session on neurology and artificial intelligence. Thank you for keeping the time as well. And if there are no other questions, I don't see any questions on the chat for any of the speakers. And I would also like to thank Qualisound, a company based in Athens, for the excellent technical support that they have been providing for the GeNeDis conference. And GeNeDis will reconvene tomorrow morning, 9 a.m. Athens time. Thank you everyone for watching, participating around the world, and see you tomorrow.